Bioinforange platformunun kuruluşunun 2. yıl dönümünde düzenlediğimiz Bioinfocongress IV etkinliğimize hepiniz hoş geldiniz.
Merhabalar, hoş geldiniz laboratuvarımıza.
Merhaba hocam, hoş geldiniz.
Merhaba, hoş bulduk. Sağ olun, teşekkürler davetiniz için, organizasyon için.
Davet için tekrar çok teşekkürler. Organizasyon da çok başarılı gözüküyor. Çok teşekkür ederim.
Tekrar teşekkürler davetiniz için. Bugün bilgisayar teknolojisiyle biyomoleküler sistemlerin simülasyonu ve ilaç tasarımı konusunda konuşacağım.
Muhteşem. Çok teşekkür ederim. Evet, hepinize merhabalar. Organize komitesindeki arkadaşlara da çok teşekkür ederim beni davet ettiğiniz için.
Herkese merhabalar. İlk önce davetiniz için çok teşekkür ederim. Buradaki diğer konuşmacılarla beraber sizlerle beraber olmak gerçekten gurur verici.
Bioinfocongress'te sunum yapmaktan beni davet ettiğiniz için çok memnunum ve sanıyorum tamamen insan hücre fonksiyonlarına yönelik olmayan daha basit canlılarla ilgili olan tek konuşma belki. Ama böyle bir değişiklik getirmekten de çok memnunum.
Öncelikle hepinize teşekkür etmek istiyorum. Bioinfocongress oldukça güzel bir organizasyon yaptınız.
Tüm Bioinforange çalışanlarına ya da gönüllülerine teşekkür ediyorum. Beni davet ettiğiniz, zamanınızı ayırdınız.
Merhaba, davetiniz için çok teşekkür ederim. Bu aktiviteyi organize ettiğiniz için de teşekkür ederim. Gerçekten çok büyük bir çalışma gerektiriyor.
Thank you, it's a pleasure. Thank you for the invitation.
İyi akşamlar. Başlamadan önce bu güzel organizasyonu düzenleyen arkadaşlara gerçekten teşekkür etmek istiyorum. Bu kadar büyük bir topluluğu bir araya getirmek, böyle bir organizasyon gerçekleştirmek gerçekten büyük bir başarı.
It's a really fantastic program that you've put together over these last few days. So it's been some brilliant science and some brilliant scientists speaking. So thank you very much again for the kind invitation.
Welcome everyone, I'm Neva Çalışkan. I'm a group leader at the Helmholtz Institute for RNA-based infection research. Today my talk will be about how genome, meaning of the genome can be altered by frameshift RNAs and how this response can be regulated by host and viral proteins.
Teşekkür ederim davetiniz için. Bugün mRNA aşılarından bahsedeceğim sizlere.
Merhaba arkadaşlar, bugün sizlere katılmaktan çok hoşnutum. İlk önce Bioinforange öğrencilerine bu güzel organizasyonu bir araya geldikleri için teşekkür ediyorum. Gerçekten benden önceki videoları izlemekten çok hoşlandım.
Merhaba herkese, buraya geldiğiniz için teşekkürler. Bugün ökaryotik genomların organizasyonu hakkında bir konuşma yapacağım.
Merhaba herkese, katıldığınız için ve dinlediğiniz için teşekkürler.
Merhabalar, hoş bulduk. Başlamadan önce böyle güzel bir organizasyonda emeği geçen arkadaşlara teşekkür ediyorum. Ayrıca davetiniz için de teşekkürler.
It's a tremendous honor and pleasure for me to be here after starting my lab just over 2 months ago.
Gün boyunca 30 saat bir yayın süremiz oldu. 3 eğitim, 17 konuşma, toplamda 20 konuşmadan süren, görüyorsunuz yorgunluğumuzu da arka planda. Öncelikle bizlere konuşmalar, eğitimler veren 20 değerli hocamıza çok teşekkür ederiz. Açıkçası davet ettiğimiz hocalardan çok yüksek oranda kabuller aldığımız için bu kadar çok yoğun bir programımız oldu. O yüzden hocalarımıza çok teşekkür ederiz. Onların bize davetleri bu kadar çabuk bir şekilde, hızlı bir şekilde bizleri kabul etmesi, konuşmalar yapması bizler için çok güzel ve bizleri gerçekten çok motive ediyor. Hocalarımızın haricinde siz şu anda 277 kişi var bu saatte. Çok teşekkür ederiz. Değerli katılımcılarımıza çok teşekkür ediyoruz. Ancak büyük bir teşekkürü de ben buradaki ekip arkadaşlarıma yapmak istiyorum. Bioinforange'ın 2. yıl dönümü, doğum günü. Buradan Bioinforange'a emek veren, gönül veren, Bioinforange'ı seven tüm bilimseverler, katkı sağlayanlar, öğrenciler, insanlar adına iyi ki doğdum Bioinforange diyorum. Bilim paylaştıkça güzel, bilim paylaştıkça ilerler. Bilimle kalın. Bioinforange platformunun bilim yolculuğunda 3.sünü düzenlediğimiz Bioinfocongress IV etkinliğimize hepiniz hoş geldiniz.
Çok teşekkür ediyorum. Mehmet, senin nezdinde tüm organizasyonu yapan arkadaşları da tebrik ediyorum. Emeğinize sağlık. İyi bakın kendinize, görüşürüz.
Merhaba İlhan Hocam, tekrardan hoş geldiniz.
Merhaba, teşekkürler davet için.
Here for scanning your valuable time, transferring your knowledge to us, and for this informative presentation. Kendim ve tüm katılımcılarımız adına çok teşekkür ediyorum hocam.
Ben teşekkür ederim.
Çok memnun oldum hocam. Sağlıklı günler diliyorum.
Sizlere bir akşamlar.
Yep, thank you guys so much. I really enjoyed it. Great questions from everyone.
Merhaba Tuğba Hocam, tekrardan hoş geldiniz.
Hoş bulduk, çok teşekkür ediyorum davetiniz için. Harika bir organizasyondu, herkese selamlar.
Değerli vaktinizi ayırdığınız, bilgilerinizi bize aktardığınız ve bilgi dolu bu konuşma için kendim ve tüm katılımcılarımız adına çok teşekkür ediyorum.
Ben teşekkür ederim davetiniz için.
Merhaba Nilüfer Hocam, tekrardan hoş geldiniz.
Merhaba, hoş buldum. Beni iyi duyup görebiliyor musunuz?
Evet, görüşürüz.
Çok teşekkür ederim davetiniz için. Çok teşekkür ederim. Öğrencilerle böyle bu sanal ortamda da olsa bir araya gelmekten.
Merhabalar. Öncelikle davetiniz için çok teşekkür ederim.
Teşekkür ederim hocam.
Çok güzel bir event ortaya koymuşsunuz. Bu kadar değerli bilim insanı arasında yer almak gurur verici. Teşekkür ederim.
Çok teşekkür ederim. Nazik davetiniz için çok mutlu oldum. Bu arada bu hani YouTube'da olan sayın seyirciler, bu saatte cumartesi akşamı gidip başka bir şey yapmayıp bu konuşmayı dinlemeye geldiğiniz için size de hürmetlerimi sunuyorum.
Çok teşekkür ediyoruz. Hem kendi adıma hem de bütün katılımcılarımız adına.
Rica ederim. Ben çok memnun oldum. Beni onure ettiniz. Çok teşekkür ederim.
Biz onur olduk hocam. Çok sağ olun. Size çok teşekkür ediyorum. Katılımcılarımız ve kendim adına.
Ben çok teşekkür ederim. Benim için de güzel bir deneyim oldu.
Bu harika sunum için katılımcılarımıza bütün ekibimiz adına çok teşekkür ederim hocam.
Ben çok teşekkür ederim beni misafir ettiğiniz için.
Thank you. Thank you so much for this space. Thank you for having me here and for this opportunity.
Bilgi dolu bir konuşma için kendi ve tüm katılımcılarımız adına çok teşekkür ederim. Ben teşekkür ederim davetiniz için.
Tekrardan çok teşekkür ediyoruz. Katıldığınız için çok mutluyum.
Ben çok teşekkür ederim. Çok güzel bir organizasyon gerçekleştirmişsiniz. Beni davet ettiğiniz için tekrar teşekkürler. İyi günler diliyorum.
Biz teşekkür ederiz hocam. Bioinforange platformu olarak düzenlediğimiz Bioinfocongress 3 etkinliğimizde çalışmalarına yurt içinde ve yurt dışında devam eden değerli bilim insanları katılımıyla toplamda 15 seminer gerçekleştirmiştik. İzninizle burada organizasyonda emeği geçen arkadaşlarımı davet etmek istiyorum. Şimdi burada ekranda olan arkadaşlarımız haricinde kongremizin düzenlenmesinde organizasyonda emeği geçen tüm ekip arkadaşlarımıza teşekkür ediyoruz. İşte sizler gibi yani burada hepimiz öğrenciyiz biz ekip olarak. Özellikle öğrencilere bir katkımız oluyorsa akademik hayatı ve işte gelecek idealleri konusunda bir fikir verebiliyorsak buradaki konuşmalar, işte bizler bir katkı sunabiliyorsak açıkçası biz bundan çok mutluluk. Tekrardan etkinliklerimize devam edebilmeyi umuyorum. Bizi sosyal medya adreslerimizden takip edebilirsiniz. Sizlerin desteğiyle biz motivasyon buluyoruz. Bizler motive olunca da böyle güzel etkinlikleri sizlere sunmaya çalışıyoruz. O yüzden karşılıklı etkileşim ve destekleşmeyi umuyorum. Tekrardan hepinize çok teşekkür ediyorum. Katıldığınız için de çok teşekkürler tekrardan ve herkese bol başarılı akademik kariyerler, güzel bir hayat, sağlıklı bir hayat diliyorum ve sağlıklı günler diliyorum. Görüşmek dileğiyle tekrardan. Hoşçakalın. Merhabalar, ben Mehmet Çalışeki. Bioinforange platformunun kurucusuyum. Bioinforange platformunun bilim yolculuğunda dördüncüsünü düzenlediğimiz Bioinfocongress 4 etkinliğimize hepiniz hoş geldiniz. 6 Şubat tarihinde ülkemizde gerçekleşen depremlerde hayatını kaybeden insanlarımızı anıyoruz. Ailelerine ve ülkemize başsağlığı diliyoruz. Bir kez daha doğal afetlerde canlarımızı kaybetmemeyi ve bu konuda gerekli tüm tedbirlerin alınmasını, bilime, eğitime ve liyakata önem verilmesini istiyoruz. Depremde biz de ekip arkadaşımız Gizem Nur Çıra'yı kaybettik. Kendisini sevgi ve özlemle anıyoruz. Tüm sevdiklerine başsağlığı diliyoruz. Kendisiyle birlikte ailesinin kurtarılması için sosyal medyadan çok uğraş verdik. Kendisini bugün aramızda görmek çok isterdik. Üzgünüz ve unutmayacağız. Depremde ve sonrasında bilimin önemi bir kez daha öne çıktı. Ülkemizde bilime ve bilim insanlarına gerekli değerin verilmesi ve onların dikkate alınmasını umuyor ve istiyoruz. Bilimin ışığında aydınlık günler görmeyi ve canlarımızı artık kaybetmemeyi diliyoruz. Bugün ayrıca 18 Mart Çanakkale Zafer Günü, başta Ulu Önder Mustafa Kemal Atatürk ve silah arkadaşları olmak üzere tüm şehit Mehmetçiklerimizi saygı ve minnetle anıyoruz. 2020 yılının Mart ayında bilim paylaşımı amacıyla faaliyetlerimize Bioinforange platformu olarak başladık ve bu kongre ile birlikte Bioinforange platformunun 3. yaşını kutluyoruz. İçimizde ülkemizde yaşanan acı kayıplardan dolayı buruk olsa da bilimin önemini bilerek önümüzdeki 2 gün boyunca bilim paylaşımı adına önemli bir kongre düzenleyeceğiz. Öğrencilerden oluşan ve kar amacı gütmeyen Bioinforange platformu olarak hem dergimiz Bioinfojournal'ın 6. sayısını yayınlamak hem de çok değerli bilim insanlarının katılımıyla 4. kongremizi gerçekleştirmenin heyecanı ile buradayız. Bioinfocongress organizasyonumuzda değerli bilim insanlarının bilim yolunda ilerlemek isteyen siz değerli katılımcılarımızı buluşturabilmeyi amaçladık. Umuyoruz ki önümüzdeki 2 gün boyunca bilim disiplinleri hakkında fikir ve bilgi sahibi olabileceksiniz. 2 gün boyunca gerçekleştirilecek olan 12 konuşmadan 6 konuşmaya katılan katılımcılarımıza İngilizce katılım belgesi gönderilecektir. Buna ek olarak katılımcılarımız vereceğimiz diğer değerli armağanları kazanma şansına sahip olacaklardır. Yoklama formları konuşmanın bitiminde soru cevap bölümü esnasında açılacaktır. Yoklama linkleri yalnızca 5 dakika açık kalacaktır. Bu konuda aklınıza takılan her şey için sitemizi ziyaret edebilir ve paylaşılan kongre kitapçığımızda ise gerekli bilgileri bulabilirsiniz. Ayrıca konuşma aralarında paylaşacağımız site ve kongremizle ilgili tanıtım videolarından da gerekli bilgileri edinebilirsiniz. Kongre sürecimizde YouTube chatimizi daha çok güzel yorumlarınız ve hocalarımıza sorular için kullanmanızı rica ediyoruz. Böylece konuşma sonlarında sizlerin de değerli sorularınızı hocalarımıza iletmemiz daha kolay olacaktır. Bu konudaki özveriniz bizler için çok kıymetli olacak. Hepinize tekrardan hoş geldiniz diyorum ve herkes için iyi bir organizasyon olmasını diliyorum. Biraz sonra değerli hocamız Sumru Bayın hocamız konuşmasıyla bizlerle birlikte olacaktır. Görüşmek dileğiyle.
Bioinfocongress IV. Dr. Sundubay'ın Age-Dependent Regenerative Mechanisms in the Brain. Dr. Sundubay'ın is a stem cell biologist with a long-standing interest in studying stem cell diversity and molecular regulation of stem cell behaviors in the brain. She completed her Bachelor of Science and Master of Science in Molecular Biology and Genetics at the Bilgi University and her PhD in stem cell biology at NYU Langone School of Medicine in the laboratory of Dr. Dimitris Plekantomakis. She completed her postdoctoral training at Memorial Sloan Kettering Cancer Center in the laboratory of Dr. Alexandra Joyner. Dr. Bayan has received a number of awards, including NIH Pathway to Independence Award, Sloan Kettering Institute Postdoctoral Research Award, and was selected as a 2020 Leading Edge Fellow.
Dear Dr. Sumrubayan and participants, welcome all of you to our Bioinfocongress IV event. Dr. Sumrubayan will give us a talk today on age-dependent regenerative mechanisms in the brain. At the end of the presentation, we will have a question-answer section prepared by our team and consisting of questions from our participants. Dear Dr. Sumrubayan, welcome back.
Thank you so much, Nazlı. Thank you everyone for invitation, and I look forward to presenting.
Thank you so much. If you wish, you can start your presentation.
All right. Can you guys see the slides full screen?
Yes, hocam. We can see clearly.
Perfect. Hello, everyone again. Thank you for the fantastic organization and invitation. I'm really excited to be here. Also, thank you everyone who's tuning in on a Saturday, which is, in these stressful and upsetting times that we're going through. It is really nice to see all of you, and I hope you get a little break and enjoy the science you're going to hear these 2 days. And there's a fantastic lineup. So I'm really honored to be a part of the speakers for this Congress. So I am a group leader in the Gurdon Institute at University of Cambridge. So my lab is very young, just about a year old, actually. We just celebrated our first birthday. And in the lab, we're interested in the age-dependent regenerative mechanisms in the brain. And today I'm going to talk to you a little bit about, why are we so interested in stem cells, developmental biology, and how we can use these to address certain disorders or effectively develop therapies using this knowledge. So just to bring everyone a little bit on the same page, I would like to kind of give a brief introduction about stem cells. And as a stem cell biologist, I've been trained as a stem cell biologist, and I've been always fascinated by how can a single cell, like a fertilized egg, gives rise to us, very complex organisms, trillions of cells, more than 200 different cell types, and then how can molecularly a single cell know eventually what to make, right? And stem cells are crucial throughout the whole developmental biology process, and also once we establish we are grown-up adults, maintenance of our bodies. So, we are not we can survive throughout diseases and injuries and so on. So stem cells function throughout all of this process at different capacities while generating different cell types, but also using tissue-specific stem cells in the adulthood to maintain the organs and so on. And then how the stem cells do this is pretty critical. They are unique cells that have the ability to self-renew, so which means they constantly replenish themselves without losing the initial stem cell population, but also they're able to undergo a lot of proliferation through progenitor states and then commit to certain lineages, which eventually differentiate and mature to specialized cells like the neurons in the brain that does the function of the tissue. And as I said, proper function of stem cells is critical for proper development, homeostasis, and something that we're really interested in the lab is in the repair or after injuries. So without proper stem cell functions, we cannot repair any injuries that causes cell loss or damage to the tissue. So, and importantly, we're interested in the balance of these processes because when the stem cell function fails, you either get degenerative diseases, or if it's overly active and they proliferate out of control, you get overgrowth or other diseases such as cancer. So understanding the balance of what regulates stem cell behaviors is very critical. So we're a neuroscience lab, so we're really interested and fascinated by the brain. And in the brain, there are multiple different cell types that works in orchestration to fulfill function. Obviously, I'm sure you're all aware of the neurons, which are the electrically active cells that transpass information from one and another to fulfill, higher order functions of the brain. But there are also other glial cells that are either oligodendrocytes that help them to conduct information faster, astrocytes that modulate their function, microglia, which is the immune cells of the brain, and adult neural stem cells, and all the niche components such as the vascular cells or other different type of cells that can be in the brain. And the question we're interested in the lab is that how is this different cell types generated? And again, the answer is through the stem cells that are either multipotent that can give rise to many of these lineages, or through different progenitors that are, we call lineage restricted. So at that point, they are still proliferative and have some stem cell capacity, but compared to their predecessor, it's a lot limited, and they cannot make various lineages. They can only make the lineage they're committed to. So I wish the brain was simple enough that there was just 3 cell types, but actually brain is one of the most diverse, one of the organs systems that has vast cellular diversity. Well, and our understanding, and I think today and these 2 days you're going to listen a lot about this, our understanding of cellular diversity has changed dramatically over the last century. So initially we would kind of look into cell shapes and understand how that may be important for morphology and function, and like classify cells based on those type of events, to now with the generation of over the past, I guess, decade now, single cell technologies and spatial transcriptomics and so on, we can now, even within the same cell type, get into molecularly diverse subpopulations within different regions of the brain and so on. And even within the stem cell compartments of the brain, we now know that there are multiple different molecularly defined progenitors. And today I'm going to talk to you a little bit about that. But from a stem cell biologist point of view and a developmental biologist point of view, in the lab, we are interested in understanding how is the cellular diversity established during development and how is it maintained. Therefore, we're really interested in a concept called lineage plasticity. So this is a little bit different than you may have heard synaptic plasticity in the brain, where neurons wire, reorganize their wiring and so on to modulate function. Here we're talking about lineage plasticity. And what I mean by lineage is, I already told you that, different stem cells, diverse stem cells function coordinatively together to generate different neuron or astrocyte types and so on in the brain during development. And this happens through committed progenitors. So if there's lineage plasticity, we argue that possibly different progenitors that are committed to different lineages may change their fate at some point and give rise to alternative progeny. So this is not really a very highly observed process in the brain. And what is more interesting for us is that, especially when there's an injury in one of these components, what are the compensatory mechanisms? Are there any compensatory mechanisms to begin with? And if so, what are the compensatory mechanisms and lineage, injury-induced lineage plasticity that is in the system to replenish the lost cells? And importantly, in the lab, we are really interested in understanding what are the molecular factors, signaling, or genes that are involved in these different processes so that we can modulate this to induce lineage plasticity upon injury. So why do we care about understanding this? So traditionally, central nervous system is, simply put, cannot regenerate, right? So when we get injuries to the brain or neurodegenerative diseases and so on, there is no efficient new neuron production to replenish the lost cells. So there are many studies that are working on trying to overcome this. And one of these approaches are, for example, generating stem cells or neurons in vitro from pluripotent stem cells, or you may have heard induced pluripotent stem cells from patients and injecting them back in. And there's a lot of promising studies, for example, for Parkinson's disease on that front. But in certain regions of the brain, we argue that this may not be as efficient. For example, one of those regions is the cerebellum, which is on the back of our brains. It has actually majority of our neurons, so 80% of our neurons are in the cerebellum. And it has these very complicated, layered and foliated structure that just injecting a ball of cells or neurons, we argue, may not have as efficient effect upon injury. Therefore, in the lab, we're interested in understanding how can we activate the stem cells of the cerebellum. First of all, are there stem cells in the cerebellum or progenitor cells? And how can we activate them upon injury to generate new neurons? But there's a lot of knowledge from developmental biology throughout to the adulthood that is missing for us to do this approach. So today I'm going to explain to you a little bit about how we are aiming to tackle this question. So as you may have guessed now, we're very interested in a specific region in the brain, that is the cerebellum. And there are multiple different reasons of why we're interested in that. So first of all, cerebellum is important for our motor coordination, how we walk, how we, our posture, everything, how we move. But also now, through its connections to the forebrain, people have been every day finding out new functions for cerebellum in pretty much every other cognitive and social behavior. The reason we're particularly interested in the cerebellum is that it also develops a little bit differently than the rest of the brain. It develops later. So majority of the growth of the cerebellum happens postnatally, meaning after birth. So here you see an MRI, MRI of a mouse brain, where after birth up to 10 days, 11 days, and then you can see how the cerebellum is growing a lot, and it's starting to form foliations to hold on to the to increase the number of cells it has to maintain them in a small structure. So this is up to 2 weeks in mice and up to 6 months in humans. So it develops a lot later, and it develops outside of the womb, which makes it susceptible to injury around birth. And actually, cerebellar injuries that happen around birth are the second leading risk factor for autism spectrum disorders. So it is really important to understand how the cerebellum develops properly and whether there are regenerative mechanisms around this stage. So over the years, starting with my postdoc research and now in the lab, we're interested in understanding whether there is increased plasticity and regenerative potential in the cerebellum around birth. And indeed, over the years, my colleagues and I were able to show that this is true, and that the neonatal cerebellum is also unique in its capacity to regenerate very efficiently. And today, I'm going to show you some of those mechanisms and how we plan to tackle this knowledge and developing this to further possible therapeutic avenues. So a little bit introduction onto how cerebellum develops and some key cell types that we will be talking about today. So as I mentioned, most of the growth of the cerebellum happens postnatally, meaning after birth. And all of these cells of the postnatal cerebellum, all the stem cells in the postnatal cerebellum are defined, committed to lineages early on in the embryo, and they are either generated from the rhombic limb, which gives rise to, in the end, all the excitatory neurons of the cerebellum, which is a subtype of classical main subtype of neurons that fire in a positive direction. And the ventricular zone gives rise to all the inhibitory neurons, and importantly, all the glia, all the support cells that I had mentioned, such as the astrocytes, and a special cell type called Bergman glia, which is an astrocyte-like, but it has a special shape that is unique to cerebellum, which we're going to talk about it in a bit. So all these adult cell types, they need to undergo a lot of proliferation during the postnatal period, and then this proliferation is maintained by, is done by different progenitors of the postnatal newborn cerebellum. So one of this is the granule cell progenitors, and they proliferate on an outside layer of the cerebellum here, and then they migrate inside to give rise to granule cells, which are excitatory neurons. So one of the questions we were interested in the lab was that the nestin expressing progenitors, which give rise to the postnatal inhibitory neurons and glia, what are the lineages that are generated from the cells? And we knew they were diverse subpopulations of nestin expressing cells. So what are those? So I'm going to summarize because this is published, but that we were able to show that there are multiple nestin expressing subpopulations, and then they were defined by whether they were gliogenic, meaning eventually give rise to Bergman glia and astrocytes, or neurogenic that give rise to inhibitory neurons. So they are lineage committed in the postnatal stages, okay? And then we were able to show that these guys were marked by either HOPX or ASCL1, which is a transcription factor critical for neuron production in the brain. So one thing to note, which is going to be very important, and that's why I put this dotted line here, is that if a cell type is from the rhombic lip, and in the postnatal case is granule progenitors, it cannot give rise to any of these ventricular zone-derived cells. So the progenies here that are defined as ventricular zone-derived or rhombic lip-derived is very lineage committed in the development. There is no lineage plasticity under normal circumstances between these cell types. But what we have done, and this was started with a previous postdoc in the lab, is that when we perform injury to the proliferating granule cell progenitors, and I'll explain you the injury in a second, we surprisingly observed that now the gliogenic HOPX positive progenitors, and actually a specific subpopulation of these progenitors that reside in the special layer called Bergman glia layer, due to the cell types they give rise to eventually, is able to now change its fate and give rise to the cell type that is affected by the injury, which is the granule cell progenitor, and then eventually give rise to the excitatory neurons. So this is kind of a classical definition of what I explained to you by lineage plasticity, that these cells that normally never make these 801 positive neurons is now with injury making this. And this process is called adaptive reprogramming. So what is adaptive reprogramming? It is a complex multi-step process. So when we kill the cells, and here we use irradiation because it selectively kills these highly proliferating granule cell progenitors, then what happens is in a day, granule cell progenitors die. And then this somehow stimulates these nestin expressing cells that are in the Bergman glia layer because they have these processes. And then these cells first proliferate, then start migrating to the site of injury. And then as they reach there, they start turning on granule cell genes. All right. So we understood a lot about molecular, on cellular level, how are these cells responding to injury, meaning, you know, increased proliferation or migration and so on. But when we started in the project, we did not know much about the molecular events that take on here. So to do this, we took on a single cell sequencing approach to understand basically at all these different stages of adaptive reprogramming, what is driving these processes, which genes or pathways are involved in this. So before I get into those results, I want to give a brief introduction to those who may not be familiar with single cell sequencing. So it is a very powerful technique that allows us to identify basically on cellular basis, on single cell basis, obviously, hence the name, the transcriptome, or based on other approaches, for example, the chromatin accessibility of individual cells. And to do those, you basically join a single cell with these beads that have particular barcodes in them that you're able to selectively identify the single cell based on which barcode it is attached to and all the individual mRNAs coming from that cell in these little lipid droplets. You lyse the cell once they're in the lipid droplets with the beads, and then you perform reverse transcription to identify, to get cDNAs from the cells, and then you collect the barcoded cDNA. So each cDNA will have a unique barcode to designate as a novel molecule, plus the barcode that tells which cell it is coming from. And then you carry on with sequencing. And in the end, and I'm sure you guys are now familiar with more of these graphs, you kind of get graphs like this, which is either done with certain levels of dimension reduction, that each dot is one cell. And then after certain computational algorithms that I won't get into in detail, first of all, because I'm not an expert at them, but based on their similarity in their gene expression, grouping cell types together so that you can identify, okay, in my sample, there's 8 cell types or 8 cell states that's doing this and that and so on. So we took on this approach in collaboration with Peter Sims and Doğukan Mızrak at University of Columbia University to identify how is adaptive reprogramming occurring. So we performed we sequenced 19,000 cells from postnatal day 5, nestin CFP cerebellum. So this means that we performed injury at day 1, which is what we always do injury, and then 4 days after we collected cells. And the reason we collected it 4 days after is that this is a stage where we see all these proliferation, migration, and transdifferentiation events occurring together. And we did this in a transgenic mice, which allows us to identify the nestin-expressing cells through a fluorescent reporter. So we got lots of cells from the whole cerebellum. We pulled out our nestin-expressing cells based on the CFP expression, the fluorescent marker expression, and then we did iterative clustering with or without injury to understand what are the changes in that. So we were able to observe our gliogenic NEBs and neurogenic NEBs and how they change with injury. So I'm going to summarize these results because this is all published, and I want to talk a little bit about more unpublished stuff. But what this allowed us to understand that when we started investigating the injury-induced changes in our NEB populations, we started observing that the neurogenic NEBs, the distinction between the neurogenic and the gliogenic NEBs, kind of started graying out a little bit. And then we observed increased ASCL1 expression in the gliogenic NEBs. And this was surprising because ASCL1, which is, I told you, a transcription factor important for inhibitory neuron production in the cerebellum is also never involved in the ATO1 excitatory neuron production. So it didn't really make sense why these guys would turn on ASCL1. So we wanted to investigate this further, and then we started looking into genes that are likely to be expressed with ASCL1 with injury, which is you see on this side of the graph. This is a probabilistic analysis of two genes being expressed together in the data. And you see likelihood of genes to be expressed with ASCL1 in the injured cells. And then we observed that in the control case, all the genes we get were restricted to the white matter, which where the interneuron progenitors are. But upon injury, we started seeing genes that are known to be expressed in the Bergman glia cells. And this is the cells that induce lineage plasticity to replenish the lost cells. So we hypothesized that actually the Bergman glial NEPs during adaptive reprogramming to ATO1 positive granule cells, they undergo through an ASCL1 positive transitory state. So we wanted to confirm this since this is this hypothesis set us as an interpretation of the single cell data. So we started performing, first of all, RNA in situ, so where we look at ASCL1 expression. And indeed, ASCL1 itself is also increased in the injury in Bergman glia layer. So we wanted to do as a developmental biology, just gold standard experiment to see if these cells do indeed give rise to granule cells with injury. We performed fate mapping. So when we perform fate mapping, just to briefly explain to the folks who may not be familiar with it, we use an ASCL promoter-driven Cre recombinase that translocates into the nucleus when we inject a drug called tamoxifen and removes the stop codon in front of an fluorescent protein. Therefore, when the tamoxifen is injected, cells that express ASCL1 will turn on red. And any cell from that point on that is generated from that red cell will maintain the red because we're modifying the genome at that point. So in the control case, when we do fate mapping and look a few days after, basically just looking at what is initially labeled, all the labeling we get is in the white matter, which is where the interneuron progenitors are. And then occasionally cells we get in the Bergman glia layer, which is the injury responsive layer, are the cells that are migrating out to form the interneurons. But when we look at injury now, as we hypothesized, we are now getting these new cell types being labeled, which has these projections to the outside of the cerebellum, like the Bergman glial express NEPs that generate Bergman glia, but now they actually are expressing ASCL1. And when we look at later, so we do injury, we label the NEPs, and then we look a couple weeks after to see in the end whether they were really able to establish granule cells. And then here you can see the quantification, which is easier to interpret. Only with injury now, we're seeing granule cell progenitors produced from ASCL1 positive cells, okay? We also see an increase in interneuron production because of the delay in development when the injury is happening. And then this is important. We did not see any increase in the Bergman glia labeling with injury. This means that once the cells turn on this transitory ASCL1 population, they are really committed and they cannot go back to make Bergman glia, okay? And therefore, we thought ASCL1 is required to inhibit this fate so that the cells can take on this new fate with injury. And then we also observed increased astrocyte production, which we don't know exactly what to make out of it yet, and curious to understand further. But this is kind of an interesting process because ASCL1, as I told you earlier, has been used heavily to induce neurons. And people actually are overexpressing ASCL1 and other astrocytes in the rest of the brain to generate neurons after injury. But this experiment was pretty exciting for us because in this case, cerebellar cells, cerebellar progenitors, are able to turn on this gene on its own. We're not doing anything. The animals regenerate very well, There is some inherent capacity of these cells to carry on adaptive reprogramming and lineage plasticity. So in the lab, we're now trying to understand what is this inherent capacity. And importantly, as you can see, and I pointed out many times today, ASCL1 is normally involved in a completely different process and cell type production, where with injury now it's different. So in the lab now, we're trying to understand how is the ASCL1 functioning to establish this new lineage with injury. And to do so, we're looking into also not just the RNA of these data, but we're performing the single cell attack sequencing, which takes into account the open chromatin regions where you can infer what is epigenetically open as opposed to epigenetically silenced, which is a way of stem cells deciding which lineages to make or not, so that we can understand how the ASCL1 function in this injury-induced state versus the developmental state is different. So the data you get from this is pretty similar, but now, again, each dot you see is one cell, but the clustering is made based on inferred expression based on the open chromatin, whether it's close to a gene or not, a promoter region of a gene or not. And then we were able to show that, and we're investigating this further. For example, with injury, there are very specific regions that just opened up that has binding motifs for ASCL1, which may be important for the lineage plasticity. So we're right now trying to understand this, but hopefully just to take a little breather and summarize a little bit here that we are pretty confident now that the neonatal cerebellum is pretty unique in its regenerative potential, and then it is a very good system to study lineage plasticity, which is not very commonly occurring in the brain elsewhere. So one other question that lab is really interested in based on these results and the uniqueness of cerebellum is also, neonatal cerebellum is unique and regenerative. Does this mean can the adult cerebellum also regenerate? Maybe it was something we're missing. And when we say brain is not regenerative, we haven't really truly explored the adult cerebellum. So just for those who may be more familiar with the neuroscience and neural stem cells, cerebellum is not a classically neurogenic zone. So it doesn't have ongoing neurogenesis in the adult like the subventricular zone or hippocampus. So we were first wanted to investigate whether there are stem-like cells or any stem cells in the cerebellum that may be responsive to injury. So we started looking into our favorite marker now that marks the NEPs, which is nestin. And we were able to show that there are actually rare nestin-expressing cells in the cerebellum. And interestingly, most of these cells, about 75% of them, are in the Bergman glia layer. And remember, this is the layer that has the cells that is able to regenerate in the neonates. And then these cells do not express mature Bergman glia markers, which is S100 beta. So they do seem to be more undifferentiated. And they are able to fulfill certain stem cell criteria, one of which is single cells' ability to grow into these round fat spheres, which is a readout of they're able to self-renew. So they are indeed able to perform this assay better than the CFB negative cells of the cerebellum. And then finally, when we do fate mapping, which is our way of understanding what do these cells give rise to, they fulfill the lineages of their neonatal counterparts, so they are gliogenic, so they don't generate neurons, but just give rise to Bergman glia and astrocytes when we do fate mapping. So this was really exciting at the time because, there is an undifferentiated looking cell type in the brain. Can this regenerate? So we performed experiments to the adult brain. So we did injury, and then we were excited to see that actually with injury, these cells are able to know that it is injured and are able to increase the production of astrocytes and neurons. But they are not proliferative and no new neurons is generated. So we think it's coming from a different cell type, which I'm not going to get into it today. But one thing right now we're in the lab we're interested in is okay, there are stem cells in the adult, but why can't it regenerate, right? So the neonatal SNPs I showed you are able to start from the stem cell state and go all the way to the neuron production with injury through these different variation of steps. Whereas adult NEPs, they know there's injury, they do something, but they are really blocked starting from proliferation step. So in the lab, we're interested in understanding how can we push these adult NEPs to regenerate neurons upon injury. So our hypothesis and our approaches is twofold. So in order to be able to facilitate regeneration, we believe that we need to understand the cell intrinsic and extrinsic mechanisms that first allow regeneration in the neonates. And I already showed you some of the approaches we're using for this. And importantly, inhibit regeneration in the adults. So how can we make the adult non-regenerative NEPs become regenerative is in the end our goal. So to do this, we hypothesize multiple different mechanisms that act together. One is once the development ends, the developmental signaling is reduced, so these cells are not actually as stimulated to proliferate or differentiate. Secondly, once the stem cells commit, they usually silence other pathways so that we don't get the differentiation or tumors or aberrant reprogramming, so that the adult tissues maintain properly. And then finally, the environment may be permissive to regeneration, pro-regenerative. And for example, the immune component in the neonatal and adult can be different. And we're investigating that in the lab, but I'm not going to talk about it today. So first, we wanted to investigate whether if we increase developmental signaling in the adult NEPs, can we induce more regeneration? So to do that, we knew sonic hedgehog pathway was required for regeneration from our previous studies in the neonates. So in the adults, after injury, we activated sonic hedgehog and look into the NEPs to see if we observe any increase. And indeed, injury plus sonic hedgehog activation increased the number of adult NEPs significantly. But this did not lead to any proliferation and neuron production. So what happened basically was we were able to push this tiny bit further, but again, there was some internal blocks. So now in the lab, we're thinking that we need a combinatorial and sequential approach where, for example, we first induce proliferation, then plasticity, and so on and so forth to really fulfill the whole lineage steps. To understand, though, we still need to understand how all these different steps are regulated molecularly. So we're going back to single cell sequencing that I mentioned to you. And we now the previous data I showed you was only just one time point, but we're now looking into all the different stages of adaptive reprogramming to understand how the lineages are made and how it changes with injury. So you can see a sample data here from all these time points that varies a lot with age. Like, for example, we get the granules hub progenitors at later time points, which is a later injury-induced cell type, and so on. And then we are able to identify all our favorite cells from this data to investigate further. So as I said, we're really interested in understanding the lineage dynamics and the injury-induced signatures. So to do this, I want to give you just the final idea of type of analysis we're interested in. We are pulling out our HOPEX expressing NEPs and ASCL1 expressing NEPs, reclustering them, and then looking into how does the HOPEX expressing NEPs change with age and injury, vice versa. You can see we're looking into the population dynamics over time. And then importantly, we perform an analysis, one of which was called RNA velocity, which orders all these single cells based on pseudo time. And for example, when we look at it for the gliogenic lineage, we can then infer different paths that the stem cells go through to make astrocytes or Bergman glia. In this case, you can see two paths, one for Bergman glia, one for astrocytes, to really understand what is driving, at this point, different lineages from these cells. And then the other type we're looking into is also same with the neuron production. Again, how the different paths that these single cells take to generate in the end neurons. And you can see, for example, we start with the progenitor here, and then as it matures, it becomes the immature interneurons that is labeled by PAX2, and then the algorithm is able to detect the directionality of this. So now we're interested in these differential nodes. Where are these? What are the molecular mechanisms that making the cells to become more mature, and so on and so forth. And then finally, as I mentioned, we are getting a lot of idea from this data, and we are already able to, I'm not going to get into detail, but get some ideas about possible mechanisms involved in all of these steps, and particularly the lineage plasticity, which we know is really inhibited in the adult. So we're trying to understand how in the adult we can induce this, and what is the signal that inducing this transitory state that is required for regeneration. So to do those, we're also using in vitro models from mouse NEPs in vitro, and then differentiate them into neurons and astrocytes to study how we can push them to one lineage or differently. And then finally, last piece of data I want to show before we finish today is that the reverse side of the coin, the silencing of developmental pathways, and how we plan to study that. So to do that, we're looking into why the adult regeneration at what step is blocked. And to do that, we initially took on a chromatin-based approach where we're again looking at the attack sequence data of the NEPs from the neonates with or without injury, and then the adult with or without injury. So in the neonatal, here you see a volcano plot, a tornado plot that has the open chromatin region. So if a region's open, it's darker blue. And this is kind of the transcription start side of genes that are involved in neurogenesis and proliferation, all right? So in the neonates, everything is pretty much open because this is still an actively developing and neurogenic status. And injury doesn't seem to change much. But you can see in the adult, all the neurogenesis and proliferation pathways are genes seem to have closed chromatin. And thus we know is the stage that is blocked. And the injury is unable to turn on these genes. Whereas upon injury, for example, the glial differentiation and stretch genes, which is from our fate mapping, suggested is able to partially turn on. So there is, besides lack of developmental signals that I had mentioned to you, there is active silencing of some of these stem cell programs in the adult NEPs, despite there is injury. So one of these programs is actually lack of MYC. MYC is a proto-oncogene that is really important for many processes, but also cellular proliferation and reprogramming actually. So in the neonatal NEPs, we know that MYC is highly expressed, and you can see also the open chromatin around the MYC gene. But in the adult, it is inhibited, it is silenced, and with injury, it is unable to turn on. So one of the mechanisms we're testing now is that if we activate MYC, can we get more proliferation of the NEPs? So we use the transgenic mouse line that induces MYC overexpression. And you can see when we overexpress MYC actually, selectively Bergman glial NEPs, and we identify them using SOX2 here, is able to proliferate where normally they're not proliferated, which was exciting. So we're hoping to now push this a little bit further to overcome the lack of proliferation. And to do so, we're collaborating with Catherine Wilson and Department of Pharmacology here to use actually modified RNAs because, you know, we don't want to develop a therapy that can give you cancers down the line because MYC overexpression is a very well-studied oncogene. So we're using modified RNAs, similar to all those RNA vaccines we all got over the last couple of years for COVID-19, that is stable, but transiently induces MYC expression in the cells and then doesn't integrate into genome and cause any other problems. And we have some preliminary data that it looks very, very promising. So the goal would be to induce MYC overexpression transiently so that we can overcome this proliferation hurdle we're having, and then build upon it on additional signals, for example, whatever that induces ASCL1 expression. So with that, I'll summarize and then finish so we can talk to questions. So hopefully today you guys were excited about, importance of stem cells and their function during development, but also how we can utilize them for regenerative therapies, and that neonatal cerebellum is a very powerful system to investigate some of these molecular biology behind stem cell behaviors and how the regulation of their self-renewal, importantly lineage plasticity and differentiation, is can be harnessed for future studies. And then in the lab, in the next, some of our primary goals is to translate our findings to human biology, but also understand whether pro-regenerative signaling is involved in tumorigenesis and whether other regions of the brain have similar age-dependent regenerative processes. So just a little bit of acknowledgements. This is my lab. Some of the data I showed you today was generated by Jens, the single cell data, and Jada has done some of the mixed studies, a list of our collaborators. My postdoctoral lab is where most of these studies have been started, and actually some of the single cell data has been generated, and our funding agency. So we're always looking into excited and talented people to join our lab. So if you were interested in any of this, please feel free to get in touch with me, and I'll be happy to take any questions in English or in Turkish, whichever you're comfortable with. Thank you.
Thank you very much, Dr. Sumru Bayan. I'm directing the first question. An increasing number of pro-aging and pro-youthful factors are being identified in the blood. To what extent do pro-aging and pro-youthful factors converge or act through different mechanisms?
Okay, yeah, that's an interesting question. Yeah, it is part of the, I guess, the cell extrinsic mechanisms we're talking about, right? So all the, there's a lot of old studies where, for example, you stitch together a young and an old mouse, and then the blood of the young mouse is able to rejuvenate the old mouse as well. So there's many different processes. So I think it is a little bit hard to say how do they act through different mechanisms, because they all in the end stimulate certain processes. But when we talk about aging, part of the mechanism is to prevent accumulation of, for example, reactive oxygen species or harmful substances in the cells and so on. Whereas some of the other mechanisms, which is what more we're interested in, is to stimulate new cell production. So it can be preventing cell from dying and functioning properly, or it can stimulate new cell production or tissue turnover. So those are two main mechanisms. But if we get into molecular mechanisms, I think there's so many different approaches that they can happen. And I need to clarify one thing. So we say it's age-dependent regenerative mechanisms, but it's also one thing I should point out. The point we see adult regeneration not happening is not really even an aged organism. It is adult, meaning right after development ends in the sense. So some of the regenerative mechanisms are really restricted to developing brain. So even if you're not aged in the sense 20-year-old, your brain still doesn't regenerate, making new neurons. So it is more cell intrinsic in certain ways than an actual aged organism, if that makes sense.
Thank you so much. I'm continuing with the next question. Nowadays, do we determine whether NAPs in adult cerebellum can be stimulated to replenish cells lost due to the injury?
That's what we're working on right now. So we know these cells are able to sense there is injury. So what I mean by that is they increase in numbers, they become more stem cell-like, but again, they cannot make neurons, which is what in the end we need, because neurons are the cells that are dying. So that is our lab's goal. So to determine this, we're doing lots of fate mapping with injury for adult NEPs after injury to see if we can get more neuron production. So stay tuned. Yes, that's our goal. But we haven't made extensive number of neurons from NEPs yet. So that's why we're going back. That is exactly actually the reason. We're going back to the development to really understand how the developing brain can regenerate so that we can replenish those signals in the adult, because right now they are unable to.
We appreciate for your answer. Thank you so much.
Mm-hmm.
I'm moving on to the next question. As an example on zebrafishes, young and elderly fishes are differentially impacted from stress, especially for young fishes. It can be turned to chronic stress, but regeneration ability isn't changed. Could we say physiological balance is highly affected by stress response and maybe after that growth and tissue regeneration?
Yeah, no, that's a great question. Again, so for example, there is a study, a lot of studies in the brain that exercise actually stimulates stem cells, for example, right? And then keeps them happy and neuron regenerating. So on the other hand, constant stress can let these cells also molecularly kind of gain epigenetic memory due to like environmental stress, chemical toxins, all sorts of things that will eventually prevent them from functioning as well. But again, coming back to our research, the adult NEPs in the cerebellum are, even default, are unable to make neurons. So we need to basically make them remember that when they were during development, know how to make this, and they should remember that and make it again. But yes, generally, I think regarding zebrafish brain, it is a great model, but in this case, it is not really the best model to compare our results to, because actually zebrafish cerebellum never stops regenerating, as the person has pointed out, and that actually there is constant proliferation in the cerebellum, specifically in the zebrafish. So at some point, we're interested in doing some evolutionary studies. Why is this potential lost, which we think is more cell intrinsic, possibly in the humans or mice, actually. So yeah, that's definitely an interesting comparison that we don't have an answer of why yet.
Thank you so much. Is ROS signaling necessary to drive reprogramming in neurons?
Yeah, that's a fantastic question. We do have, and we're working on a paper from Alex's lab, my postdoctoral lab, that ROS seems to be the alarm signal. What I mean by alarm signal is that the dying cells release more ROS, which stimulates stem cells. And if that step is failed, we do not get reprogramming. So it is crucial. And this is pretty well studied in many other injury paradigms, that initial cell death is, and whatever that secretes, basically, and ROS is kind of the initial, one of those key damage-associated molecules that people call them, is required for stimulating the wound healing or injury response in many cases. But yes, and we haven't studied this extensively in the adult, and whether if that signaling is somewhat impaired, that may affect. But given that our cells in the adult still know that there is injury, I assume it's not this step that is inhibited. Mm-hmm.
Thank you so much. I'm moving on to the next question. Brain has a highly energy needed system by its own. So if there is a dysfunction on mitochondrial homeostasis, can efficiently affect on axonal energy homeostasis. Could we clearly characterize the association between mitochondrial status and aging?
Yeah, so there's a lot of studies going on that, and mitochondrial function, or let's say motor dysfunction, is one of the key signs of aging that people use in the context of, what are the molecular differences that occurs in cells with aging. So that's absolutely true. And one thing, and again, this is kind of leading into some of the differences between our developing NEPs and adult NEPs. So in the case of the developing cells, these cells are still very immature and they haven't fully made neurons, and they use a different type of metabolism, we think, compared to the adult cells. And being more, metabolically, I don't have evidence for this, but I think it's more glycolytic metabolism, and stem cells tend to be more glycolytic and then turn on oxidative phosphorylation or mitochondrial mechanisms as they differentiate. This is kind of widely more or less studied in different stem cell contexts, which may be also why they are more permissive to reprogramming. So there's multiple layers within the stem cell differentiation paradigm. Yes, mitochondrial status is probably very critical for regenerative capacity, and also in the organ level, as the person has suggested, in terms of the aging aspect of like axonal biology and neural biology. There is lots of studies that shows that mitochondrial health is crucial for proper function.
Thank you so much for your informative answers. Do you think aging can be associated with increased inflammatory cytokines, including lymphotoxins, in the dorsal root ganglia?
Yeah, so I don't know much about directly the dorsal root ganglia, but I think generally, yes, more inflammation you have, you do worse, and the cells don't like that. And that's kind of, I didn't talk to you guys about why we're looking into microglia as well, and whether in the neonatal, in our hypothesis is during neonates, microglia, because they're actually functioning in their developmental state, is not inflammatory, where in the adult injury, actually, microglia is becoming more inflammatory, and therefore it is inhibiting regeneration. So yes, more inflammation, and this is again signs of general environmental stress or chronic stress, or toxins and so on that may lead to more inflammatory like state, is never good for any types of system. Mm-hmm.
Thank you so much. I'm continuing with the next question. How far-reaching are the effects of aging and promote factors in the brain?
Yeah, I don't know. It's a hard question to answer. I think, the thing is, depends on what we mean by pro-youth factors, right? So, and whether what we're trying to stimulate. So we're trying to, for example, do a very controlled approach here, where we're hopefully trying to selectively stimulate just the stem cells or injury responsive cells. For example, there are some old studies when you force neurons to proliferate. Let's say we can use that as a possible pro-youth factor, maybe. Not really exactly what I think the person means by pro-youth factors, but most neurons die. And when we activate MYC in all the brain, for example, we only see through proliferation in the Bergman glial NEPs, which may mean that other cells, either they have other regulatory mechanisms that they couldn't induce proliferation, or they just are unhappy proliferating. So there's a lot of cell type dependent context, depending on which pro-youth factor we're talking and which cell we're talking on. So it is a little bit hard to answer this without more context, I think.
Thank you so much. Thanks for presentation. How much of the interactions between extrinsic and intrinsic regulators of neural stem cell aging could be uncovered? What are the benefits of this knowledge?
Yeah, so let's start from the second question, I guess. Benefits of this knowledge is that gives us power of regulating, right, when something goes wrong. For example, in our case, when neuron production is inhibited, if we know why is it inhibited and what factors are there, we can modulate this to induce stem cell function. To what extent this can be uncovered is, I think, this is generally, I think, biological sciences with new tools and with new developments. The extent always we uncover something that we didn't know that even was possible. And that's why, for example, in our research, we're really excited to use single cell technologies, which gave us great power on, individual cell-based, individual cell type, cell state, understanding of over time, how are these cells are functioning. So I think we don't know what we don't know, let me put it that way. But our goal in the long term is to get a full understanding of these mechanisms, but we're also limited by the technologies we can use, right?
Thank you so much again for your informative answers. Can systemic therapy approach to brain aging reveal an age-related duality in blood based on both pro-young and aging accelerating circularity factors?
Yeah, so that's a great question. I think there is possibility, but again, our results show that it's not just cell extrinsic factors or blood. We activate sonic hedgehog signaling, for example, systemically, and we still don't get the full reprogramming. So therefore, we are more rooting for a combinatorial approach of activating different levels of signaling. And this is assuming we can overcome the cell intrinsic inhibition. So it is to be determined, but from our results, we think that it's not going to be a single magic molecule or blood transfusion that can solve this problem. Yeah.
Thank you so much again. One of the YouTube question again. Why do you think Bergman glia differentiate into GCP during injury? With less Bergman glial cells, are there any chances in tissue environments?
Yeah, so that's a fantastic question. Something we're interested in looking into. Why do I think? I don't know. It was pretty surprising for us that we decided to study this, obviously. So Bergman glia cells are a little bit special, and I didn't have time to get into their differences. They are resembling more the embryonic neuronal progenitors that are called radial glia, for example, and they start their life as radial glia and then become Bergman glia eventually. So maybe we think they have some memory of this embryonic, more the more plastic state. We absolutely think there's role of tissue microenvironment in the pro-regenerative and anti-regenerative states. It's not really related to number of Bergman glia. I think it's more about overall the signaling and the extracellular, maybe the stiffness of the tissue or other things. And then we think why Bergman glia is the cells that are being able to regenerate is because they actually are in physical contact with GCPs during injury. So I think that's why they know what to make. But we're now testing if we stimulate, for example, ASCL1 and Bergman glia without injury, would we still get GCPs, or do we need cell death for Bergman glia to know what to make? Because we don't always get this response in other cell types. Mm-hmm.
Thank you so much. How well understood is the interlineage and cell-to-cell interactions required in vivo for expansion of different progenitors and survival of Purkinje cells?
Oh yeah, that's a great question. I didn't talk about Purkinje cells a lot today. Not at all, actually. Purkinje cells basically are kind of a central hub for all these processes we talked about. And Purkinje cells are the cells that secrete sonic hedgehog, which is required for regeneration and also just development of cerebellum. So we have some we have some understanding of some of these molecules, but we are still obviously expanding this knowledge. There's definitely interlineage communication, just as similar to the previous question I said, between GCPs and Bergman glia, and I think it's because cell-to-cell contact took part, and without Purkinje cells, none of this would have happened. But also there is the 3D structure of the cell with the layers that may be important for the migratory processes and so on. So it is, some of it we understand, some of it we hypothesize, but don't really have experimental proof, but it is absolutely crucial. And there is a lot of interlineage communication that gets into it. And one thing to note, I guess, on last bits, the interlineage aspect, the cell-to-cell, the regeneration stops when the circuitry is more established. So the cell-to-cell interaction and neurons firing may also affect the whole regenerative processes.
Thank you so much. I would like to thank you for your informative presentation and answers. Lastly, I would like to ask that what advice would you give to students studying in scientific field?
Yes, thank you for the invitation too. I really enjoyed them. Fantastic questions, everyone. I think the biggest advice is to read a lot, be proactive, talk to, try to kind of absorb as many different fields and information as you can at these early stages, so that you can, decide on what excites you the most in terms of biological questions or techniques or whatever. And I think in terms of network well, and I think all the Bioinforange people I met, sounds like this is part of the point of the society. So networking and getting to know people is something we sometimes don't do well, but it is important for future careers. And I mean, it goes without saying, work hard, read a lot.
On behalf of myself and all our participants, I would like to thank you for spending your valuable time, transferring your knowledge to us, and for this very informative presentation.
Thank you very much, and enjoy the rest of the talks.
Thank you so much. I wish you healthy days.
Bye-bye.
At 3:15, Assistant Professor Dr. Defne Bayık will be with us to give a speech on considerations of biological sex for immunotherapy of glioblastoma. Thank you.
Bioinfocongress IV. Assistant Professor Dr. Defne Bayık. Consideration of biological sex for immunotherapy of glioblastoma. Assistant Professor Dr. Defne Bayık is a cancer immunologist focused on uncovering host regulatory pathways as targets for brain tumor immunotherapy. She completed her undergraduate degree in the Bilkent University Department of Molecular Biology and Genetics and received her PhD in 2016 through the National Institute of Health Bilkent University Graduate Partnership Program. Her dissertation work, under the mentorship of Drs. İhsan Gürsel and Denise M. Klinman, identified differentiation pathways of immature myeloid cells to inflammatory and immunosuppressive macrophages. In 2017, Dr. Bayık joined the laboratory of Dr. Justin D. Latia at the Cleveland Clinic as a postdoctoral fellow, where she gained experience in neuro-oncology and translational cancer immunotherapy research. She demonstrated that sex differences in the anti-tumor immune response inform targeting of immunosuppressive myeloid cells in high-grade brain tumors. Her postdoc work also defined epigenetically regulated pathways that inform the functional heterogeneity of myeloid cells. She joined the University of Miami Silvestre Comprehensive Cancer Center as an assistant professor in December 2022.
Dear Dr. Defne Bayık and participants, welcome all of you to our Bioinfocongress IV event. Dr. Defne Bayık will give us a talk today on consideration of biological sex for immunotherapy of glioblastoma. At the end of the presentation, we will have a question-answer section prepared by our team and consisting of questions from our participants. Dr. Defne Bayık, welcome back.
Betül, thank you very much for that very kind introduction, and I thank you very much for your invitation to give this talk. I'm very excited to be here with you guys virtually and looking forward to connecting afterwards as well. So as Betül alluded to, I am a very new assistant professor who started I started my lab just 3 months ago, and we are interested in understanding how biological sex could be an important factor determining immune response in brain tumors and how we can take advantage of that for immunotherapy of glioblastoma. So just to briefly outline my presentation, I would like to very briefly introduce the role of the immune system in glioblastoma in brain tumors. I would like to briefly recap some of my postdoctoral work, which focuses on sex differences in myeloid-derived suppressor cells and how epigenetics is important for their regulation. And I would like to talk about 3 ongoing projects in my laboratory that focuses on myeloid cell regulation and aims to identify new therapeutic targets for brain tumors. So what is glioblastoma? Glioblastoma, or in short GBM, is grade 4 glioma. So this is the most common primary malignant brain tumor. This is a very aggressive brain tumor, and the standard of care has not changed over a decade. When we look into what constitutes standard of care, it is surgical resection, followed by radiotherapy and chemotherapy, which is temozolomide. And these treatment modalities lead to a median survival of somewhere around 14 to 16 months. More recently, there are other treatment strategies that are getting incorporated into glioblastoma treatment, and one such strategy is tumor treating fields that manage to extend the survival duration up to 20 months. So this is still not a great outcome for these patients. There is a growing appreciation for the use of immune therapies in many solid and hematological malignancies, but these strategies did not lead to a good therapeutic response in patients with glioblastoma. As many of you are aware, anti-PD-1 and anti-CTLA-4 and the scientists who discovered these molecules got Nobel Prize a couple of years back, are now becoming standard of care. But these two reagents did not lead to any extension of survival in patients with glioblastoma, and the response rate is somewhere around 2%. So there is really great need to understand what immune mechanisms are involved in treatment of glioblastoma and application of immune therapies. And there are several considerations for that. So this is a tumor of brain, so there is blood-brain barrier that prevents effective accumulation of therapeutics in brain tumors. There are stem cells that lead to therapeutic resistance, and after surgical removal, the presence of these cells lead to recurrence. But there is also the immune system. So if we understand what kind of immunological changes are happening in the tumor microenvironment, we can target these for effective treatment strategies. And if we look at glioblastoma, there are many immunological changes. So these are data from patients, and what we observed was that patients with glioblastoma had very low levels of T cells, almost equivalent of patients with HIV. So these T cells usually end up in bone marrow, sequestered in bone marrow, and this is induced by the tumor itself, as well as the treatment, the chemotherapy we use in patients. In the tumor microenvironment, what we observe is accumulation of immune suppressive type of cells, or exhausted T cells that cannot fight with tumors effectively. Glioblastoma is not unique, but it's different in the sense that there is very heavy myeloid cell presence. So if we again look into the tumor microenvironment, there are resident microglia cells, tumor-associated macrophages, and my population of interest, myeloid-derived suppressor cells. Collectively, these three myeloid cell populations can constitute up to 30 to 50% of tumor bulk. So there are almost as many immune suppressive myeloid cells as tumor cells in this microenvironment. And when I joined my postdoctoral fellowship, we had studied the role of myeloid-derived suppressor cells and patient outcomes. And what we observed was that in patients, if there's a reduction in the frequency of myeloid-derived suppressor cells after initial treatment, these patients have tendency to do better. If we observe an increase in the frequency of circulating myeloid-derived suppressor cells over time, these patients usually have a poorer outcome. So these studies really defined myeloid-derived suppressor cells as biomarkers of disease and biomarkers of brain tumor in particular. But at the time, we did not really study the individual role of myeloid-derived suppressor cell subsets. So what do I mean by that? So when we look at the MDSCs, there are broadly categorized into two subsets as monocytic MDSCs and granulocytic MDSCs. Both these cells are generated in the bone marrow. They are immature, but they have different properties. We can use phenotypic markers to define these cell populations. There are different markers we use for human and mice, and this helps us to discriminate monocytic subsets from granulocytic subsets, or polymorphonuclear is another name for this subset. We know that both subsets are capable of suppressing T cell response, and that is the functional role of MDSCs, the suppressed T cells. But recent studies suggested that they can undertake additional roles in the tumor microenvironment. Specifically, a study in the context of breast cancer showed that monostic MDSCs can promote stem cell maintenance, cancer stem cell maintenance, whereas granulocytic MDSCs can promote differentiation, metastasis to distant sites. However, the role of these cells were not defined in the context of brain tumors. So for the purpose of this talk, it's important to know that glioblastoma is a disease in which we observe sex differences. So if we look at the incident rates across age groups, what we observe is that 60% of all glioblastoma patients, or all glioma patients, which are the brain tumors, are males. So this is a male prevalent disease. If we look at the outcome, we observe that male patients have a poor outcome compared to female patients. So what is the basis of that? Looking epidemiologically, this is not unique to glioblastoma. If you look at the various diseases from autoimmunity to cancer, we observe a strong sex bias. Females overall are prone to autoimmune diseases and inflammatory conditions, whereas very non-reproductive cancers such as liver, lung, are more common in males compared to females. And this is something we can also see in immunocompetent models as well, or model in immunocompetent models. So in lab, we frequently use syngeneic mouse glioblastoma cell lines that we implant orthotopically into brains of mice. If we use immunocompetent mice, what we observe is that, and I do realize this data points don't look well, is that male mice, which is I try to highlight with my pointer, succumb to disease earlier than female mice. And this is consistent across different models of glioblastoma, which have different driver mutations. If we repeat the same experiment with NSG mice, which lack intact immune response, we observe that there is no difference between the median survival of males versus females, suggesting that there is a role for the immune system. So that's what I tried to identify as a postdoctoral fellow. And long story short, these results got published several years back, showed that there's increased accumulation of monocytic subset of myeloid-derived suppressor cells in the tumor microenvironment. And these cells were more abundant in males compared to females. And we observed this both in animal models as well as patients. We observed a role for granulocytic MDSCs in females. Specifically, there was an increased expansion of these cells peripherally in females. And in patients, we observed that high levels of granulocytic MDSC, or score for a granulocytic MDSC, correlate with a worse outcome. So we wanted to target these differences with the hypothesis that if we target the sex differences, we can get differences in immune therapy response. So we performed gene expression analysis and then combined this with a drug prediction analysis to define targetable pathways. And what we ended up observing kind of validated our hypothesis. So when we targeted monocytic MDSCs non-specifically with a chemotherapy named fludarabine, we observed that male mice with glioblastoma tumors had a better survival, whereas this treatment had virtually no effect on female mice with tumors. In contrast, if we block IL-1 beta, which was mainly secreted by granulocytic MDSCs, this time, there was no outcome differences in males, but in females, there was a significant extension of survival, suggesting that neutralization of IL-1 beta could be a good therapeutic approach for females and not males. But at the time, we really didn't address what drives this differences in MDSC subsets. So that's what I tried to do later in my postdoctoral career by using adaptive transfer approaches. So in this setting, we implanted mice with tumors, and then we adaptively transferred MDSCs to these tumor-bearing mice to measure the survival outcomes. As we were doing that, we used matched sex donor and recipients, as well as donor and recipients from opposite sexes. So the aim of this this design was that by focusing on the donor sex, we tried to identify cell intrinsic regulatory mechanisms Whereas by looking into recipient sex, we wanted to understand host factors or environmental factors that are unique for males versus females. So in this series of Kaplan-Meier curves that I'm going to walk you through, it actually turned out that there was a role for both cell intrinsic and environmental regulation. So the first pair we are looking into here is male male donors that go into male recipients. And in this setting, what we observed was that transfer of monostic MDSCs significantly accelerated tumorigenesis. That is blue line compared to the black line. We didn't observe the same tumor driving effect when we transfer granulocytic MDSCs. That is orange line compared to black line. When we use female donors and put them into male recipients, we observed a similar effect in which monostic MDSCs drove the disease and accelerated the tumor progression, but the magnitude of effect was much limited. In contrast, when we put these cells into female recipient mice, there was virtually no effect in the survival outcome. So collectively, these experiments suggested that monostic MDSCs were the tumor driving factors, and it didn't matter whether they originate from a male or a female mice, but they were only able to exert this activity in a male microenvironment. So we wanted to understand what makes a monostic MDSC a tumor driver compared to granulocytic MDSC. And to do so, these cells are generated in the bone marrow. So we sorted them out and collaborated with a researcher from Case Western University, Peter Scaccieri, to perform ataxic. So this is a method in which we can detect open chromatin regions by using linker sequences. So when we did that and brought to characterize the epigenetic architecture of these cells, we observed that the main driver was cell identity. So what I mean is that in this principal component analysis, you can observe that 70% of the variation in the epigenetic structure was introduced by cellular identity. Monostic MDSCs were clustered separately from granulocytic MDSCs. There was still a role for sex that accounted for 27% of the variation. So since the cell identity was the main driver of the epigenetic profile, we wanted to understand what genes were located in these open chromatin regions in the monostic MDSCs and which pathways they were linked to. When we did this analysis, our top pathway was cell adhesion. So cell adhesion-related genes were more available in monostic MDSCs compared to granulocytic MDSCs. To further validate these observations, we actually went ahead and did a surface flow staining for cell adhesion molecules, which are integrins. And we observed that integrin beta-1 and integrin beta-7 were highly expressed in monostic MDSC subset, but not in granulocytic MDSC subset. We wanted to understand whether this expression had a role. So we did the same adaptive transfer experiments I shared with you earlier. But this time, we blocked the surface integrin beta-1 or integrin beta-7 before transferring these monostic MDSC into recipient mice. So in this setting, the cells get pretreated and not the mice. And in what we ended up observing is that when we block integrin beta-1 on the surface of monostic MDSCs, they lose their tumor driving effect. So this is solid line compared to dashed blue line. In contrast, when we block beta-7 on the surface, we don't abrogate their tumorigenic function. These results suggested that integrin beta-1 could play a role in the trafficking of monostic MDSCs, or at least their function as tumor-promoting cells. So, as I briefly mentioned earlier, monostic MDSCs are immature cells that are capable of differentiating into macrophages or dendritic cells when they encounter right cytokines. And since macrophages are very prevalent in the tumor microenvironment, and tumor-associated macrophages are actually main drivers of immune suppression, we wanted to understand whether by blocking integrin beta-1, we were abrogating their differentiation into macrophages. So again, these are the adaptive transfer experiments I shared with you earlier. vehicle are control mice, and we transfer isotype treated monostic MDSCs or monocytic MDSCs that were pre-treated with an anti-integrin beta-1 blocking antibody. When we look at the frequency of different myeloid cell populations, immature cell subsets on top and more differentiation subsets in the bottom, what we observed is that when we transfer isotype-treated monostic MDSCs, there's an increased prevalence of macrophages. Whereas when we block integrin beta-1, this effect is diminished, and in fact, we actually observe more dendritic cell subsets. So this really suggested that we are abrogating monostic MDSC macrophage communication by blocking integrin beta-1. These are all preclinical studies, and obviously we always have in mind to translate these observations, basic observations, into the clinic. So we wanted to therefore understand whether integrin beta-1 expression and these differences we observe in mouse are relevant to patients. To do so, we actually turned into publicly available single-cell data sets and created signatures for monostic MDSCs or granulocytic MDSCs. Then we compared the integrin beta-1, beta-2 as a control, and beta-7 expression levels between these two subsets. So the patient data kind of validated what we were observing in preclinical models. Both integrin beta-1 and beta-7 expression levels were higher in the monostic subset compared to the granulocytic subset. We also confirmed this by performing flow cytometer staining. We got patient specimens, blood and tumor specimens from patients with glioblastoma and stained for integrin beta-1 and beta-7 on the surface of the cells. We observed that there were very high levels of integrin beta-1 expressed on the monostic MDSCs compared to granulocytic MDSCs. So, to our surprise, integrin beta-7 was not highly expressed by either cell population, and it was not different, which was kind of controversial compared to the RNA expression levels, but we did validate this with two separate antibodies. So this really is a take-home message that RNA and protein don't always correlate. But collectively, combined with the mouse data, these results suggested that integrin beta-1 could be a good candidate to target monostic MDSC activity in patients with brain tumors. The only problem is that integrins are very cardinal. They have a very central role in human physiology. So though there are inhibitors and though they are in clinical trials, they are not the best drug candidates. Therefore, we went back to our cell adhesion signature and tried to understand what other genes were related to integrin beta-1. And one such gene was dipeptidopeptidase 4, or DPP4 in short. You can appreciate that if you look at the peak graphs here. These peaks are more prominent in monostic MDSCs compared to granulocytic MDSCs, suggesting that this gene is getting actively transcribed. And in fact, when we look at the RNA level, we see that monostic MDSCs has significantly higher levels of DPP4 expression compared to granulocytic MDSCs. And we also wanted to again validate this in patients and looked for their expression levels in patient monostic MDSCs versus granulocytic MDSCs using the single-cell sequencing data sets. And again, DPP4 was highly expressed by monostic MDSCs, but not granulocytic MDSCs, which is also shown here. So this top purple part is the monostic MDSCs, whereas this bottom gray part is the granulocytic MDSCs. So is DPP4 a relevant target? And it appears so. If we look at the expression levels across tumor grades using the Cancer Genome Atlas, we observed that patients with grade 4 tumors, which are glioblastoma, had significantly higher levels of DPP4 expression compared to patients with lower grade tumors. Furthermore, only in the glioblastoma subset, if we look whether DPP4 is predictive of survival, we observed that high levels of DPP4 correlated with worse outcomes, suggesting that DPP4 might have a tumor-promoting effect. So what is DPP4 or CD26? So this is a multifunctional protein that is present on the plasma membrane. There's also a soluble version of DPP4 that is secreted from these cells. So on the surface of the cells, DPP4 forms interactions with different kind of proteins, including integrins, and activates phosphorylation pathway. Soluble DPP-4 is important for regulation of glucose metabolism. DPP-4 is an exopeptidase which has ability to cleave cytokine, chemokines, and incretin hormones. And this cleavage through incretin hormones is important for glucose-insulin balance. And in fact, there are many clinically approved inhibitors against DPP-4 that are used in patients with type 2 diabetes. So there are very commonly used drugs that can target DPP-4, and they relatively have good safety profile. So we were very excited about that because the standard of care of glioblastoma, as I mentioned earlier, has not changed over a decade. So the main treatment approach is to do clinical trials. And if we have drugs that are readily available in clinic, we can repurpose them for glioblastoma use. That's why we wanted to understand whether targeting DPP-4 could lead to therapeutic opportunity for glioblastoma, and we turned into our syngeneic models. So again, if we implant immunocompetent mice with a syngeneic model, GL261 in this case, and treat them with a DPP-4 inhibitor sitagliptin, we observe that these mice that receive drug have a much better outcome compared to those that get the vehicle. If we do the same experiment in immunocompromised animals, we fail to extend survival by any means, suggesting that the effect is likely through regulating the immune response. And in fact, when we analyze the tumors from this vehicle versus the sitagliptin-treated mice with flow cytometer, we observed that there was a reduction in tumor-associated macrophages. So what else? Like, is this related to myeloid cells? It doesn't appear to be so. So these are more recent studies that we are currently pursuing. What we observe in vitro, at least, is that if we treat CD8 positive T cells with sitagliptin, there's an increased proliferation. So there's a potential that we are activating T cells as well. But when we looked into tumor microenvironment, that's not what we observed, because there was still slight reduction in the frequency of CD8 and CD4 T cells. It wasn't statistically significant. But when we also look at the activation status by measuring the levels of CD69, which is a marker of T cell activation, and PD1, which is the checkpoint, a marker of T cell exhaustion, sitagliptin treatment did not change the expression levels. So then the effect is, at least the anti-tumor effect we see is mainly through myeloid cell modulation. And it appears to be the case. So to better understand the outcomes, we did a migration assay to see whether we are actually preventing cells from reaching to the tumor microenvironment. And in this in vitro migration assays, we observed that sitagliptin treatment significantly reduced the migration of monocytic MDSCs towards tumor cells. We also observed that there was a reduction in the phosphoric levels in monocytic MDSCs treated with sitagliptin, which kind of shows us that the drug is effective. So our current studies are trying to understand what DPP-4 does in the tumor microenvironment. I mentioned to you earlier that it's an important molecule that regulates insulin glucose metabolism. So in this tumor-bearing mice, we looked at unfasted insulin and glucose levels, and we didn't observe any change with sitagliptin treatment, which is to be expected because this drug only works in patients with aberrant insulin glucose metabolism, and it doesn't necessarily change the baselines in healthy individuals or individuals with no metabolic diseases. We also did with a targeted cytokine analysis from plasma of these mice, and we observed that there was a reduction in the CXCL5 levels. So CXCL5 is a chemokine that involves recruitment of immune cells, granulocytic cells, and macrophages. And in patients, what we observed was that high levels of CXCL1 correlated with worse outcome compared to patients with low levels of CXCL5. So in conclusion, these published and unpublished results suggested that there was an epigenetic regulation that affected the behavior of myeloid cells. Specifically, we observed that the monocytic MDSCs had increased cell adhesion pathway availability, And this led to higher levels of surface integrin beta 1 and DPP4 expression. When we block integrin beta 1 or DPP4, we were able to reduce the frequency of tumor-associated macrophages in preclinical models. So our current research focuses on understanding the role of DPP4 in myeloid cell regulation and also trying to understand the role of CXCL5. So CXCL5 is a ligand for the receptor CXCR2, and we want to understand whether if we block this axis, whether we can further alter the immune microenvironment and get favorable outcomes. So what else we are working on? So we are, I mentioned to you that there is a role for intrinsic regulation and environmental factors. So these results that I shared with you were intrinsically, epigenetically making monastic MDSCs more proteinogenic. But what are the environmental factors that contribute to differential MDSC subset activity? So there is a growing appreciation for the involvement of the neural system in regulation of tumor genesis. This is especially a very hot field in the context of brain tumors, but there's new research suggesting that this is also applicable to peripheral tumors. So several groups, Michel Monge's group and Frank Winkler's group, has shown that tumor cells can integrate into neuronal networks and can respond to electrochemical cues. Furthermore, factors secreted by neurons or other neural cells, such as astrocytes, can alter the tumor cell behavior. But what is not known in this field of cancer neuroscience is that what role the immune system plays. And there is a reason why we are interested in, because several studies in inflammation and autoimmunity have suggested that neurotransmitters can regulate the behavior of immune cells. Specifically, these immune cells were shown to express receptors for various neurotransmitters. And if we alter the neurotransmitters, we can control the inflammation. And when we had done gene expression profiling of monastic MDSCs and granulocytic MDSCs, which led to identification of drug candidates that I shared with you earlier, one of the top differentially expressed genes between these two subsets was GABA receptor B. So GABA receptor B is a G protein coupled receptor, so it has a signaling function. These cell subsets were not expressing GABA receptor A, which is the ion channel. And in fact, when we looked at the drug prediction algorithm and tried to understand which drugs can readily affect the behavior of granulocytic MDSCs, one of our top drug classes was GABA modulators. So this was very interesting to us and kind of brought up the idea whether we can modulate GABA levels to control the activity of granulocytic MDSCs. One thing before I move into my next slide is that this drug prediction algorithm does not show directionality, meaning that these drugs do not necessarily block the function of granulocytic MDSCs. So that's what we tried to do, and we were very optimistic and hope that we can target GABA to block granulocytic MDSCs and use the GABA analog pregabalin. So pregabalin, again, is a clinically used GABA analog that is important to manage neuropathic pain. When we treated our mice with this drug, tumor-bearing mice with pregabalin, and when we hoped that we would block the granulocytic MDSC activity, we actually observed an opposite outcome. So female mice that received this drug succumbed to disease earlier than males, earlier than vehicles. In contrast, we didn't see the same effect in male mice. As always, we repeated the experiments using NSG immunocompetent mouse models. And again, we didn't observe differences in survival outcomes, suggesting that pregabalin treatment was driving disease in females in an immune-dependent manner. So we looked at whether there's a role for granulocytic MDSCs. And we did so by getting these cells out from bone marrows and stimulating them with GABA. Stimulation with GABA led to moderate changes in the expression profile, and we ended up observing an increase in 130 genes. But this was unique to female granulocytic MDSCs. When we repeated the same experiment with male granulocytic MDSCs or monastic MDSCs from either sex, we observed that there were no differences in the expression profile. So therefore, we focused on this as GABA response in female granulocytic MDSCs and looked what genes were upregulated and which pathway that was linked to. So one important pathway that came up from this screen was antiviral innate immune response. To further validate what was unique to females, we compared the expression profile of GABA-treated granulocytic MDSCs from females and males, and we observed that complement 1Q was highly expressed in female granulocytic MDSCs that encountered GABA. So you guys might remember this from your undergrad course, which is one of the least favorite topics in immunology. The complement system is an important innate immune system. Just briefly to recap the complement system, there are 3 ways that will lead to complement activation, and the one we are referring to here is the classical pathway, which was initiated by complement 1Q. So complement 1Q forms complexes with C1R and C1S, and collectively this complex recognizes the antibodies that are circulating in patients in host, or apoptotic cells, which could be more relevant in the tumor microenvironment. This C1Q immune complex formation leads to the cascade of cleavage events and production of additional complement molecules such as C3A and C5A. So this is important for opsonization and fighting against infections, but there could be a role in the tumor microenvironment as well. Several studies suggested that endothelial cells and tumor cells express receptors for these complement molecules, and binding of these C1Q, C3, or C5A to these receptors can lead to increased angiogenesis or can alter tumor cell phenotypes by increasing their proliferative capacity or stemness. So we wanted to understand whether complement 1Q was indeed a relevant target. And we return back to our preclinical models. So we wanted to do this by an unbiased way. Therefore, we sorted out monocytic MDSCs, granulocytic MDSCs, and macrophages from tumor microenvironment based on markers that they express. We also sorted out monocytic and granulocytic MDSCs from peripheral blood of mice with tumors. So this analysis, which is single-cell analysis from these populations, led to the identification of 9 separate clusters. And when we looked at the gene expression levels, we observed that tumor-infiltrating myeloid-derived suppressor cells has significantly different expression profile than those in the peripheral circulation. We also again identified sex as a driver of expression profile, and different clusters were represented at different levels between males and females. So what genes were differentially regulated within these pathways, within these clusters? And we observed again that subunits of complement 1Q were highly expressed by tumor-infiltrating granulocytic MDSCs, which are likely get exposed to GABA compared to any other cluster. I would like to highlight that IL-1 beta again emerged as a therapeutic target, which is expressed mainly by peripheral granulocytic MDSCs, and that really matched our earlier data, which was very reassuring. So since we again identified complement 1Q as a therapeutic target, we wanted to understand whether these RNA levels changes correlate with a disease outcome, or whether it has physiological protein level outcome. And we did an ELISA to measure C1Q levels between male mice and tumors and female mice with tumors. And collectively, we observed that female mice with tumors had significantly higher levels of complement 1Q compared to male mice with tumors. There are transgenic mice that we can use, so that's what we did. And we collaborated with a researcher who had those, and we implanted C1Q knockout mice or wild-type mice with tumors, with brain tumors. And in this setting, we observed that there was no difference in the survival duration of wild-type male mice with GBM tumors or C1Q knockout mice with GBM tumors. In contrast, in females, we observed that C1Q knockout female mice survive for a longer duration compared to wild-type litter mates. So collectively, these results indicated that C1q knockout mice. C1q might be a tumor promoting factor, specifically in females and not in males. We wanted to validate these observations in patients, and we did so by again looking into two publicly available databases, the Cancer Genome Atlas and Chinese Glioma Atlas. In this setting, we observed that C1q expression level is not predictive of outcome for males, but in females, we observed that high levels of C1q correlate with a worse outcome compared to patients with low levels of C1q expression. So why are we interested in C1q? Well, there are several reasons, but one of which is that, again, there's a clinically used inhibitor that we can repurpose. C1 inhibitor is an inhibitor of C1 esterase, and it is used for treatment of angioedema. So in very preliminary studies, we grabbed this inhibitor and tested in our in vitro in our bone marrow cultures. So we sorted out bone marrows and put them in MDSC polarizing conditions and included C1 inhibitor in our cultures as well. So in this setting, we didn't observe any phenotypic changes of a relative representation of monostic MDSCs, granulocytic MDSCs, but what we observed was a functional change. At the very beginning of my talk, I mentioned that the main function of MDSCs is their ability to suppress T cells. So when we do a T cell proliferation assay, in which we co-culture MDSCs and T cells and measure T cell proliferation by activating T cells and looking for their division rate, we observed that we could reverse the suppressive capacity of granulocytic MDSCs. So here the black bars are controls where we observe proliferation of T cells. Blue ones are the ones that are cultured with monostic MDSCs, in which we observe a reduction in the proliferation capacity of T cells, and C1 inhibitor had no effect on this compared to vehicle control. But when we look into orange bars here, we observe that vehicle-treated granulocytic MDSCs are more capable of suppressing the proliferation of T cells compared to those that encounter C1 inhibitor. So this really brings up this hypothesis that we are currently pursuing. We think that the GABA produced by astrocytes or neurons in the tumor microenvironment functionally alter tumor-infiltrating granulocytic MDSCs, which express GABA-B receptors. We observe that this GABA-B receptor activity leads to increased C1q production, and this somehow drives brain tumors. And we also believe that this is a dominant mechanism in females, which is characterized by higher granulocytic MDSC activity. So what are we trying to do now? We are trying to understand the role of GABA in myeloid cell regulation, as well as in other immune cell types. We are trying to understand whether it's the GABA or GABAergic neuronal activity that leads to these functional changes. We are trying to, again, identify the cell intrinsic changes in granulocytic MDSCs upon exposure to GABA and pathways that are activated. So what about C1q? Yeah, we are very interested in understanding what it does in the tumor microenvironment. And we would like to obviously therapeutic the target and incorporate that as part of the standard of care in brain tumors. So with that, I want to leave ample time for questions. So I will acknowledge my postdoc mentor, Justin Latia from Cleveland Clinic, who was very supportive of my career and my transition as an independent investigator. We had a fairly large lab, and not all these people were present at the same time, but I had the privilege to work with this brilliant scientist across 5 years, I guess, that I was in Justin's lab. It takes a village to do high-impact studies, so it's really important to collaborate with clinicians and scientists with different expertise, and this is what happened. You can see that there were many internal and external collaborators that help with different aspects of this project, and Antonio Arborone is my actually faculty mentor who is helping me to start my own lab. So we are a very new lab. I mentioned as earlier, we started our lab 3 months ago, and I was very lucky to be able to start working with two great scientists, Oriana Teran Pumar, started a month ago, and Asmita Pathak will soon join. with that, before moving on to questions, I actually want to switch to Turkish and want to make some remarks. So those who don't speak Turkish, I apologize. Arkadaşlar, hepinize katıldığınız için çok teşekkür ederim. Benim söylemek istediğim, yaşadığınız zorlukları farkındayız. Biz özellikle yeni veya Türkiye'den ayrılalı çok olmayan bilim insanları olarak durumu farkındayız ve size elimizden geldiğince yardım etmek istiyoruz. Bunun hani ekonomik zorluklar olduğunu farkındayız, eğitimsel zorluklar olduğunu farkındayız, pandemi, daha sonra deprem nedeniyle. Bu yüzden eğer olur mu olmaz mı bilmiyorum ama Hakan Ceylan ve Murat Yıldırım'la bir platform başlatmayı deniyoruz. Dediğim gibi belki başarırız, belki başaramayız. Çok yeni bir oluşum bu. Ama bu platformla size staj olanakları yaratmak istiyoruz veya en azından sizi Türkiye'de bulunan veya yurt dışında staj olanaklarıyla eşleştirmek istiyoruz. Böyle bir misyonumuz var. Bu yüzden aktif olarak diğer araştırmacılara hem yurt içinde hem yurt dışındaki araştırmacılara ulaşıyoruz ve bir platform kurmaya çalışıyoruz. Yine bunun bir parçası olarak sizin başvurularınıza yardımcı olmak istiyoruz. Yani bunu daha eğer büyüyebilirsek tabii daha iyi bir şekilde yapabiliriz ama en azından hani o kurulana kadar da bize de ulaşabilirsiniz. Mesela CV hazırlama gibi bir yardımda bulunabiliriz veya özgeçmişinizi özgeçmişinizi hazırlamanıza yardımcı olabiliriz veya nasıl e-mail atılır, Amerika için ayrı, Avrupa için ayrı beklentiler oluyor. Bu gibi şeylerde size danışma için yardımcı olmaya çabalayabiliriz. Ekonomik zorluk tabii ki çok önemli bir etken burada. Bizim zamanımızda belki bu kadar ciddi bir ekonomik kriz yoktu. Sizin kadar etkilenmedik. Ama size maddi destek imkanları yaratmak istiyoruz. Ne yazık ki çoğu enstitüde staj için gelecek öğrencilere maddi bir yardımda bulunma olanağı yok. O yüzden bazı yerlere ulaşarak belki programlar oluşturmak ve staj için destek sağlamak gibi bir misyonumuz var. Dediğim gibi bu çok daha kurulma aşamasında olan bir şey. O yüzden ne kadar başarırız bilmiyorum. Ama sadece yanınızdayız. Bunu bilmenizi istiyorum. Ve belki bu kuruluna kadar mesela ben kendi adıma söyleyeyim. Elimden geldiğince size yardımcı olmak isterim. O yüzden her zaman bana Twitter'dan ulaşabilirsiniz veya her zaman bana e-mail atabilirsiniz. Okumamı istediğiniz bir şey varsa veya yorum yapmamı istediğiniz bir şey varsa ben yardımcı olmaktan çok gurur duyuyorum. Hakan ve Murat'ın da muhtemelen aynı şekilde hissettiğini düşünüyorum. Hepimiz çok farklı alanlardan gelen insanlarız. Farklı dünya görüşü, farklı politik görüşü, farklı sosyal görüşü olan insanlarız ama aynı çatında altında buluştuk. Hepimizin bilime olan inancı var ve hepimizde size yardımcı olmak gibi bir misyonu var. Bu da aklınızda bulunsun. O yüzden again, switching into English, I would be happy to answer any questions you have regarding the slides I presented today or anything else I can help with. Looking forward to the discussion. Thank you again for inviting me.
Merhaba hocam. Öncelikle desteğiniz için gerçekten çok teşekkür ederiz, minnettarız. Ben de şu anda İngilizce olarak devam ediyorum. I appreciate your wonderful presentation, Dr. Defne Bayık. If you wish, we can go to the question and answer section prepared by our team and consisting of questions from our participants.
Sure, would love to.
Okay. Our first question is, gonadal sex steroids are found in sex chromosomes and affect on neuroanatomy and neurochemistry. For this information, can we say glioblastoma treatments can be related to connectivity among neurobiological changes but not hormonal changes?
Yeah, so that's a wonderful question. We actually don't know the answer. Let me start with that. So they're not just in glioblastoma, but the sex differences are getting extensively studied across tumor types. And we actually know that androgen receptor signaling could play a potential role in the regulation of mostly T cells, to be honest, not myeloid cells, but T cells and their anti-tumorigenic function. That's something I am very much interested in studying because it's been shown that estrogen and progesterone, more so progesterone, is linked to neuronal regulation. So there could definitely be link between hormones and their, direct effect on the immune system or in the microenvironment, and that could lead to potential outcome differences in glioblastoma. With that being said, I will also tell that the sex differences in incidence is considered across age range, meaning that even early puberty to very late in life, there is a difference between males and females, suggesting that the hormones may not driver. So I don't think I know it doesn't answer the question all that well. I think bottom line is that we don't have a good idea how the whole equation works, and that's something I'm very much interested in studying.
Thank you for your answer. Next question is, the reason that you have been used GL261 type mice because they have highly immunogenic relativity to human GBM, right?
Actually, no. So I will tell you, if anything, GL21 model is not the best model. The reason being that it's a hypermutated mutated model. So GL261s are generated by random mutagenesis. And in patients, what we observe is that there are very limited mutations in patients with glioblastoma. The mutation burden is very low. So that's why it's not the best model to use. But that's also why we use multiple models in the lab, such as SB28 and KR158, which have targetable mutations that are identified in patients with glioblastoma. I will tell you, as in many cancers, modeling glioblastoma is very hard in mice. And that's kind of why we always try to validate whatever results we get in preclinical models. We try to validate in patients to confirm that those mechanisms are conserved between the species, and models are indeed good to study those mechanisms.
Thank you so much. I'm moving on with the next question. Hormonal estrogen support to men. High probability we see hormonal changes on sex-based behaviors, but in social cultural frame. These behaviors can be reduced for the very good treatment's efficiency.
Yeah, I guess if I understand the question right, the question is the impact of social sex behavior. I guess the difference between gender and sex, which I know we don't have the same concept in Turkish, toplumsal cinsiyet ve biyolojik cinsiyet. So again, I don't know how much roles does the hormones play in this equation. I don't think they are the major drivers. I think the chromosomal or epigenetic drivers are the main effectors of the outcome in the context of glioblastoma. With that being said, that's actually a very important point because there are several studies that suggest gender differences are important for tumorigenesis as well. So one example I can give is that males are more likely to smoke than females, and that is a major contributor of lung cancer incidence. So how do you discriminate between gender behavioral differences versus biological sex? That's tough. I mean, I think in mouse models it's obviously easier because there are not many gender differences. Certainly there's differences in aggression, that kind of stuff, but not life choices that would affect the glioblastoma outcome. So role of hormones still under investigation. I can tell you that, again, we have a study under review. A friend of mine led that study and focuses on androgen receptor, androgen signaling, and glioblastoma outcome. But I don't know whether estrogen has a role and how that plays into behavioral aspect.
Thank you for your response. I'm moving on with the next question. Glioblastoma has male gender incidence bias, but is there currently clinical trial data examining differential effects of treatment between genders?
Yeah, so Yes and no. That's what we are hoping to achieve. We are trying to initiate clinical trials by using this information. It's very hard to exclude the sex from a clinical trial because of regulatory reasons, but we are pushing. There's a very big push to account that when you measure treatment response. And there is no currently any. That's something we started doing, but there are no clinical trials that specifically measure sex differences in the context of glioblastoma. But recent studies for checkpoint inhibitors, again, which are getting widely used in lung cancer, melanoma, GI cancers, and those studies suggest that there is a difference in response rate based on sex, as well as there's a difference in the adverse reaction that is observed in patients. So clearly this is an emerging field, and we are just starting understanding how patients respond differently based on sex and what to expect.
Thank you for your answer. And next question is, how feasible is it today to independently investigate sex hormones and chromosomes for sex-dependent immunotherapy responses in GBM?
Yeah, that's a wonderful question. There are models to do that. And one such model is called 4-core genotype model. So 4-core genotype model is a model in which there are four genotypes. One of them is XX, the other one is XY. The other two is that XY without the SRI gene, which is the gene that promotes testicular development. So those XY minus SRI mice don't have testes, but other than that, they are chromosomally X and Y. And the fourth genotype is XX plus SRI. So basically, they move the SRI gene to an autosome in those mice. So those mice are chromosomally female, but they do develop testes. So those models are very useful to dissect the role of sex chromosomes versus sex hormones. But I agree that they are very hard to maintain. They are getting used more frequently. But that's the one mechanism we can use. The other mechanism that we can use, which is more feasible, is basically doing ovarectomy and castration and see how that could affect the outcome. But when we do that, that's obviously not the perfect model, in the sense that you do reduce the circulating hormonal levels, but you do not alter epigenetic or imprinting changes that happened early on life that is the result of exposing to sex chromosomes. But that's still a way, at least, to study the relative contribution of sex hormones versus chromosomes.
Thank you for your answer. Another question is, can sexually dimorphic genes play a role in stratifying survival outcomes in a large GBM patient dataset?
Certainly. I mean, I don't think anybody has done that, but that's certainly a possibility. And that's kind of from answering that from an immune perspective. We know that, for example, X chromosomes express many immunological genes. One such example is FOXP3, which is important for development of regulatory T cells, and toll-like receptor 7, which is important for innate immune regulation. And in fact, toll-like receptor 7 is one of the genes that escape dosage inactivation, meaning that females have higher levels of toll-like receptor 7 compared to males. So it's certainly a thing, something we should consider. We should look more deep into whether sex differences, sexually dimorphic genes, is a relevant contributor of patient outcomes. The other thing I will briefly touch on is that both X and Y chromosomes do express epigenetic modifiers. So even if we don't see maybe chromosomal level changes, these epigenetic modifiers are likely to regulate gene expression levels differently and lead to different outcomes. But we should definitely look into it.
Thank you so much. I continue with next question. The tumor microenvironment occurs by specific interaction between the tumoral and the host biological factors. For these relationship conditions that happen during medical treatments, is there a way to decrease tumoral function and parallel that increase the biological factors mechanism efficiency?
Is there a way to decrease tumoral function and in parallel increase the biological factors mechanism efficiency? Yeah, that's an intriguing question. I will say yes. And I don't think we know that. That's the bottom line of it. But again, going back to the GABA story that I shared with you, so that we initially tried out GABA analogs, hoping that that would be the mechanism, but which didn't turn out to be the case. But there are GABA inhibitors that we can use, and that would likely to decrease the tumoral function, but also affect the biological mechanism. But we haven't tested that yet. So if we use, for example, GABA production inhibitors or receptor antagonists, maybe we cannot do both.
Yeah, thank you so much. Do sexually dimorphic genes tend to enrich for immunological signatures in both gender?
Yeah, I think that kind of we touched on that a bit, that there are definitely immunological genes that are related to sex. But we understand that better in the context of autoimmune diseases. Again, going back to toll-like receptor 7, it's been linked to prevalence of autoimmune disease in females. That's we need to come up with those signatures for patients with tumors as well.
Okay. I appreciate for your answer. Another question is from YouTube. Does identifying predictive biomarkers of immune checkpoint inhibitors response for sex bias can be any benefit to cancer therapy?
100%, yes. The only problem in the context of glioblastoma is, as I briefly mentioned, that the checkpoint or the immune therapy strategies have failed in glioblastoma. So when you have a response rate of 2%, that's kind of not a good outcome to do it. But definitely for lung cancer, melanoma, that's something that should definitely be further pursued.
Thank you very much for your response. Another question is, considering the data of the Cancer Genome Atlas revealing different sex patterns and association to multiple cancer types, what is the hardness of coordinating a big amount of data?
Yeah, the biggest hurdle is how you collect the data, right? This data come across institutes, and you also don't know what this history of these patients, how the data is collected, how the specimens are processed. So I think the lack of multi-institutional standardized things is the main problem handling large data sets. We try to overcome that by generating our data. We have a large number of patients that are going through treatment in our institute, and we are. I mentioned that my faculty mentor he's very much interested in doing single cell sequencing with those patients and identify gene signatures that could correlate with outcome or identify treatment opportunities based on that signature. So that's what we'll keep doing. I think we'll generate our own large data to do so.
Okay, thank you so much for your answer. I'm moving over to the next question from YouTube. Nowadays, do we know how each of the MDSC subtypes contributes to pro-disease processes in men versus women will provide?
Yeah, we do and we don't. So I guess the question is how that is relevant in patients. So we know that they serve as biomarkers in many different tumor types. We know that each tumor type has slightly different association. For example, in pancreatic cancer, granulocytic MDSCs seem to be more important than monocytic MDSCs. In livers, the other way around, monocytic MDSCs seem to play a more bigger role than granulocytic MDSCs. So I think by studying the patient data more on different tumors, we can get a better understanding of how each subtypes interact with these different disease processes, environment, and tumors, and how that could be an important driver of the disease.
Thank you very much. Another question is, There is a complex interaction among sex-based behaviors, hormones, and genes, and as a result of this could have affected the adaptive immune system. Is it possible to collect and coordinate all of the efficient factors in one spot?
Patients are very complex, right? So it's very hard to I don't think we have all the data to make those correlations. So when patients walk into clinic, we don't, for example, measure their androgen levels or estrogen levels, and we don't see how that could play a role. But that's what we are hoping to do, in a sense that if we could identify female-male specific disease biomarkers, for me that's immune, probably for others that's like different contributors, different mechanisms, we could potentially address that. But I think it's just very hard to bring all this data together due to the patient variation, which is something we oversimplify using mouse models.
Yeah. Thank you so much. Another question is, apart from gender-related treatment response, is it necessary to evaluate pediatric patients separately in GBM?
100%. Sorry.
Are there differences in tumorigenesis in adult and pediatric patients?
Yes, 100%. And all the data I presented today is for adult brain tumors, not pediatric brain tumors. Pediatric brain tumors also have tendency to show sex differences. Again, it's unfortunately more common in male kids than female kids, but there are different driver mechanisms. It's a very much understudied area. We don't really actually have a good understanding of immune infiltrates in pediatric brain tumors, but there are definitely different mechanisms in place. So with those young adult and pediatric tumors are a completely different field of study.
I appreciate for your answer. Next question is, in the light of all this information, could sex consider as a biological variable for development of effective immunotherapeutic strategies against GBM as well as other cancers?
Yes, and that's what we try to do with the IL-1 beta as well. So the drugs we usually use are, again, clinically approved drugs for different purposes, so that we can basically repurpose them for anti-tumor. And we did try to initiate one such trial using IL-1 beta inhibitors. But the challenge is that you need an industry partner who is willing to do that. I'm happy to discuss the business aspect of the stuff, but what I can tell you is that we were about to start a clinical trial doing just that, but then the company backed down because their priorities have changed. So I think overall, is it possible? Yes, but there are some challenges to make that happen.
Okay, thank you. And next question is from YouTube. Functional and targetable sex differences present within certain myeloid populations. Can GBM infiltrating T cells actually mediate sex difference in overall survival?
Yes, the answer is yes. And I don't know whether you guys are familiar with the bioarchive. This is where people deposit their preprints before pre-review, which is a great way of sharing science freely before pre-review. So that's kind of the catch there. So not all the papers are great, but certainly that's a platform we commonly use to get the data out there. And Julian Lee is the first author of that paper that focuses on T cells and sex differences in the context of glioblastoma. I had the privilege to work with her as a postdoc. She was a postdoc in the same lab as I was, and you could find more information there.
Thank you. And another question is, we can examine androgen receptors due to the location of AR gene on the X chromosome. Depending on the different location, types of AR gene can cause a higher prevalence GBM in men. Based on the facts can be found AR antagonist-based treatments for men and women.
So is it the androgen receptor and how it affects how it can be combined with treatment? Yeah, there are androgen receptor blockers, right? So I know that there are studies now in BRAF melanoma that incorporate androgen receptor antagonists to increase the efficiency of immune therapies. Jennifer Vargo is the lead senior author on that paper. In glioblastoma, several studies have suggested that tumor cells can express androgen receptors, and that could be an important factor. We have not studied androgen receptor signaling so far. But that's a good point that we should definitely look into.
Thank you so much. We have last question. What advice would you give to students studying in scientific fields?
Yeah, so is the person who asked the question Turkish?
You can answer both. Yeah, it doesn't matter. Yeah, you can answer.
Okay, then I'll switch to Turkish. Yeah. Ne gibi şöyle scientific network'ünüzü genişletmeniz gerekiyor. Çünkü aslında akademinin içinde olunca insanın anladığı şeylerden biri sizin arkanızı kollayan birinin olması ne yazık ki çok önemli. Belki bunu hiç düşünmüyoruz ama danışmanınız olsun veya yurt dışında tanıdığınız biri olsun. Onların size verecekleri referans mektupları her şey için çok önemli. Bağlantı kurmak çok önemli. Bunun için hani siz zaten buraya gelip hani konferansa katılarak yapıyorsunuz. Virtual olsa bile bu yani mesela bana bir gün yazarsanız işte ben sizin işte buradaki konuşmanızı şey yapmıştım, bana mektup verir misiniz? Ben tabii falan şeklinde yaklaşırım ama en önemli şey ne yazık ki bağlantı kurmak. Aynı zamanda tabii yapacağınız kendinizi bilim açısından geliştirmeniz de çok önemli. Yeni teknikleri öğrenmek veya en azından hani teknik olarak bilmeseniz bir de hani alanlar nereye ilerliyor işte ne bileyim single cell sequencing mi, CRISPR mı, işte spatial transcriptomics mi? Hani hangi alanlara doğru böyle bir şey olduğunu anlamak ve o alanlara yoğunlaşmak gerçekten çok yardımcı oluyor. Çünkü o sayede daha hani daha iyi makale basabiliyorsunuz, daha etkili bilim yapıyorsunuz. Ama verebileceğim en büyük advice, sizi destekleyecek insanlar bulun. Mentorlarınızı çok dikkatli seçin. Sizi destekleyeceklerinden emin olun. Ne yapacaksınız? Yine kendimden örnek verecek olursak mesela ben doktoraya aslında Türkiye'de başladım. Ama benim danışmanım beni NIH'e yolladı doktoramı bitirmem için. Ve benim için o çok ilk kapıyı o şekilde açtı. Ondan sonra belki kalma niyetim yoktu. Hani hayat kalmaya şey yaptı, beni doğru itti. Ama onun o verdiği destek çok önemliydi. Mutlaka sizi destekleyecek birilerini bulun ve mümkünse sizi destekleyecek bir sürü insan bulun. Size verebileceğim en büyük şey bu olur.
I will switch to Turkish now. Hocam öncelikle tavsiyeleriniz için gerçekten çok teşekkür ederiz. Bizim için çok önemli. Ayrıca daha önce verdiğiniz destek için de teşekkürler. Yani biz Türk genci olarak gerçekten bunlara çok ihtiyacımız var. O yüzden gerçekten çok sağ olun. And I'm switching to English now. On behalf of myself and all our participants, I would like to thank you for spending your valuable time transferring your knowledge to us and for this great informative presentation.
Thank you very much for inviting me again, and looking forward to seeing you guys down the road. You are doing some great stuff here.
Thank you. I wish you healthy days.
You too. Bye bye.
At 4:30, Associate Professor Dr. Zeynep Baharoğlu will be with us to give a speech on epigenetic adaptation to antibiotics in gram-negative bacteria.
Bioinfocongress IV. Associate Professor Dr. Zeynep Baharoğlu. Epigenetic adaptation to antibiotics in gram-negative bacteria. After graduating from Pierre Loti High School in Istanbul, Dr. Zeynep Baharoğlu studied cell biology and microbial genetics at University Grenoble and University Paris Cité, respectively, in 2000. She spent her last undergraduate year at the University of California, Irvine, as part of an exchange program. She received her PhD from Sorbonne University in 2008 for her research on E. coli DNA replication and repair mechanisms in the CNRS. In 2008, she joined the bacterial genome plasticity team at Institut Pasteur as a postdoc. As a researcher at Institut Pasteur since 2014 and group leader since 2020. She has focused on the response of gram-negative bacteria to low-dose antibiotics and more recently on the impact of RNA modifications.
Dear Dr. Zeynep Baharoğlu and participants, welcome all of you to our Bioinfocongress IV event. Dr. Zeynep Baharoğlu will give us a talk today on epigenetic adaptation to antibiotics in gram-negative bacteria. At the end of the presentation, we will have a question-answer section prepared by our team and consisting of questions from our participants. Dear Dr. Zeynep Baharoğlu, welcome back.
Thank you.
If you wish, you can start your presentation.
Okay, thank you very much for the introduction. So I would like to thank the organizers for having invited me. It's an honor to be here, so I'm very happy to be here today. So thank you very much. As you said, I will present you today my research topic about antibiotic resistance and tolerance and involvement of epigenetic modifications of RNA. So I just would like to mention that a few words just about the earthquake that happened. So my family is from Iskenderun, and so I would like to say that my thoughts are with everyone who suffered from this, and I just wanted to share a nice photo to remember it, nice things, and to be to have some hope for the future. So, okay, so now I'm at the Institut Pasteur in Paris, France. So my group is studying bacterial responses to stress in the presence of antibiotics. So antibiotic stress is, of course, very important nowadays because of antibiotic resistance development. Because bacteria are becoming more and more antibiotic resistant. So in the right here, you can see the years of introduction of several antibiotics here in the market, and the years where resistant bacteria appeared. For example, here for tetracycline, you can see that tetracycline was introduced in the 50s, and in 1959 there were resistant bacteria which appeared, but now this happens even more frequently and in a shorter time. For example, here you need just for ceftaroline, it took only 1 year for resistant bacteria to appear, so it's really a major problem. So there is also tolerance to treatment for sensitive bacteria. I will talk about this a bit more in the next slides. But there can also be relapsing infections of susceptible bacteria, which are not resistant, but which still tolerate antibiotic treatment. And so it's a major public health issue. So in the lab, we are more concentrated in understanding the effect of antibiotic stress at antibiotic concentrations below the lethal concentrations. So we use antibiotics at doses that do not kill bacteria, which we call sub-MIC, for sub-minimal inhibitory concentration. And these kind of doses can be found in the environment because of industry waste, healthcare waste, or even farming and animal husbandry, where the waste containing antibiotics goes in the soil and in the aquatic environment at quite high doses, which are still below lethal doses for bacteria. And the bacteria can encounter these low concentrations of antibiotics in their environments. And these concentrations do not kill bacteria, of course, but they are stressing agents, and bacteria have to develop strategies to grow well in presence of this kind of stress. So it has been shown by different labs that these kind of sub-MIC antibiotics are stressors, and bacteria respond to these stresses. And there is a question about the fact that antibiotics grow in presence of low doses of antibiotics, and whether this can lead to antibiotic tolerance and resistance. So just a definition slide of words that I will be using in my presentation. So these are mostly about resistance and tolerance. So what is resistance? Bacteria are called resistant when when we need more antibiotics to kill them. So here you have the susceptible bacteria, where the MIC, minimal inhibitory concentration, is 2 here. And if you have a resistant clone, if resistant strain of bacteria, you need more antibiotics to kill it. The MIC is increased here to 62 in the example. So this is resistant. Unlike that, tolerance is a bit different. So tolerance does not change the MIC. So if you have a susceptible population or a tolerant population, their MIC is the same. So you need the same dose of antibiotic to kill all of the population. But the difference is that if you do a killing curve, so you take bacteria, you treat them with the antibiotics, and then you see the proportion of survival, you see that susceptible ones in green here die faster than tolerant ones. So here, for example, when you look at 6 hours of treatment, you still have some tolerant bacteria which have survived the treatment, while susceptible bacteria were cleared. So tolerant populations usually do not resist to higher doses, but die more slowly. And this is usually also, it can be measured in competition experiments. For example, in the lab, if you mix two populations at equal levels, and then you at equal proportions, sorry, and then you grow them for a number of hours, and then you measure which bacteria is present in the culture. You can see that one has invaded the culture, and it's the tolerant one usually, or the one that grows better, which is the tolerant one. So in the lab, we use as model the cholera pathogen Vibrio cholerae, which is a gram-negative bacteria. And we study its response to submic antibiotics, and we asked whether we could find some genes or that are important for growth and response of Vibrio cholerae to submic stress, submic antibiotic stress. So to do that, we take the Vibrio cholerae genome, and we use a transposon, which is called Mariner transposon, which can insert in the genome in a random way. So we build large libraries of Mariner transposon insertion libraries into Vibrio cholerae genome. And in these libraries, theoretically, we have for each gene at least one insertion in this gene. And when the transposon gets inserted in a gene, usually the gene is inactivated because there is some random chunk of DNA inside. So then we take these libraries and we grow them either without antibiotics or with different antibiotics at concentrations which do not kill bacteria. So we took half of the MIC. And we grow them for 16 generations, which is not very long for bacteria. If you're a microbiologist, you would know it's like an overnight culture. So it's not a very long evolution. And then we sequence the regions flanking the transposon, which gives us the locus where the transposon was inserted. So it means that this gene is inactivated. So for example, here after 16 generations without antibiotics, we have gene A, gene B, and gene C, in which we detect insertions in blue here. And if we look with antibiotics to the right, we see that in gene B, we have lost detection of insertions. It means that when there were insertions in gene B, when gene B is inactivated, the bacteria containing these insertions were lost. They died. So they couldn't survive the stress, the antibiotic stress. So by comparative analysis of sequences with antibiotics and without antibiotics, we can infer the genes which were important for growth in the presence of this antibiotic. It is not depicted here, but we can also have the opposite. So for example, we can have a gene C, for example, where the number of insertions would increase in the presence of antibiotics. And that would mean that the inactivation of this gene is beneficial, and bacteria with insertions in this gene grow better and invade the population. So we did that with two different antibiotics because they have different targets. So tobramycin, which targets the ribosome, and ciprofloxacin, which targets the DNA. And then we analyzed all the sequences that we have, and we end up with genes in which we have increased insertions or decreased insertions. So here I show volcano plots. So in the x-axis, to the right, it's the number of insertions which increase. and to the left, it's the number of insertions which decrease. And the y-axis is the inverse p-value, which means that the higher the gene is, the more significant is the result. So what we see here is that, for example, we see thus B here in the right side of the plot. It means that when we have insertions in this gene, thus B, the bacteria grow better in presence of tobramycin, which means that deletion of thus B is beneficial for growing in tobramycin. And to the other side, we have TGT here. When you inactivate TGT, bacteria get lost. It means that TGT is important for growth in the presence of antibiotic. And we did the same thing with ciprofloxacin. And in both cases, we identified many genes associated to RNA modifications. So tRNA modifications and ribosomal RNA modifications. So I will explain this more in the coming slides. But when we do gene ontology enrichment analysis of these genes, you can do it online. So you have a list of genes that have different levels of insertion. You take these genes and you see if there are significant changes in gene ontology groups classified by function. So for example, here you can see that with the red arrows, we can see that RNA modification genes appear in a significant way in this list. So it means that it's not just that we are seeing more RNA modification genes, it's that it's statistically significant. Okay, so before I go into RNA modifications, I would like just to show again the central dogma that many of you know, of course. So the central dogma is that we have DNA. The DNA gets transcribed into mRNA, messenger RNA, which gets translated by the ribosome to proteins. And so genetic mutations can happen in the DNA. They are hereditary, so they can be transmitted from generation to generation. And there is also epigenetic modifications. And for example, when we hear about epigenetic modifications, usually it's DNA methylation that we talk about. And so DNA can get methylated. And this is not genetic, it's not a mutation, but it's an epigenetic modification of DNA. And DNA methylation in bacteria was all associated with virulence, antibiotic resistance, cell cycle regulation, etc. But there is also modifications in RNA. So the messenger RNA can be modified. This is rare in bacteria. In eukaryotes, it's much more present. And other RNAs can also be modified, which are transfer RNAs and ribosomal RNAs, which compose the ribosome. So it can be methylations, but there are also other modifications. And so this kind of modifications in bacteria were very few phenotypes were described. Their role is quite not very well known. So as I said, there are different RNAs inside the cells. So inside bacterial cells, you have messenger RNAs, transfer RNAs, which bring amino acids for transcription to the ribosome, ribosomal RNAs inside the ribosomes, and some non-coding RNAs. So at least 3 of these RNA types are involved in translation, protein synthesis. So we could imagine that if RNA modifications are present or absent, that could impact tRNAs, mRNAs, and rRNAs, so protein synthesis. This is just to show you, to give you an idea where modifications can be. So this is ribosomal RNA on 2D depiction of ribosomal RNA. So this is the 16S ribosomal RNA. This is the 23S ribosomal RNA. And each colored square or round or triangle are different modifications that are present in ribosomal RNA. In E. coli, they are well described. They are mostly involved in the stability of the ribosome or the structuration of the ribosomal RNA, but for some, the function is not really well known. For tRNAs, so this is a tRNA. This is the anticodon of the tRNA, which binds to the codon. And here you have the amino acid that is attached to deliver the amino acid for translation elongation. So in E. coli, there were almost 80 modifications that were described for tRNAs. So many of these modifications happen at the anticodon loop. So here in the positions 34, 35, 36, 37. But you have also other modifications elsewhere on the tRNA. And what's interesting is that when you delete genes which do these modifications, usually you have no phenotypes, no growth phenotypes. So bacteria in the lab continue growing well like the wild type. They have no growth defects, except for 3 of them. So that was interesting because it could mean that these genes are not important during growth in rich media in the lab, but maybe they have some function in another situation or in response to stress. And since in our screen with antibiotics, we identified many of these genes. They are here in red square. All these genes, we have identified them in our screen as important during growth in the presence of tobramycin. So what we did is that we deleted each gene. So we did single deletion mutants in Vibrio cholerae of each gene. And we did some competition experiments. So we mixed the wild type and the mutant strain at equal proportions. We grow them together and then we plate and we see if one of the 2 strains wins or if they are still at equal proportions at the end. So this is presented here in these graphs. It's the competitive index in a log scale. So 0 means that they are still at equal proportions. There is none of the strain wins. And here, as you can see, it's the non-treated condition. So we just grow them in rich media. And as you can see, the majority of the mutants are at 0. It means that after the competition, they are like the wild type. They have no growth defect or they don't grow faster, except for a few of them which grow slower and which get lost. But then we did the same thing with the antibiotics in the presence of aminoglycosides, ciprofloxacin, trimethoprim, carbenicillin. So these are 4 families of antibiotics which target different targets in the cell. So carbenicillin is the membrane, trimethoprim is the DNTP pool, ciprofloxacin is the DNA, and aminoglycosides is the ribosome. And here you can see that we have differences now. For example, here the TGT, it gets lost from the culture. So you have almost 100 to 1,000-fold less delta TGT strain than the wild type at the end of the competition. It means that it has a really large growth defect in the presence of tobramycin, which is not the case, for example, in the presence of Cipro or the other antibiotics. We can also have another case where the mutant wins. Here, when you delete this gene, the mutant strain wins, which means that the deletion of this gene is beneficial. And it's not the case for other antibiotics. And for each antibiotic, we have some different genes that have an effect. For example, here for trimethoprim, it's this one. So doing that, we tested 25 tRNA or RNA modification mutants. And what we see is that we confirm that what was known in the literature, that when you delete them, there is no growth phenotype in the absence of antibiotics usually, but in the presence of antibiotics, we have sometimes strong growth defects or benefits, and this is dependent on the antibiotics or the gene, so it's a specific response. It's not like you delete all the genes and they all become slow growers and they all lose. It's not like that. It's really specific. So then we decided to see how this can happen. So we decided to characterize more one of the deletion strains, which was the TGT. So TGT makes a modification called chiosine on some tRNAs. And it was mostly studied in eukaryotes. So in mammalian cells, this modification is associated to cancer, cell proliferation and malignancy, and also to oxidative stress responses. In yeast as well, it was associated to oxidative stress because it influences mitochondrial protein expression. But in bacteria, there is almost no study linking this chiosine modification with any specific phenotype. So now we have one strong phenotype and we decided to study it. So as I said, to show it again here, this is a competition experiment. NT means non-treated, so without any antibiotics, both strains are equal, but when we put the tobramycin in it, the TGT loses, delta TGT. We also tested this with high doses of tobramycin, lethal doses, so 5 times more than the minimal inhibitory concentration here. So we grow bacteria, then we take them and we treat them with this dose of antibiotic, and we plate them at different time points to see the survival rate. And we see here in the blue that the wild type dies at this rate that we can see, and the delta TGT mutant deleted for this modification dies faster here. So it is less tolerant to tobramycin. So TGT modifies 4 tRNAs in the cell, which are tyrosine, aspartate, histidine, and asparagine. So we wondered whether one of these amino acids were more important in the response to tobramycin. So just to show you how modification of tRNA could influence codon decoding, this scheme, so it shows again the central dogma, DNA, messenger RNA, and protein. So here I put the example of tyrosine codon. So tyrosine has can be coded by 2 codons, TAT or TAC. So when this is transcribed into messenger RNA, it becomes TAT becomes UAU and TAC becomes UAC. So this UAC codon has a tRNA anticodon which can recognize it, which is AUG, which forms a Watson-Crick base pair here. And so, like depicted here, so this is the expected situation. But when you have a chiosine modification here at the G becomes a Q, and then here we don't really know what happens, if this is more stable or less stable than this, for example. So for the other codon, TAT, as I said, messenger RNA is UAU, and you would expect a tRNA which is AUA, but this tRNA is not present in the genome. in gram-negative bacteria. So they do not have this tRNA AUA. So this codon has to be decoded by the same tRNA decoding the TAC codon. So what we have is depicted here again. So the UAU is decoded by the AUG. And here you have a mispair. But this is the real life situation. And when you have Q-zine instead of a G, again, we don't know what happens if this affects the stability of the codon decoding and codon recognition. So here we can see that modification of the anticodon of a tRNA can impact the codon decoding and recognition in general. So this happens for the tyrosine, but also for the other tRNAs that are modified by Q-zine. So what we did is that we constructed a fluorescent reporter, GFP, green fluorescent protein, where we introduced some codons at the beginning of the gene. So we wanted to test the four amino acids for which tRNAs are modified by Q-zine, and these are all the 8 codons here that I show. It's not very important, but we constructed reporters where we put repeats of the codon that we want to test, and if the codon is correctly decoded, the green fluorescent protein will be synthesized and it will be fluorescent. But if there are problems for decoding the inserted codon, then the fluorescence will go down because there will be less protein GFP made. And so what we saw is that only for one codon we had a difference between the wild type and the non-modified delta TGT strain, and it's the TAT codon, the tyrosine TAT codon. In the delta TGT mutant, it was more fluorescent. It means that more proteins are made when the modification isn't there at the TAT codon. So this suggested that there is more efficient amino acid incorporation in the absence of modification at tyrosine TAT. So we constructed another reporter, which is a bit different. So it's a beta-lactamase gene which codes for carbenicillin resistance. It's quite well described, and we know that there is a tyrosine in it that is important for its function. So if you remove this tyrosine, the cells become sensitive to carbenicillin, even if they have this gene. So the beta-lactamase gene, this tyrosine codon is a TAC, and we made a synonymous mutation. So we changed the TAC to TAT, which is still a tyrosine. So both versions of this gene code for the same protein, okay, but it's just not the same codon. So we introduced these two versions in wild type and in delta TGT strain. So in wild type, both grow at the same rate in the presence of carbenicillin, but in delta TGT, one grows faster, and it's the TAT version. So even when we have two genes coding for the two same proteins, the codon in it is very important for the expression of this gene. So in the absence of modification in delta TGT, when you have a TAT here, you have more beta-lactamase protein that are made, probably. So these experiments showed that the TAT codon is better decoded in the absence of Q-zine, but then we would like to know what's the link between this and the fact that delta TGT is sensitive to tobramycin. So to ask this question, we did some proteomics. So we took proteins from, we grow the cells in the presence of tobramycin, and then we quantify proteins from the wild type and proteins from the delta TGT, and we compare the abundance of all the proteins in mass spectrometry. And what we saw is that there was one protein which was really much more abundant in delta TGT than in wild type, which is RS6A here. There were 13 times more RS6A in delta TGT, which is a lot in this experiment, in mass spec proteomics experiments. 13 times, it's really a lot. So we wondered if this high abundance of RS6A could explain first the sensitivity to tobramycin. So to understand that, we just expressed this protein RS6A in the wild type, so we overexpressed it. And when we overexpress this protein, we see that the growth in tobramycin is decreased. So the overexpression of RS6A can explain why TGT is more sensitive to tobramycin. Okay, so what is RS6A? Briefly, RS6A is an inhibitor of oxidative stress response. So we have this is not really important now, but I will just to explain. We have SOX-R, oxidative stress response regulator, which is active when there is oxidative stress, but when we have more RS6A protein, it inactivates SOX-R and the oxidative stress response. So if you have oxidative stress and too much RS6A, then the oxidative stress response of the bacteria is diminished. And what we think is that maybe in delta TGT, since we have more RS6A, then maybe we inactivate the oxidative stress response, and maybe that's why the top sensitivity happens. We thought that because we know that in the presence of tobramycin, there is some oxidative stress in the cells. We had already observed that previously. So as I said, if you overexpress RS6A, even in the wild type, it grows much more slowly in the presence of tobramycin. And what was interesting in this protein is that it has 6 tyrosine codons, and 5 of these 6 tyrosine codons are TAT. So in the Vibrio or gram-negative bacteria genome, if you randomly take any protein from the genome, if it has 10 tyrosine codons, 5 are TAT and 5 are TAC. Usually it's 50%, 50-50, as shown here. But for RS6A, you have 80% of TAT, which is called a codon usage bias. So RS6A has a bias towards TAT codons. So what we did, we thought maybe RS6A is much more expressed in delta TGT because of these TAT codons. So we replaced these TAT codons by TAC codons, which are also tyrosine codons, and they make the same protein. And we asked if this TAC version is expressed at high levels like the TAT version. And so this we checked that using fusions again with the green fluorescent protein. So we measure fluorescence here. So with the TAT version, we confirm that in delta TGT we have much more RS6A. But when we replace the TAT codons with TACs, we lose that. So we don't have this high expression level anymore. So it means that RS6A is highly expressed in delta TGT because it has more TAT codons than TAC. So this brings me to the concept of modification tunable transcripts, which were defined in eukaryotes first in this paper here as transcripts, so mRNAs, which use specific codon biases, so which do not have the same codon usage as the rest of the proteins in the genome, which encode stress response proteins, and for which translation is influenced by RNA modifications. So in eukaryotes, usually they are involved in cell cycle progression and cellular stress. So we propose that RS6A can be qualified as a modification tunable transcript because it has a codon usage bias. It has more TAT than TAC codons. It encodes a stress response protein because it regulates the oxidative stress regulator, and its translation is influenced by the chiosine modification, TGT. But for this to work, you need to see if the modification levels of inside the cell can be dynamic, if they can vary. Because if they do not vary, you cannot. They cannot be a stress response. They cannot function as a stress response, because when there is a stress, the modifications have to go up or down so that the cell responds to that. So we wanted to see whether there are conditions where the TGT gene is more or less expressed. And we found that yes, some conditions. So here we measure mRNA levels of TGT, and we saw that in presence of tobramycin or in other conditions, there are some conditions that upregulate TGT. And then we wanted to see whether upregulation of TGT is associated to more chiosine in tRNAs. So we looked at chiosine levels in tRNAs by mass spectrometry, and we see that in this condition where TGT is highly expressed, we also have more chiosine in tRNAs. So this shows that chiosine modification levels can be dynamic, and maybe they can be used by the cell to respond to antibiotic stress or other stresses, maybe oxidative stress. So what we propose here as a model is that when there is a stress, so in our case it's antibiotic stress, but it can be other, starvation, temperature, acidity, RNA modification enzymes can be activated or inactivated. This also can happen. So in our case, it was TGT. This leads to changes in modification levels. So maybe we have more chiosine in some tRNAs. And this impacts protein synthesis on specific proteins. So you have specific proteins that are up or down-regulated, which creates some heterogeneity in the phenotype, because even if the genome is the same, the expression levels of some proteins change. And this leads to adaptive response to stress and leads to homeostasis. So then we wondered, so we found RS6A protein in a proteomics experiment, but we wondered whether we could look in silico for gene sequences to find some candidate genes which could be regulated by the same modification mechanism. So in Vibrio cholerae, we looked at genes which have more TATs. And one category here was interesting. It's DNA repair. So DNA repair genes are shown here in this heat map. And you can see that, so in the left, it's the TAT codon. And when it's red, it means there are more enriched. And in the left, it's the TAC codon. And you can see that the rec genes, rec A, rec O, rec R, which are involved in DNA repair are enriched in TAT codons. They have a TAT bias. And so we thought if DNA repair genes have a TAT codon usage bias, then maybe in the delta TGT strain, where there is no cuisine, since TAT codons are better decoded, maybe you have more of these DNA repair genes. So maybe those strains would be more resistant to DNA damaging agents. So we tested that using UV resistance. So UV, ultraviolet rays damage DNA, and so the cells die. And so at 60 joules here, you have the wild type, and the delta TGT was 4 to 9 times more resistant to this dose of UV than the wild type, which was consistent with the cells having more DNA repair genes. We also looked at it in E. coli, but in E. coli, these genes do not have the same codon usage. They are not more enriched in TAT, and you don't have this UV resistance increased in delta TGT. So now our model is that in presence of stress, there are changes in RNA modifications which impact translation and lead to translational reprogramming of proteins with codon biases, which leads to phenotypic adaptation. This can be a layer of regulation on top of transcriptional regulation of genes that is much better described. So there is an epigenetic or epitranscriptomic, we can call it epitranscriptomic as well, adaptation to antibiotics. This can also be much more generally adaptation to changing conditions, for example, gut versus urine for E. coli, for example, who has different niches, the gut, the urine, or the external environment, gut versus water. So because, for example, Vibrio cholerae has two niches, the host gut, and the aquatic environment. It can be a way to regulate stress, transcriptional regulators. When you look at these regulators in the genome, usually they have codon usage biases, not necessarily for tyrosine, but also for others. Ribosomal proteins as well, they have high codon usage biases. So maybe they can also be regulated by other modifications. So that's an interesting thing to study further in the future. And maybe we can think that such tolerance due to transient modifications of tRNA, maybe they can increase the chances of bacteria to acquire genetic mutations, which would make them resistant to antibiotics. So that's it. I would like to thank all the people involved in this work, who are the group members here, and all our collaborators. And most of all, thank you all for your attention. I hope it was a bit interesting for you. Thanks.
Thank you so much, Dr. Zeynep Baharoğlu. If you wish, we can go to the question and answer section prepared by our team and consisting of questions from our participants.
Okay, I stopped the sharing.
Okay, it's okay. I'm directing the first question. Epigenetic studies in bacteria often rely on deactivating methylation-related genes. Instead, could studying the expression of genes involved in DNA methylation help elucidate the pathways involved in this mechanism?
In the mechanism of DNA methylation, you mean?
In the mechanism, the DNA methylation in the mechanism.
Yes, so DNA methylation is quite well described. So you have the DNA restriction modification systems, which are a bit different because it defends the cells from DNA coming from outside, for example, from phages. But you have also the orphan methylases. they are called orphan because there is a methylase, but there is no restriction enzyme, for example. And these sometimes they participate in regulation of the cell cycle, but sometimes we don't know what they do, like the RNA modification. So it's also an interesting thing to study more.
Thank you for your answer. I'm continuing with the next question. Do we know whether the transcriptional change induced by antibiotic stress can later become genetically fixed in the population by natural selection?
Yeah, that's an interesting question. We think it's possible, but we don't have any evidence yet. So, yeah, I think if you allow bacteria, for example, with the tRNA modifications to become more tolerant and better and reply to an antibiotic stress, then they will have more chance, they will divide more and will have more chances to fix some mutations that would occur in their genome and become more resistant. It's an interesting point.
Thank you for your answer. I'm continuing with the next question. This comes from YouTube. In bacteria, does expression changes affecting hundreds of genes across the genome is the exception or the rule? Thanks.
Yeah, it's difficult because I presented some results and I said we find one protein RS6A and we use it to say that it's because of this protein that maybe we see the effect that we see with tobramycin, but I believe that it's much larger than that. RS6A is one protein, but I believe that many other proteins are regulated this way, and I think the sequences of these transcripts have evolved with this bacteria in their niche. For example, the codon usage of a given gene that is very conserved, like REK, for example, in Vibrio cholerae and E. coli are not the same. So I think every bacteria evolve in their niche and adapt to that niche using their, thanks to their codon usage, for example, and hundreds of genes are can be regulated this way. If it's an exception or not, I don't know. It has to be studied more in bacteria, at least. It's not very well known.
Thank you for your answer. I'm moving on to the next question. Can bulk or single cell methods be able to link the heterogeneity of single cell susceptibility to the population scale response to antibiotics?
Yeah, that's also very interesting. So we did, for this study, we did everything in bulk. But yeah, we have to do single cell methods to visualize heterogeneity and to see whether one phenotype conferred by modification, for example, if a subpopulation of bacteria have more modification, if we can visualize that to ask the question whether these cells are more or less sensitive to antibiotics, and we could make that link, it would be really nice. Yeah.
Thank you for your answer. Another question is, while bacteria adapted to different antibiotics often show overlap in their sets of mutated genes, what is the importance to know about the extent of parallel evolution at the transcriptomic level?
Yeah. So I think antibiotics are some stress for bacteria, but it's not the major stress that they encounter. For example, in nature, the major stress now would be phages or temperature stress. And so I think what we see when we study response to antibiotic stress, even if they are different antibiotics from different families, I think those stress responses are integrated in larger stress responses of bacteria. So, for example, what we see usually is that when we try to see what happens with tobramycin or ciprofloxacin, we end up with oxidative stress response. So maybe bacteria use the stress responses that they already have for other stresses to respond to antibiotics. So maybe that's why there are these overlaps that we see.
Thank you for your answer. Another question is, new non-classical methods that can detect modification, like example as phosphorylation in bacterial DNA backbone. How can we control isogenic bacterial population within different heterogeneity among bacteria of new relations between epigenetics and biochemistry?
Okay, so if I understand correctly, how can we detect the different levels of modifications between different bacteria? Maybe that's the question. So actually, the reason why the RNA modifications in bacteria are not so well studied for now was the technical issue that we could not really detect so well the modification levels and modifications. But now there are new techniques that are emerging. There are some techniques based on next generation sequencing and chemical treatments, and now nanopore sequencing, which is booming, and some labs are really experts on these, and there are papers going out every day. So I think the nanopore technology development and the analysis of data and the emergence of specialists will really help this field in the next years.
Thank you for your answer. I'm moving on with the next question. There is a potential importance of genomic expression patterns in shaping antibiotic-resistant profiles. On the other hand, how can we understand of global transcriptomic rewiring induced by resistant mutations?
Yeah, so I think there is a balance between bacterial growth in the absence of antibiotics, and sometimes antibiotic resistance genes have a cost, and this cost is balanced when bacteria encounter antibiotics. So there is a balance between bacteria having these resistance genes and bacteria which have them but grow slowly maybe, and bacteria which don't have it but they die when there's the antibiotic. Maybe populations are mixed and there is a balance between different populations having or not these genes. And sometimes the expression of antibiotic resistance genes are also regulated. They are not always expressed. So that can be a strategy.
Thank you for your answer. Another question is, much information has been gained from recent studies on epigenome modification of the host by pathogens. What difficulties are experienced in transferring these findings from the laboratory to critically ill patients?
Yeah, so that's not my expertise, really, the clinical part. But yes, there are more and more labs studying the effect of bacteria on host modification in the host, so the human cells. So yeah, I don't know. I'm not very specialist on that.
Thank you. The next question is how can we determine the single-cell response to antibiotics while screening different bacteria and molecules having different mechanisms of action?
Yes. In many cases, the stress responses are found in many different bacteria. And so usually what we do is that we have fluorescent reporters of different stress response regulators or stress responses, and in either in time-lapse microscopy or microfluidic devices, we can follow basically fluorescence of reporters. That's what we usually do.
Thank you for your answer. The next question is droplet-based approach. The question comes from the YouTube. The if current text is the dependence on populations to achieve a consensus sequence, how can we determine the epigenetic heterogeneity which exists with different cells of isogenic populations?
Yeah, again, this is done usually with either sequencing techniques or nanopore, but few labs do it.
Thank you for your answer. The next question is, droplet-based approach can provide insights into the heteroresistance of bacterial populations. So are there any limitations to these methods?
Yes, so it's a powerful approach because you can have many droplets and you can put many bacteria in every droplet. The limitation would be rare events because, for example, if you study persistence, so this is a subpopulation of bacteria which are supposed to be dormant or they are not resistant, but they don't die when there is the antibiotic treatment. And these populations are quite rare. So, for example, they are at 10 to the minus 5 in the total population. So you would need 10 to the 5 droplets, and it won't be statistically significant anymore. But other than that, the droplet-based approach is actually a really cool way to study this because you can also change the media and follow what happens to the fate of the population for as long as you want. And also the other limitation, of course, is data analysis because it generates a lot of images and a lot of data. So you need to be able to store these and analyze it.
Thank you for your answer. I'm moving on to the next question. To overcome microbial therapies that efficiency goes rough because of antibiotic resistance, can antibiotic-loaded nanocarriers determine infected cells and act like a phagocytose mechanisms to release suitable drugs? This can be designed for worldwide non-individual treatments.
Yeah, definitely we need new strategies to be able to treat people with antibiotics. So there are many strategies. There is this one. There can be combination of different molecules in order to enhance existing antibiotics action. There's also the phage therapy. Yeah.
Thank you for your answer. I continue with the next question. Especially in our country, like any other disease treatment, antibiotics are chosen increasingly. Because of that, maybe with this small effect that we make on bacterial genetic variation, we change the resistance to antibiotics. Can we classify bacteria and the relationship between antibiotics and the result of survival time?
Yes, so for the increasing usage of antibiotics, yes, it can. It does select for more resistant bacteria. Unfortunately, there were some studies of which translate experiments done in the lab for persistence, for example, to a more clinical strain of Pseudomonas inside. So these are in vivo experiments. So usually it's in mice, for example. And we see that what we see in the lab happens also in mice. So probably also in humans, but it's not very well known yet.
Thank you for your answer. The next question is, many of bacterial nucleotide-associated proteins do not appear to be homologous to eukaryotic proteins. So could they be suitable targets for new antibiotics?
Sure, but yes, of course. But then we have to find a molecule that targets them and that can enter the bacteria and that does not enter the human cells. So usually it's the problem is that the antibiotic research is expensive, but the benefit for industries and companies is not so high because it's not the benefit is not so high. So the private companies do not always want to invest on these studies. So that's why the research on antibiotics is a bit not advancing as it could.
Thank you for your answer. Next question is, while the embryo is developing, the tasks are defined by methylation takes place with the GPG method. Can stem cells transform into any tissue because these tasks are not given?
Yeah, so I'm not
And also,
yes.
Sorry for the interrupting. Can we prevent certain DNA regions from being methylated?
Yes. So I'm not at all an expert on CPG methylation in eukaryotes, but I know discussing with colleagues that, for example, in the brain tissue, only one methylation of one ribosomal RNA determines the cell fate, whether one cell goes at some point of the brain and not to the other part. So I guess in the embryo it must be the same, but I don't know more.
Thank you for your answer. The next question is, during bacterial infection, the pro-inflammatory response can delay bacterial spread and avoid infection. With evolution results, bacteria's effectors allow bacteria to get colonized, changing host cytonic pathways to control their survival periods. This widely affecting cycle can be blocked by the host biochemical reorganized novel treatments.
Yeah, that's an interesting point as well. I think that's a good field that will grow in the future as well.
Thank you so much for these informative answers. Lastly, I want to ask that what advice would you give to the students studying in the scientific fields?
Oh, that's a big question. I think one should really follow their passion, and if you really want to continue in that field, I think you should be persistent, because sometimes it's difficult and it seems that there are not many chances to get a position and things like that. But I think if it's really your passion, then go ahead.
Thank you for your helpful advices. On behalf of myself and all our participants, I would like to thank you for spending your valuable time, transferring your knowledge to us, and for this informative presentation.
Thank you very much, and thanks for inviting me, and I was very happy to be here today.
I wish you healthy days.
Thank you. You too.
Thank you. At 6 p.m., Associate Professor Dr. Ahmet Coşkun will be with us to give a speech on secrets of healthy life hidden in cell and tissue architecture. Thank you.
Bioinforange platformunun kuruluşunun 2. yıl dönümünde düzenlediğimiz Bioinfocongress II etkinliğimize hepiniz hoş geldiniz.
Merhabalar, hoş geldiniz laboratuvarımıza.
Merhaba hocam, hoş geldiniz.
Merhaba, hoş bulduk. Sağ olun, teşekkürler davetiniz için, organizasyon için.
Davet için tekrar çok teşekkürler. Organizasyon da çok başarılı gözüküyor. Çok teşekkür ederim.
Tekrar teşekkürler davetiniz için. Bugün bilgisayar teknolojisiyle biyomoleküler sistemlerin simülasyonu ve ilaç tasarımı konusunda konuşacağım.
Muhteşem. Çok teşekkür ederim. Evet, hepinize merhabalar. Organize komitesindeki arkadaşlara da çok teşekkür ederim beni davet ettiğiniz için.
Herkese merhabalar. İlk önce davetiniz için çok teşekkür ederim. Buradaki diğer konuşmacılarla beraber sizlerle beraber olmak gerçekten gurur verici.
Bioinfocongress'te sunum yapmaktan beni davet ettiğiniz için çok memnunum ve sanıyorum tamamen insan hücre fonksiyonlarına yönelik olmayan daha basit canlılarla ilgili olan tek konuşma belki. Ama böyle bir değişiklik getirmekten de çok memnunum.
Öncelikle hepinize teşekkür etmek istiyorum. Bioinfocongress oldukça güzel bir organizasyon yaptınız.
Tüm Bioinforange çalışanlarına ya da gönüllülerine teşekkür ediyorum. Beni davet ettiğiniz, zamanınızı ayırdınız.
Merhaba, davetiniz için çok teşekkür ederim. Bu aktiviteyi organize ettiğiniz için de teşekkür ederim. Gerçekten çok büyük bir çalışma gerektiriyor.
Thank you, it's a pleasure. Thank you for the invitation.
İyi akşamlar. Başlamadan önce bu güzel organizasyonu düzenleyen arkadaşlara gerçekten teşekkür etmek istiyorum. Bu kadar büyük bir topluluğu bir araya getirmek, böyle bir organizasyon gerçekleştirmek gerçekten büyük bir başarı.
It's a really fantastic program that you've put together over these last few days. So it's been some brilliant science and some brilliant scientists speaking. So thank you very much again for the kind invitation.
Welcome everyone, I'm Neva Çalışkan. I'm a group leader at the Helmholtz Institute for RNA-based infection research. And today my talk will be about how meaning of the genome can be altered by frameshift RNAs and how this response can be regulated by host and viral proteins.
Teşekkür ederim davetiniz için. Bugün mRNA aşılarından bahsedeceğim sizlere.
Merhaba arkadaşlar, bugün sizlere katılmaktan çok hoşnutum. İlk önce Bioinforange öğrencilerine bu güzel organizasyonu bir araya geldikleri için teşekkür ediyorum. Gerçekten benden önceki videoları izlemekten çok hoşlandım.
Merhaba herkese, buraya geldiğiniz için teşekkürler. Bugün ökaryotik genomların organizasyonu hakkında bir konuşma yapacağım.
Merhaba herkese, katıldığınız için ve dinlediğiniz için teşekkürler.
Merhabalar, hoş bulduk. Başlamadan önce böyle güzel bir organizasyonda emeği geçen arkadaşlara teşekkür ediyorum. Ayrıca davetiniz için de teşekkürler.
It's a tremendous honor and pleasure for me to be here after starting my lab just over 2 months ago.
Gün boyunca 30 saat bir yayın süremiz oldu. 3 eğitim, 17 konuşma, toplamda 20 konuşmadan görüyorsunuz yorgunluğumuzu da arka planda. Öncelikle bizlere konuşmalar, eğitimler veren 20 değerli hocamıza çok teşekkür ederiz. Açıkçası davet ettiğimiz hocalardan çok yüksek oranda kabuller aldığımız için bu kadar çok yoğun bir programımız oldu. O yüzden hocalarımıza çok teşekkür ederiz. Onların bize davetleri bu kadar çabuk bir şekilde, hızlı bir şekilde bizleri kabul etmesi, konuşmalar yapması bizler için çok güzel ve bize gerçekten çok motive ediyor. Hocalarımızın haricinde siz şu anda 277 kişi var bu saatte. Çok teşekkür ederiz. Değerli katılımcılarımıza çok teşekkür ediyoruz. Ancak büyük bir teşekkürü de ben buradaki ekip arkadaşlarıma yapmak istiyorum. Bioinforange'ın 2. yıl dönümü, doğum günü. Buradan Bioinforange'a emek veren, gönül veren, Bioinforange'ı seven tüm bilimseverler, katkı sağlayanlar, öğrenciler, insanlar adına iyi ki doğdum Bioinforange diyorum. Bilim paylaştıkça güzel, bilim paylaştıkça ilerler. Bilimle kalalım. Bioinforange platformunun bilim yolculuğunda 3.sünü düzenlediğimiz Bioinfocongress ilk etkinliğimize hepiniz hoş geldiniz.
Çok teşekkür ediyorum. Mehmet, senin şeyin nezdinde bütün organizasyonu yapan arkadaşları da tebrik ediyorum. Emeğinize sağlık. İyi bakın kendinize, görüşürüz.
Merhaba İvan Hocam, tekrardan hoş geldiniz.
Merhaba, teşekkürler davet için. The first time we go very different, transferring your knowledge to us and for this informative presentation. Kendim ve tüm katılımcılarımız adına çok teşekkür ediyorum hocam.
Ben teşekkür ederim.
Çok memnun oldum hocam. Sağlıklı günler diliyorum.
Sizlere bir akşam.
Yep, thank you guys so much. I really enjoyed it. Great questions from everyone.
Merhaba Tuğba Hocam, tekrardan hoş geldiniz.
Hoş bulduk. Çok teşekkür ediyorum davetiniz için. Harika bir organizasyondu. Herkese selamlar.
Değerli vaktinizi ayırdığınız, bilgilerinizi bize aktardığınız ve bilgi dolu bu konuşma için kendim ve tüm katılımcılarımız adına çok teşekkür ediyorum.
Ben teşekkür ederim davetiniz için.
Merhaba Nilüfer Hocam, tekrardan hoş geldiniz.
Merhaba, hoş buldum. Beni iyi duyup görebiliyor musunuz?
Evet, görüşürüz.
Çok teşekkür ederim davetiniz için. Çok teşekkür ederim. Öğrencilerle böyle bu sanal ortamda da olsa bir araya gelmekten.
Merhabalar. Öncelikle davetiniz için çok teşekkür ederim.
Teşekkür ederim hocam.
Çok güzel bir event ortaya koymuşsunuz. Bu kadar değerli bilim insanı arasında yer almak gurur verici. Teşekkür ederim.
Çok teşekkür ederim. Nazik davetiniz için çok mutlu oldum. Bu arada bu YouTube'da olan sayın seyirciler, bu saatte cumartesi akşamı gidip başka bir şey yapmayıp bu konuşmayı dinlemeye geldiğiniz için size de hürmetlerimi sunuyorum.
Çok teşekkür ediyoruz. Hem kendi adıma hem de bütün katılımcılarımız adına.
Rica ederim. Ben çok memnun oldum. Beni onure ettiniz. Çok teşekkür ederim.
Biz onur olduk hocam. Çok sağ olun. Size çok teşekkür ediyorum. Katılımcılarımız ve kendim adına. Ben çok teşekkür ederim. Benim için de güzel bir deneyim oldu. Bu harika sunum için katılımcılarımız ve bütün ekibimiz adına çok teşekkür ederim hocam. Ben çok teşekkür ederim beni misafir ettiğiniz için.
Thank you. Thank you so much for this space. Thank you for having me here and for this opportunity.
Bilgi dolu konuşma için kendime tüm katılımcılarınız adına çok teşekkür ederim.
Ben teşekkür ederim davetiniz için.
Tekrardan çok teşekkür ediyoruz. Katıldığınız için çok mutlu duydum.
Ben çok teşekkür ederim. Çok güzel bir organizasyon gerçekleştirmişsiniz. Beni davet ettiğiniz için tekrar teşekkürler. İyi günler dilerim.
Biz teşekkür ederiz hocam. Bioinforange platformu olarak düzenlediğimiz Bioinfocongress 3 etkinliğimizde çalışmalarına yurt içinde ve yurt dışında devam eden değerli bilim insanları katılımıyla toplamda 15 seminer gerçekleştirmiştik. İzninizle burada organizasyonda emeği geçen arkadaşlarımı davet etmek istiyorum. Şimdi burada ekranda olan arkadaşlarımız haricinde kongremizin düzenlenmesinde organizasyonda emeği geçen tüm ekip arkadaşlarımıza teşekkür ediyoruz. Ve sizler gibi yani burada hepimiz öğrenciyiz biz ekip olarak. Özellikle öğrencilere bir katkımız oluyorsa akademik hayatı ve işte gelecek idealleri konusunda bir fikir verebiliyorsak buradaki konuşmalar, işte bizler bir katkı sunabiliyorsak açıkçası biz bundan çok mutluluk. Tekrardan etkinliklerimize devam edebilmeyi umuyorum. Bizi sosyal medya adreslerimizden takip edebilirsiniz. Sizlerin desteğiyle biz motivasyon buluyoruz. Bizler motive olunca da böyle güzel etkinlikleri sizlere sunmaya çalışıyoruz. O yüzden karşılıklı etkileşim ve destekleşmeyi umuyorum. Tekrardan hepinize çok teşekkür ediyorum. Katıldığınız için de çok teşekkürler tekrardan ve herkese bol başarılı akademik kariyerler, güzel bir hayat, sağlıklı bir hayat diliyorum ve sağlıklı günler diliyorum. Görüşmek dileğiyle tekrardan. Hoşçakalın. Bioinforange platformunun bilim yolculuğunda üçüncüsünü düzenlediğimiz Bioinfocongress IV etkinliğimize hepiniz hoş geldiniz.
Çok teşekkür ediyorum. Mehmet, senin nezdinde bütün organizasyonu yapan arkadaşları da tebrik ediyorum. Elinize, emeğinize sağlık. İyi bakın kendinize, görüşürüz.
Merhaba İlhan Hocam, tekrardan hoş geldiniz.
Merhaba, teşekkürler davet için.
Here for scan video available time, transferring your knowledge to us, and for this informative presentation. Kendim ve tüm katılımcılarımız adına çok teşekkür ediyorum hocam.
Ben teşekkür ederim.
Çok memnun oldum hocam. Sağlıklı günler diliyorum.
Sizlere bir akşam.
Yep, thank you guys so much. I really enjoyed it and great questions from everyone.
Merhaba Tuğba Hocam, tekrardan hoş geldiniz.
Hoş bulduk. Çok teşekkür ediyorum davetiniz için. Harika bir organizasyondu. Herkese selamlar.
Değerli vaktinizi ayırdığınız, bilgilerinizi bize aktardığınız ve bilgi dolu bu konuşma için kendim ve tüm katılımcılarımız adına çok teşekkür ediyorum.
Ben teşekkür ederim davetiniz için.
Merhaba Nilüfer Hocam, tekrardan hoş geldiniz.
Merhaba, hoş buldum. Beni iyi duyup görebiliyor musunuz?
Evet, görüyorsunuz. Evet.
Çok teşekkür ederim davetiniz için. Çok teşekkürler. Öğrencilerle böyle bu sanal ortamda da olsa bir araya gelmekten.
Merhabalar. Öncelikle davetiniz için çok teşekkür ederim.
Teşekkür ederim hocam.
Çok güzel bir event ortaya koymuşsunuz. Bu kadar değerli bilgi insanı arasında yer almak gurur verici. Teşekkür ederim.
Çok teşekkür ederim. Nazik davetiniz için çok mutlu oldum. Bu arada bu hani YouTube'da olan sayın seyirciler, yani bu saatte cumartesi akşamı gidip başka bir şey yapmayıp bu konuşmayı dinlemeye geldiğiniz için size de hürmetlerimi sunuyorum.
Çok teşekkür ediyoruz. Hem kendi adıma hem de bütün katılımcılarımız adına.
Rica ederim. Ben çok memnun oldum. Beni onure ettiniz. Çok teşekkür ederim.
Biz onur olduk hocam. Çok sağ olun.
Size çok teşekkür ediyorum. Katılımcılarımız ve kendim adına.
Ben çok teşekkür ederim. Benim için de güzel bir deneyim oldu.
Bu harika sunum için katılımcılarımız ve bütün ekibimiz adına çok teşekkür ederim hocam.
Ben çok teşekkür ederim beni misafir ettiğiniz için.
Thank you. Thank you so much for this space. Thank you for having me here and for this opportunity.
Bilgi dolu bu konuşma için kendim ve tüm katılımcılarımız adına çok teşekkür ederim.
Ben teşekkür ederim davetiniz için.
Tekrardan çok teşekkür ediyoruz. Katıldığınız için çok mutluyum.
Ben çok teşekkür ederim. Çok güzel bir organizasyon gerçekleştirmişsiniz. Beni davet ettiğiniz için tekrar teşekkürler. İyi günler diliyorum.
Biz teşekkür ederiz hocam. Bioinforange platformu olarak düzenlediğimiz Bioinfocongress IV etkinliğimizde çalışmalarına yurt içinde ve yurt dışında devam eden değerli bilim insanları katılımıyla toplamda 15 seminer gerçekleştirmiştik. İzninizle burada organizasyonda emeği geçen arkadaşlarımı davet etmek istiyorum. Şimdi burada ekranda olan arkadaşlarımız haricinde kongremizin düzenlenmesinde organizasyonda emeği geçen tüm ekip arkadaşlarımıza teşekkür ediyoruz. İşte sizler gibi yani burada hepimiz öğrenciyiz biz ekip olarak. Özellikle öğrencilere bir katkımız oluyorsa akademik hayatı ve işte gelecek idealleri konusunda bir fikir verebiliyorsak buradaki konuşmalar, işte bizler bir katkı sunabiliyorsak açıkçası biz bundan çok mutluluk. Tekrardan etkinliklerimize devam edebilmeyi umuyorum. Bizi sosyal medya adreslerimizden takip edebilirsiniz. Sizlerin desteğiyle biz motivasyon buluyoruz. Bizler motive olunca da böyle güzel etkinlikleri sizlere sunmaya çalışıyoruz. O yüzden karşılıklı etkileşim ve destekleşmeyi umuyorum. Tekrardan hepinize çok teşekkür ediyorum. Katıldığınız için de çok teşekkürler tekrardan ve herkese bol başarılı akademik kariyerler, güzel bir hayat, sağlıklı bir hayat diliyorum ve sağlıklı günler diliyorum. Görüşmek dileğiyle tekrardan. Hoşçakalın.
Bioinfocongress IV. Assistant Professor Dr. Ahmet Coşkun. Secrets of Healthy Life Hidden in Cell and Tissue Architecture. Assistant Professor Dr. Ahmet Coşkun is currently a Bernie Marcus Early Career Professor of Biomedical Engineering at Georgia Institute of Technology and Emory University. Dr. Coşkun is a systems biotechnologist and bioengineer working at the nexus of multiplex cell imaging and quantitative tissue biology. Dr. Coşkun directs an interdisciplinary research team at the Single Cell Biotechnology Laboratory, an interdisciplinary program that is strategically positioned for multiparameter imaging one cell at a time by spatial context and function. Dr. Coşkun holds 5 issued patents and is also the co-author of more than 40 peer-reviewed publications in major scientific journals. Previously, Dr. Coşkun was an instructor at Stanford University. Dr. Coşkun received his postdoctoral training from the California Institute of Technology. He holds a PhD degree from the University of California, Los Angeles. His research has been supported by the National Institutes of Health, Wellcome Leap, Burroughs Wellcome Fund, NSF CMAT, American Lung Association, American Cancer Society IRG, Multicellular Engineered Living Systems, and Regenerative Medicine Center. He is a recipient of the Student Recognition of Excellence in Teaching, Class of 1934, Course Instructor Opinion Survey Award. In addition, he leads outreach programs to engage K-12 students and undergraduate students through BioCrowd Studio, an innovative crowdsourcing program bringing together interactive virtual media distributed by kits and collaborative spatial discovery.
Dear Dr. Ahmet Coşkun and participants, welcome all of you to our Bioinfocongress IV event. Dr. Ahmet Coşkun will give us a talk today on secrets of healthy life hidden in cell and tissue architecture. At the end of the presentation, we will have a question and answer section prepared by our team and consisting of questions from our participants. Dear Dr. Ahmet Coşkun, welcome back. We're so glad to have you here.
Thank you, Efekan, for the kind introduction and also for this invitation. And I'm very excited to be here and also share some of our work with the students in especially bioinformatics. And also thank you for this also wonderful organization. I think it will help increase the impact of the international science, right, through online forums like this one. And therefore, I appreciate your support and your efforts on this. So yeah, I'll get my...
Oh, sorry, sir. I'm sorry. Please.
I was just going to say I'll get started, but do you have anything to add?
Oh, no, please.
So then we'll get started. Thank you for sharing the slides. So today we're going to talk about architecture, right? So particularly if you look at this slide on the left, you're seeing the building that was designed in the shape of a cell and how also intercellular or multicellular structures would look, right? The architectures, they design these buildings, and on the right, you're looking at multiple cells. Inside, you're looking at the specific organelles and nucleus in the purple color, right? And the organization also is there. So what's interesting is that there are similarities between a building and cells, right? So today I'd like to highlight this. So therefore, why cell and tissue architecture matters, right? Why is it important? For instance, when you look at inside a building or inside a flat or an apartment, you'll be looking at different compartmentalized parts, right? You'll be looking at the bathroom, you'll be looking at the kitchen and study room, right? And the living room and whatnot. When you focus and zoom into a cell, then you'll be looking at the organelles, right? The endoplasmic reticulum, the mitochondria, the nucleus, and whatnot. So I'd like to, give a general analogy between the two. First, parallelization. So in an apartment or house, actually you can do multiple events. While your family member cooks here in the kitchen, right? Then some of the other family members, they can study in the study room, and some of the others can watch TV, right? So therefore, multiple events and processes can go in parallel. In the cell also, while some proteins are getting phosphorylated to perform a function, while the mitochondria give rise to more energy, some other organelles, they perform the waste disposal, right, of the cell, and some others also carry the proteins from some location to other locations. So therefore, a lot of parallel events. Second, the compartments. If you don't have a compartment in an apartment or in a cell, then it would be challenging to perform in separate multiple events, right? For instance, the kitchen, the cooking will interfere with studying, right? Or the living room will get interfered with another thing happening in the laundry room, right? Same thing with the cell. If you didn't have a compartment such as mitochondria, all these molecules would not be dense enough to generate enough function to perform especially the necessary functions of the cell. Then it's goal defined, meaning that if this cell is a kidney cell, then it performs the function of a kidney. If it is a cardio cell, then it performs the function of a heart. Then if it is a brain cell, then it performs the function of a brain. And similarly, an apartment or a house could be a personal use, or could be a business use, right? Or could be a study, a school or another facility, right? That can play the specific functions. So therefore, you can design it, and users also can design it. And the cells, of course, they self-assemble and design themselves, but they are modular. Then they're also tolerant, right? So in the case of failures, for instance, cells reassemble, change the positions, and change the particular number of organelles and their size and whatnot, right, and show us. So in the case of apartments as well, if you are not comfortable with certain passages, right, and how apartment is organized in terms of the locations, then you can reorganize to the best of your ability, of course. And therefore, there are a lot of things in common, right? And when you then look at the health and disease and zoom in to, for instance, outside the cell and inside the cell, then you're going to see that this architecture also changes between health and disease, right? For instance, the first right image, I will highlight this image. That's an organelle and 3D image of a subcellular region from a healthy donor. And the one on the right is from an obese donor, which means that that's not non-healthy, it's obese donor, right? So in the obese or non-healthy cases, then you start seeing these yellow circles popping up, right? These are the lipid droplets. So this study is actually from Harvard study, where they published this in Nature and showed that the subcellular architecture organization gets altered in disease, right? But also in normal, it has this original organization. And the same thing in the matrix or outside the cell, right? The matrix architecture, matritecture also changes. For instance, if you look at this blue-yellow regions, in the disease, it shows a bubble, right? While in the healthy region, it shows nice features of the extracellular matrix supporting the cell function. So therefore, it actually controls the cell and tissues. And to be able to understand this architecture, there are molecular screening technologies, right? There are sequencing, mass spec technologies, and many others. When you think about sequencing technology, you start with these cells, the architecture of the cells right in the tissue. But the problem is that we then dissociate this architecture, right? We basically use the lysine buffers to dissociate everything, and then you extract the DNA, RNA molecules and do the sequencing. But this is a problem when you want to understand the architecture. So therefore, we perform in our vision an image-based sequencing technology, which we call in situ sequencing, or now people call it spatial sequencing, that can help us sequence the cells before dissociating them. Basically, we keep the cell intact. We do not dissociate them. We keep their original positions in the cells and tissues. Then we perform multiple barcoding strategies to be able to read DNA, RNA molecules, as well as we will overlay them also with metabolites. Then we use typically microscopes or advanced imaging systems to be able to look at these molecular subtypes. Then we will use the big data computers, right, or supercomputers to be able to convert these data sets to sequences. So this is, for instance, advantages. Why? For instance, if you just do a single-cell RNA sequencing experiment, you get this soup of the cells, right? And this is actually similar to a fruit smoothie, right? If you start with the bunch of fruits, then put it in a grinder, right? Then you're gonna make a smoothie. And that smoothie then loses the original context of that fruit bowl, right? So similarly, single-cell RNA-seq also loses that spatial context. You can also only tell how many of these cells are there, and possibly also what are their types in the smoothie. But in the case of spatial or image-based technologies, for instance, this purple one is the H&E, or the histological image that the typically pathologists would use, right, in the hospital. Then you're looking at different parts of these tissues to understand the health and disease. And again, this is the fruit bowl itself, and you do not dissociate this. You keep this intact, right? You keep this intact, and hence you can tell the different structures of this. So, but this is a spatial technology, right, which is called spatial transcriptomics, originally invented in Sweden and now commercialized by a company in the US called 10x Genomics. And here, then you're looking at the spots that are being measured, right, for across the tissue samples. But these spot sizes are around 50 to 100 micrometer. And if you remember the average cell size, that is about 10 micrometer. So each spot size basically would cover a very large area. Here, for instance, a spot size would cover this multiple cell area, right? But it doesn't necessarily tell you the single cell information about molecular densities across these tissues, right? So this is spatial technology. It still gives you the architecture, but one problem is that it averages out still a few cells, not millions of cells, but at least a few cells. Then there are third directions, right, around this, which can keep both spatial and single cell. And again, you start keeping the cells intact. You do not dissociate it, but you keep hybridizing labels and also fluorophores, the light emitting molecules, such that you can create these barcodes. And these barcodes then will give you this picture. And here in this picture, you are looking at each circle with different colors, and each circle, especially blue, indicates nuclear structures. And the different colors indicate, and particularly different distributions of RNA molecules inside these tissues. So therefore, these technologies can both provide single cell information, because you can see these single bubbles here, single circles, but also the colors indicate different cell types and different functions, right? So this is a timeline of this field. If you Google spatialomics, you will see there are a lot of materials coming up nowadays because it's a very hot topic. And why? Because after 2013, there was a boom and there was an interest to be able to measure more RNA molecules, more protein molecules, and also see the spatial information about how tissue looks, right, and how the architecture looks. And remember the first architecture that I mentioned? So that architecture is very key when you study the tissue, because the cancer, immune cells, and how they basically interact also is coordinated by the architecture of these tissues, right? So my entrance in this field is actually around 2014 when I was doing my postdoc at Caltech, where we invented the sequential FISH technology. It is a technique that can measure RNA molecules at very high resolution, at even super resolution levels. Also, it can measure the spatial information at in the tissues. Then around 2018 also, I was at Stanford doing my instructor job, junior faculty job, where we also developed the Codex technology to be able to measure proteins and multiple of these proteins across the images, right? You can look at the previous image that I showed at the single cell level, look at about 30, 40 proteins all at once. So now the field is, of course, progressing indeed fast. And if you look at major journals, spatial, single cell, right? And all these titles are always coming up in the recent journals. So therefore, it's very important to consider to study the cell and tissue architecture the special omics technologies, they basically landed themselves to these problems. And my lab specifically, we are interested in not only the cell types, but also function, right? So when cells are in these tissue and cell architectures, right, or special tissue architectures, they are changing their identities, right? They could be the kidney cell, and they could be the epithelial part of that kidney or another immune part of that kidney. But while they're changing their decisions, they're always using these networks of molecules. So what are these molecules? For instance, the blue one is signaling, the black one is metabolic, and especially this red one and these networks are gene regulatory RNA level interactions. But if you can see, even in the subcellular level, the architecture matters, right? So there will be the molecular organization that can help us understand the interactions of molecules, and then finally it leads to decisions, right? And the cells either grow, right? They differentiate, or they commit to apoptosis and things like that, right? So therefore, we are interested in looking at signaling and metabolism along with the RNA networks, right, to be able to understand the cell function. So particularly, we are excited about mainly 3 directions. One is gene neighborhood networks, how to model high-dimensional data for interactively and also link to the patient outcome. For instance, here, you're looking at one single image, and inside of it, you have all these different smaller regions colored by individual groups, right? And this is not actually a US map, right? This is actually a single cell. But here, each group indicates that there are RNA molecules that are concentrated around those regions, right? So therefore, we will be talking about a technology that can model these interactions or neighborhoods. We call these neighborhoods, right? Architecture refers to the houses, apartments, and in the apartments, you have also your neighbors, right? You have one neighbor, another neighbor, and between you are basically talking and interactions with your neighbor, you decide on several things in your environment. So therefore, we're also looking at these neighbors among the RNA molecules, right, to also help us understand some of the cellular functions. Then we're excited also to study subcellular spatial signaling networks, where here also you're looking at these multiple colors indicating the signaling molecules at the subcellular level, and we are visualizing these networks of more proteins, right? And then our ultimate goal is to be able to trace the signaling transduction visually in the cell. Then we're also excited to study single-cell spatial metabolomics, particularly to understand how cells alter their metabolism, and also they follow a trajectory of metabolic pathways, right? They, for instance, choose the way that the glucose is used more, or fatty acids or fats are used more, right? Then we are also projecting these trajectories on the architecture maps, right, or tissue architecture maps as shown in here. So the first section that I'm going to talk about today is about neighboring RNA, right? So until now, people use these kind of technologies on the left, right? We had technologies to measure RNA molecules in the cell. Again, this is a single cell, and you're looking at these distinct colors. They correspond to different RNA molecules, and each dot corresponds to a single RNA molecule, right? Then it's dispersed across the cell, especially in the cytosol, right? And as you know, the RNA processing mostly occurs, especially translation process, mostly occurs in the cytosol, and you see this very high density. And this is pretty much common, but what's not common is that how do we look at these RNA molecules, and how do we also look at more than hundreds to thousands of different RNA molecules all at once by these images. And we generate these images by a technology called fluorescent in situ hybridization. But now using multiplexing strategies, we can then generate the maps of RNA for about hundreds to thousands of them. And then what's also common was that you can count number of RNA dots, as you see on the bottom left, right, across a single cell and say, oh, actually my blue dots here, we have about 2,000 of them, right? So therefore, actually this is currently about 1,000 dots. My RNA gene expression is about 1,000, right? Then if you zoom into other colors, I mean, the other colors are a little bit, of course, covered by the blue ones, but for instance, black one is also higher, right? So then you can look at the other black ones and other colors, right? And look at the gene expression by just counting these number of dots, right? Again, counts of dots will give you the gene expression. This is the akin to actually a single-cell RNA-seq expressions for the FPKM values. But in the case of these experiments, you're looking at just the counts. But beyond the counts, actually, we have another information that, for instance, the red RNA molecules are in the cytosol, but blue ones are in the nucleus. I mean, why are they in the nucleus? Because these are, for instance, non-coding RNA molecules called link RNA, and this is particularly MALAT gene. So therefore, it is operating in the most nucleus, right? It is different than the mature genes. But the interesting part is that the blue and red genes, they are non-neighboring, right? They're non-neighboring. Then, alternatively, you could also have genes like the bottom two, where the blue and red are neighboring each other, right? Neighboring each other. So how do you distinguish these two? Then we developed these computational frameworks, again, to cluster the individual RNA molecule positions, but ignore their RNA copy numbers, right? Ignore their intensities, ignore everything, but just keep their positions. That we have the X value, Y value, and if this is also in 3D, you have the Z value. So then cluster those positions using Leiden, and then it tells you basically multiple clusters, right? Let's say you can generate up to 30 clusters. Then you can zoom into one region here, a small region here, and inside that region, then you could study RNA neighborhoods by using the nearest neighbor algorithm, right? For instance, here, blues are interacting or neighboring with each other, but here, the blues and reds are interacting and neighboring each other. So therefore, you can distinguish these two cases. And again, these kind of technologies are generated by multiplexing these in situ hybridization methods. So for instance, if you have this RNA molecule here, this RNA is trapped or it's fixed inside the cell, right? There are a lot of proteins. You add formaldehyde, it basically creates this mesh of networks, and proteins are all trapped, and inside and near proteins, then the RNA molecules are trapped. So then you can add these red and green molecules and hybridize and image this under the microscope, and you see this dot on the cell image. Then you can remove this label by an enzyme called DNase-1, and DNase-1 removes all the molecules here except the RNA, right? RNA will be left intact or here. It won't be digested, but the rest of the molecules, they're digested, and then they're floating away the buffer, right? Inside the buffer. So then you can relabel the same RNA with a different color, right? And then generate a barcode for this RNA. For instance, you could color this with first green, then blue, and create this barcode of green, blue for this RNA. Then you can also perform an additional experiment to stain proteins using the antibodies that are tagged by fluorophores, and that tells you especially different subcellular architectures and how they co-localize with all these proteins, all these RNA molecules. Then this experiment you can do about 20 different cycles, right? And each hybridization removal and hybridization removal is defined as a cycle. Then you can perform the clustering, as just we mentioned, digitally, right, to perform the analysis that I mentioned before. So when you zoom in to a cell, then after the clustering the positions of these RNA molecules, you would obtain these boundaries of different regions, the region 1, 2, 3, 4, 5, 6. And we call these for now, and we define these as patch. And these patches, it will be each row, right, will be patches, and each column will be the numbers of these genes, right? So you're looking at the gene 1, which is the orange, orange color, and that would be the number of genes within this patch. But instead of just looking at number of genes per patch, we're also looking at the neighborhoods that I mentioned. So you're creating this network of genes when they are neighboring each other. So we then call this technology SPA-GNN, Absolute Spatial Resolved Gene Neighborhood Networks. And this technology is unique because it has the properties in comparison to other technologies that can help us understand unique features, right? For instance, if you use co-expression, transcription factor binding analysis, computational net inference, or if you just look at some of these large spatial spot data, right, you could make all these networks of gene-gene interactions. But they don't necessarily immediately tell you the RNA neighborhoods, right? Because they're predictions typically. Either if the two genes are high, then you expect that they are basically networks. But in our case, it's a physical analysis, right? We call when the two RNA molecules are nearby or they're neighbors, then they are the networks. So therefore, they are different than the other technologies. So for instance, we tested this in one of the data sets called Merfish, originally invented at Harvard, but we also used this data set to demonstrate the idea. And for the same cell, you're looking at about 130 genes colored, right, in different part of the cells, right? So this is the same cell again over and over, but on the right panel, we just highlighted different gene subsets, right, just to show how it looks. Then you can create these heat maps of correlations across patches. You take the cell, chop it into small patches, then you correlate the RNA molecule densities across these patches, and this is what you get. And here, basically, you can at least understand the enrichments of RNA across different patches. Then you could look at the neighboring RNA molecules, which gives you high correlation value across the patches. But if the molecules, then red and blue, they are not nearby or they are not neighboring, then these patches will give you low correlation scores, right? So that's why you can use this metric to quantify non-neighboring. Then you can again start with the cell, zoom it in, and create these nearest neighborhood networks. So basically, you start with a central cell RNA molecule here. Then this RNA molecule, you find the nearest 10 neighborhoods. The nearest 10 neighborhoods, you count this, and you repeat this process for every single RNA molecule until you create this large network, right? And when you're creating this large network, so this is the large network, you start with looking at 10 nearest neighborhoods, then you do 1,000 permutations, then we perform z-score analysis of these permutations, especially for RNA neighborhoods, and that tells us the map of small network here or large network, and blue shows that they are not neighboring, and red shows that they are neighboring, right? Then these z-scores are also illustrated for this orange part of, for this purple part of the cell. And here, different RNA molecules, they are giving you different scores, right, based on how close they are, right? For instance, this blue and MALAT and THB-S1, they are not close to each other. Therefore, your z-score is very low. But for instance, these top two, the IG F2R and FBN2, they are near each other. That's why they are giving you high scores. Then we also tested the same analysis by gene in another cell line, right, for the same technology, but this is a different cell line. Now this cell looks different, but we are getting the same different correlations here, right, for the second cell line. But the concept technically works, right, in even different cell line. Then when you look at two cell lines, right, and compare them for the same genes, right, fibroblast is one cell type and U2OS is another cell type, then you could see similar RNA neighborhood patterns, or you could see different RNA neighborhood patterns. So even if that you select two genes that are the same genes, across cell types, you might see similarities or differences, right? Then you could also compare these across many cells and generate all these patch correlations, right? And they compare them across the cell type. So which means that SPA-GNN, this technology can tell you, this algorithm can tell you RNA neighborhood differences across different cell types, even if you're looking at the same RNA. Alternatively, if you again look at the two different regions of the fibroblast versus the U2OS cells, and you pick three different cells here, right? And then you look at their nearest neighborhoods of RNA molecules, then you may obtain distinct RNA neighborhoods. Some of them might be similar, but in this case, the blue, which is fibroblast, it has a lot of more proximities or the neighborhoods of RNA compared to the U2OS cells. This suggests that there's a cell type specific RNA neighborhood is going on. Then beyond just looking at these images, we combine this across, for instance, 200, 300 cells. Then each dot corresponds to a single cell here. Then we classified these cells on a t-SNE or high dimensional reduction map, right? If you just use single cell RNA count and look at two cell types that I showed, then they're decently separated. I mean, they're close to separation, but they're also a continuum. Or if you use patch correlation, then still they're closer. But if you use the network or these patches or different regions of the cell, if you use those and then look at their variability, then you can perfectly separate this. For instance, by looking at the confusion metrics, you expect these diagonals to be high and other opposite black diagonals to be low. And the network variability gave us the perfect separation of cell types, right? The fibroblast versus U2OS. because it's the most sensitive one for the cell type separation. Then you could use less number of genes and perform this analysis, and still you will have the same similarity of the clusters with 0.9 RAND score, which suggests that this analysis can also work with less and less number of genes. You may not always need hundreds of genes. Then you could also compare 100 genes and 50 genes and define these networks and look at specifically the neighborhoods. And neighborhoods are preserved across the subset of RNA-RNA pairs, right? So that also suggests that number of genes doesn't necessarily change the RNA-RNA neighborhoods. Then, alternatively, we did this technology. I think there was an image it didn't show here for some reason, but we did this technology, right? We used this technology to compare stem cells used for regenerative medicine treatment. So the first two types are human umbilical cord mesenchymal stem cells. The second one is human bone marrow mesenchymal stem cells. And the third one is human chondrocytes, right? So this is actually an output of the mesenchymal stem cell differentiation. But we wanted to compare these two cell types to the third one. And essentially also we wanted to see the differences of HUC and HBM. Even if they are mesenchymal stem cells, still we expect them to show some differences. So when we perform this, again, analysis of single cell into multiple patches, and you have the cluster numbers, then we have the correlations across these, right? And we then zoom into different regions of these stem cells and perform the nearest neighbors analysis, and you'll be obtaining these networks of RNA molecules, right? Then in the same cell, for instance, for this cell, different part of the cell shows a different RNA neighborhood network. And another part of this cell also shows the second cell type shows different RNA neighborhood networks. Again, not only single cell now varies, but different parts of subcellular regions also vary. And then finally, this third cell type, right, in different color regions, right, the green region versus the pink region, we all have different RNA neighborhood networks. So, but using this kind of, again, network variability concept, by using the RNA count or expression. So this is how you do normally cell classification. You just use the RNA count and you classify genes, or you compare the correlation across these small patches, or you look at the network variability. So what we found out is that actually you can define cell type separation better, right? With this is only count, this is only patch, and the third one is the network. Again, you want this colorful diagonal to be high and black diagonals to be low. And we got the lowest black diagonal numbers in the confusion metrics, right, to achieve a better separation of cell types. So this suggests that actually the network neighbor variability approach can be a good way to separate the cell types. Then you could also truncate, look the same cell into a smaller cell, but just truncate from the edges, and you'll obtain still similar patch architecture. Then we could also look at the same cell and another cell type, right, the stem cell, for instance, and measure the genes responsible for cytokines. And we every now and then observe some of the genes are actually localized at the edge of the cell. Then you could use a different geometry of patches. Remember the previous geometry, it was a US map almost, but now you're looking at the almost geographical map of the latitudes and altitudes almost inside the cell. Then you could also define these RNA neighborhoods across different parts of this cell. Then using this information, you could compare the cells that have edge expression versus non-edge expression. For instance, this cell on the left shows the edge expression, but on the right, you're looking at that these edge expression ones are only the pink ones, or this color, bright color, but blue ones are the non-edge expression ones. Then you could use this concept to combine RNA and protein neighborhoods. So for instance, here, collagen 1 is a gene encoding for collagen molecule, most abundant molecule in the human body. And these are the green molecules. So then the CoNA and WG are protein stains for endoplasmic reticulum and Golgi molecules. So we know that actually collagen is associated with endoplasmic reticulum, right? Just because it is playing an important role when the collagen is secreted, right? The ER basically regulates this process. Then we further, again, did this a patch map, the US map type of map inside the cell. Then we created these networks, and we showed that when you look at correlations and z-scores, particularly the blue, which is that the collagen RNA molecules are enriched on ER molecules, but not Golgi, right? Blue tells us that the collagen is enriched on ER. Again, it tells you that the collagen is the neighbors of endoplasmic reticulum, but not Golgi molecule. So this was the story about how RNA neighborhoods can be measured in a subcellular architecture, right? So next, I'd like to switch gears to signaling biology to understand how signaling molecules, which are proteins, right, are organized in the cell. And we are studying signaling because for especially lung cancers, unfortunately, existing chemotherapy approaches, they can only extend the lifetime by 5 to 11 months. But the most recent drugs, which is called osimertinib, those are tyrosine kinase inhibitors, so they can extend the lifetime by about 20 months. So, but still, even if they extend the life by 20 months, some patients, the red ones especially, they become resistant, unfortunately, and we lose our loud ones just because of these. And we think what's happening in these kind of red patients is that the signaling pathways are so complex that even if you block this EGFR, right, this molecule, right, this is a signaling pathway, still the other pathways, they regulate the process and then they're leaky. They basically bypass the drug as well as the effect of the drug. Then they lead to finally the growth of tumor, right? They don't get basically suppressed. So therefore, our goal is to be able to use this technology, implement it on a microscope using microfluidics. We perform antibody stains of protein, and then we repeat this multiple process. And this is how it shows multiple staining and labeling. Basically, we are now using robots to perform our experiments, right? Of course, students and researchers are great in pipetting, but we are trying to reduce these times, right? And then finally, we are generating these kind of setups where you have the tubes, the autosamplers, and then the microscope has this stage that has the multiple fluidic inputs and outputs. And you just basically take the sample, put it on a microscope, and study these cells. And this is a technology that we use to understand signaling resistance. And when you compare it to the other technologies, the red ones actually are important. We are cutting down the experimental time to only a few hours per cycle. That allows us to generate rapid profiles. And specifically, what we want to do is to localize protein molecules in the cell by images, then use these images to define signaling networks. Until now, what people did is that they took the cell, they used an antibody molecule, they targeted all these proteins, then they pulled down all these proteins and looked at which protein was bound to which protein, right? But now what we want to do is that don't dissociate the cell, just keep the cell as is, then tag protein molecules and measure the distances and their interactions in the cell. And this is how we do these things, right? So for instance, each rectangle here shows 3 cells. But this is, of course, among a large plate of experiment where you have like thousands of cells. But then these 3 cells show epigenetic markers in the nucleus, and the signaling markers are in the cytosol, right? I guess can be seen in these images. And then the inside of the cells, you see a dark hole here because the proteins for signaling typically are enriched in the cytosol, but not in the nucleus, right? So therefore, you see these holes. But then interestingly, you can do 2 things. One is you can identify the nuclear region and cytosol region, and the red here is the nucleus. Then this type of an analysis allows us to study the translocation of protein molecules between nucleus and cytosol, right? And hence you can understand the signaling dynamics. Then we perform correlation or intensity analysis across multiple single cells and their pixels. And for instance, the MOL2 signaling molecules, they show correlation in these multiple single cells. Then we perform data science with this approach. For instance, take all these 30 markers, multiple protein molecules, put it on a single map of UMAP, right? So this is a UMAP. It's a high dimension reduction. Then color them using light and coloring. These colors then are projected back to the cell image. So this outside boundary is the cytosol of the cell. Inside boundary is the nucleus of this cell. Then the colors indicate the colocalization of subset of signaling molecules, right? If you have AB molecules, it will appear here. If you have CD and EF molecules, right, then they will show different groups. So this suggests basically the cell has this very complex signaling organization. Again, back to the architecture, signaling molecules even use different part of the cell to perform different tasks, right? It's actually important to compartmentalize even signaling molecules. then how these architecture of the subcellular regions look, right? You could see all these individual single cells. They are totally different, right? So you have basically one apartment complex, you have another apartment complex, and their architecture is different, right? So then you could see exactly the same thing here. All these individual cells, they have different signaling architecture, right? And when you zoom onto the same colony even, right? Let's say you have one apartment complex, and inside each apartment, of course, you have a different organization of the intra apartment, for instance, where the chair is, where the TV is, right? So then you're looking at all these protein molecules playing a function in different parts of the cell. For instance, this green part that I indicated before was Wnt-AKD molecule, right? The other part was PAKT. And this red part is the, again, epigenetic molecules, right? Here, these are the epigenetic. And mTOR is also on this side and things like that. So basically, it shows us how signaling organization can be different in different architectures of these cells. And here's the three-dimensional visualizations of this, where you're looking at the brown as the nucleus, and outside boundary is a single cell. And then the other colors show the signaling colocalizations with subset. For instance, this blue one is different from one cell to another, and it suggests that the single cells are heterogeneous, and that's also important to control signaling resistance. And to study our initial hypothesis that how signaling, spatial organization of signaling molecules are altered by disease, and then how that leaky signaling pathway can give rise to resistance, right? We then tried this direct treatment of osimertinib, and this is used in clinics. We compared the direct treated with untreated one to see how it affects our cells. First, when we perform the multiplex experiment, we start to see that the PAKT pathways, they get suppressed, and then PEGFR start to get more discontinuous, which means that they are blocked on the cell surface. But then the Wnt start to become more higher expressed, just because that's a leaky pathway in the cell. So Wnt resistance is known in lung cancers, but we also observed this at the cellular level. Then we compared the untreated and treated cells, and PEGFR and PAKT were the markers suggesting that the drug worked. Then we also compared cells that are treated with control and all the way up to 60 nanomolar concentration of drug. And the more we treated the cell, the more the PEGFR got more discontinuous. What does it mean by discontinuous? Do you see how these patchy green here, those ... structures, they are discontinuous. But in the original one, PEGFR were more continuous. So everywhere you had a PEGFR. So this suggests that physically the molecules are bound, and then they are basically inhibiting your molecules. So then we compared no drug with drug at the single cell level, and the blue ones are with drug and red areas are without drug, and each row is a single signaling molecule. These are proteins. We were able to also recapitulate the stratification of these. Then we did the same high-dimensional clustering of pixels in the single cells. So here, these are the, again, cluster colors coming from 30 multiplex markers. And in the control versus drug-treated one, we observe clear differences, right? How do we visualize it? We will use network visualizations. For instance, here, each bubble shows the percentage of cluster of all clusters. And then the pixel-to-pixel closeness will be the blue lines. So they're basically spatial proximity. If you have a blue pixel, right, and a yellow pixel, then between them, if they're very long, then you measure this in the image, then you project it onto here, and then it will be longer, right? So then in the control versus direct-treated one, what we observed was a direct-treated one started to break these distances across into smaller distances, right? And then these bubbles start to get bigger. So for instance, 2 and 6, in the previous one, it was smaller, and this one was high. After drug treatment, it becomes higher. This suggests that the Wnt pathway is leaky, even if there is a drug treatment occurring. And for instance, the 14 here is getting smaller. This suggests that the drug treatment is working. But Wnt pathway is suggesting that that's becoming more prevalent among the clusters of your cells. So then the 6 and 2, they're getting bigger in terms of the dot size, right? So that's important to distinguish. Then we did this also in patient tissue. So here's the tissue architecture again. The yellow is cancer, and the green is non-cancer. We just focused on the cancer regions and showed how signaling molecules are distributed differently across different regions of this tumor. Then we compare these across 21 patients, and for each patient we have 3 different replicates. Then when you zoom in, it's basically a heterogeneous expression across tumors, and particularly this bright area corresponds to tumor area, dark area corresponds to non-tumor area, which means that tumor upregulates signaling, but the level of upregulation is unique to each patient, right? It is heterogeneously expressed in each patient. Then in the last part of my talk, I'd like to highlight the metabolism and cancers. So as you know, metabolism is altered in cancers, particularly it is increased in cancer cells, and immune cells also, they reprogram their metabolism. And also our life also alters our metabolism, right? Depending on how we exercise, how we eat, fat diet, or we are obese or not, right? Or do we sit every day, or we stand up every day, right? So this metabolism alters. So we suggest that actually we should think about cell and tissue architecture from the point of view of how we can incorporate the lifestyle and metabolic measurements such that you prescribe drugs, right, for especially cancer patients, considering both. And then you might be, and you might end up getting better treatments, right? Because we know a lot about us and the tumor molecular profiles, and then we can treat them better. But the problem is that all these metabolic assays are also like making smoothie. They're not giving you the individual single cells. So we think that we can study single cell metabolism across tumors, right? And also we can compare metabolism across patients. And when you zoom into a tumor, that's what you see. The normal tissue will be on the left. And inside of the tumor, you see all these multiple interactions of cells that some of them actually, they compete with the molecules, right? They actually, for instance, secrete lactate, then they change the immune profile, or they compete for the glucose inside the tumor region. So there's a lot of competition and interaction going on. And we developed a technology for this where we started with H&E imaging, or the histological imaging of this tissue, annotated different regions. Then we stained another separate tissue with these antibodies that are tagged by not fluorophores, but something called isotopes. We are using isotopes because we can look at 35, 40 molecules all at once. Then we perform one imaging using a secondary IMMS spectrometry. It says 3D-SMF, this is our unique name, but it's a metabolic imaging technology. Then we perform protein imaging, then we overlay these two. And in the imaging, we perform processing and clustering and data analysis. So this is a crowded slide, but you're gonna see in the next slides. So first, in the tumors, we can use protein staining using isotopes and on the antibodies, right? Remember isotopes. Then you can look at all these proteins for cancers, stromal cells, macrophages, and T cells. Then when you zoom onto different regions, you can color these cells by their identity. Then you could then create these graph networks right at the end. So each dot corresponds to a single cell type. Then we can look at their interactions in the different parts of the cell. Then you start with this again, isotope and antibody complexes, right? You can use 30 of them and stain your tissue, then perform metabolomic imaging, protein imaging, and then combine these two data sets. And zooming into one lung cancer patient biopsy, here the orange color shows the phosphate molecule found in your DNA, but there's no label. This is label-free. Then spatial metabolomics shows you all the metabolites distributed across your basic nucleus region. Then nuclear metabolites that are tagged by antibodies targeting the histone antibodies inside the nucleus. Then also these right colors are the surface receptor antibodies that are targeting the CD8, CD68, collagen, and Ki67, and all that at the cellular level. So it tells you the cell type. So then one can quantify the cell types from the protein information, then overlay with metabolic information. That's what we did. At the single cell level, we got the lung cancer patient samples on a microarray. Then we performed proteomic classification. Right here, the C shows how the bright colors are the immune cells and the cyan color is the cancer. Then we grouped this as tumor, stroma, and others, and showed that the tumor has a higher expression of several lipids, glycine, compared to stroma or non-tumor areas. Then we could also go back to the original image and color the metabolism of lipids, glycines, and cholesterol at the single cell level. So this is actually very exciting for us because for the first time, we can do all these kind of studies for hundreds of metabolites at the single cell level. Then the data science allows us to compare the profiles of about 23,000 cells for 40 metabolites. And bottom, you have these 20 different proteins. Then you could use the protein for cell types and metabolites for metabolic analysis. And further, you can subgroup the metabolites into glucose-associated, cholesterol, and amino acid associated, right? As well as the lipids. So then several tumor regions have higher expression of lipids, but several others are higher in the stroma regions, which suggests that lipids are depleted or generated in tumors. And it's basically time-dependent and energy-associated process. Then we can further associate the tumor immune stroma, separations in the tumor. Right here, you're looking at 3 different colors in the tumor region, right? And then by counting number of immune cells and their metabolism, you can create also these immunometabolism maps. Immunometabolism is the metabolism of immune cells inside cancers, right? That's what you're looking at. So then we can also compare the metabolic profiles across patients. But one thing that I want to highlight is that because this is a multimodal data, we have metabolites and proteins. You have the 2 different UMAP or the high dimensional representations here. If you were to use the existing data integration approaches, so this is about bioinformatics problem, right? You can look at the one data, it shows all these colors. The second data shows similar colors, but there are a lot of shared features. But what we found out is that in our case, the metabolites and proteins are 2 different worlds, because proteins can tell you the cell types, but metabolites, if you have a lipid on the cell surface or cell membrane, that lipid may be every cell type, right? So you cannot basically distinguish them. So therefore, my student actually developed this variational autoencoder approach that combines 200 metabolites with 20 proteins. Then this is integrated in the latent space of this variational autoencoder. Then we create the cell type specific maps of metabolites, right, in multiple patients. Well, what did we learn? We learned that the metabolism and protein expressions are unique in different patients. Then several metabolites are lowly varying. For instance, each row here is a different patient, and then they are lowly varying. But also there are some other metabolites that are highly varying, right? So the blue and then orange one, they're highly varying. Then, for instance, when you use the t-SNE after the data integration, then each color shows the different regions for the same patient. For instance, orange, they're closer to each other. These are from the same patients. And for instance, here the purple, they're from the same patients, but different regions. So it shows that the metabolic changes of cell types are patient specific, but also they can be different from one and another, right? So it's a very complex regulation. So we think that actually now you can combine immunotherapy with metabolite supplements to increase the survival. And what we propose is that now use the lifestyle, diet, and BMI for designing patient therapies, right, especially for immunotherapy. And you could also think about simple diet prescriptions to boost the immune system, especially for immunotherapy responses, right, how they increase them. So there is a good in the Cell paper, right, demonstrating the initial bits of it, but that's what we are moving after. We have now the technology that can measure single cell metabolism and proteins. And now we'd like to also use metabolites on top of immunotherapy to be able to increase the responses to these lung cancer treatments. And now I will be summarizing. So we have shown today how we could do graph-based images of proteins and metabolites, right, where you can look at single cell differences. We also talked about spatial signaling networks and how these distinct colors show the protein molecules and their networks. Then in the first part also, we talked about RNA neighborhood networks, where we also highlighted how distinct part of the cells show different networks. And I'd like to also highlight that the virtual reality can be also integrated into such an exciting data where you can discover the patterns, right, of neighborhoods, right, RNA neighborhoods, or protein-protein interactions, right? And all these at the cell using virtual reality. So this was published in Cell Reports Medicine. You might want to read this. It's a fun article showing how biomedical field can use virtual reality. Again, even for bioinformatics community, this could be enabler, right? It's an interactive technology. So with this, I'd like to wrap up, and I'd like to thank to our collaborators from Emory University and Georgia Tech. And I'd like to gratefully acknowledge the support of generous funding agencies. And of course, without the hard work of the team behind such a work, right, this would have not been possible. So since from 2019, when I started my lab, now we are growing, we are branching out to multiple directions, as well as collaborating with large consortiums like this one. And with this, I'd like to conclude, and thank you again, Bioinforange, for this kind of invitation. And I'm happy to take any questions if you have. Thank you.
Thank you very much for this precious presentation, Dr. Coşkun. Then I'm starting with the first question. Why are preclinical models inadequate, especially in tumors? What are the challenges in developing prototypes that mimic the tumor microenvironment?
Oh, yes, I think this is a great question. We just actually published with a colleague of mine a paper, a model to mimic tumor microenvironment, right? So this microenvironment is typically framed as organoid. There are mini organs, and you basically mix cell types with the extracellular matrix compartments in a dish, right? You take the matrix with cell types, you mix them in a dish, and then you let them self-assemble, grow, and make it synthetic tumor microenvironment, right? In our last work, which was published in Nature Materials, that's what we showed, that the lymphoma organoids can be recapitulated by using the chemistry that's mimicking our body's collagen chemistry, right? And then you mix with the cell types, and then they grow and give rise to the functional signaling and, for instance, metabolism, right? Profiles of cells in the organoid platform. So it's actually combining our also spatial omics technologies, right? Because important that once you make the synthetic environment, you want to also understand how they're similar to the real tumor microenvironments, right? You could actually look at their expressions, light, high, low, as well as their spatial architectures, right? Are they preserving the cell neighborhoods, right? Are they preserving the original architecture, right? So that's why we are currently actually developing experimental as well as computational frameworks to establish this, right? Synthetic tumor microenvironments benchmarked by the architecture concepts, right? And then use iteratively, right? Use chemistry and bioengineering to make additional, new synthetic tumor microenvironments. But it's a very important one, just because also right now there's a trend in the US and in internationally also that you want to decrease the animal use in your research. Can you use an in vitro platform like an organoid to be able to study the disease response, right? And then at the end of the research, you should be able to replace the animal studies with just a bunch of microwells, right, or microfluidic chips, right? Then you can actually get the answers of which drugs will be more efficient, which patient will most likely to respond to which subset of drug, right? All these kind of responses. Therefore, I think your question is really important for us to impact the future of medicine by reducing the animal use, right, but increasing the clinical relevance of synthetic tumor microenvironments.
I appreciate your answer, sir. This is from one of our YouTube participants. And next question is, a current challenge in two-dimensional analysis of tissue is the detection of structure independent of the cutting angle. How can we get robust morphometry of tissue structures?
Thank you, Ayşenur Kaya, I think, for this great question. So I think what Ayşenur is specifically referring to is that when you look at the tissues, right, there are a lot of features. Some of them are sharp, like their edges. Some of them are smooth, they are more continuous. So identifying the features in this tissue, along with, of course, expression of many markers, is a challenge in image processing. Actually, I'm teaching the medical image processing in my institute, so therefore we teach all these filtering and other methods, right, to perform edge detection and beyond. But now classical edge detection algorithms, classical image processing algorithms are lacking behind. So what we are doing in my lab actually is to be able to identify these, for instance, graph-based architectures, right? You make the individual dots in your tissue, right, as a graph. Then you take that graph and then project it in a machine learning, deep learning interface. Then that interface, as you compare multiple tissues and multiple different tissue regions, is much more robust, right? So you might want to check, or I can refer to new articles studying the morphology using this kind of approaches. For instance, there's an algorithm called Vampire, developed by Johns Hopkins professor Dennis Wirtz, and they're defining the cell shape by getting the edge of the cell as a point structure, right? They basically take a cell and make it point, and then they take these points and their positions and project it on a PCA, or principal component analysis, high-dimensional, three-dimensional space. Then there, they can go back and say, oh, actually, my cell type is round. My cell type is elongated. My cell type is, like a square. I mean, nothing is a square cell, but my cell type is like square, right? So those kind of methods actually are much more robust. So therefore, I highly recommend considering integrating morphological analysis of tissue and tissue structures, right, by not just the filtered or image processing techniques, right, but also new machine learning algorithms to group them as the graph-based methods, right? That's what I would highly recommend. But thank you, it's a very good question.
Thank you so much for your answer. The next question is, why are targeted therapies for neuroendocrine prostate cancer and the castration-resistant adenocarcinoma tumors not available? Could tissue architecture be modeled?
Thank you. Well, this is very specific. I'm surprised. So this refers back to our paper also with the same team that we published 2 years ago in Advanced Materials, where we also use the same chemistry, hydrogel chemistry, to mimic the prostate cancers, right? And then these are the NAPC, right, and CRPC adeno. So these are specific therapies that shows a different molecular profiles, and they are not necessarily responding to what's available as a treatment, right? So I think what the question is asking about, tissue architecture can be modeled. Yes, I think if you can model the prostate architecture, distinguishing the NAPCs, CRPCs, which in that Advanced Materials paper 2021, we have the organoids made by our colleague, and we also looked at the NAPC and CRPC tissue microenvironment in also cancer patients, and we saw a lot of similarities, right? We saw a lot of similarities, but there were a lot of still differences. So currently we are optimizing this further, and as you mentioned, if the tissue architecture further mimics the exact NAPC and CRPC tumors, then at the end of the day, you'll be able to mimic this much easier and better, right? In terms of finding the better treatments for these unique prostate subtypes, prostate cancer subtypes. But thank you. I think this is a very nice question, suggesting that clinic has some unmet need, and bioengineering and bioinformatics can meet together, right, to answer these questions.
Thank you so much for the answer. Our next question is, how can we identify and quantify organ-specific microanatomical domains from biological images without previous knowledge?
Okay, great. I think without previous knowledge is key there. Thank you, Nagihan. So how can we identify quantify organ-specific microanatomical domains from biological images? So assume that, for instance, we collect tissue images from multiple organs, right? And let's say you do kidney, prostate, right, lung. Then you get all these H&E features, right, or the histological features, or even immunofluorescence-like features. Then you create these, maps of their cell stains, and then their anatomical features, right? You can look at, this is my epithelial region, this is my, other part of the, prostate, or this part of the kidney, right? So then using these three, what we would do is we would take the single cell information from this, basically color one cell at a time, right? One cell at a time. For instance, in the prostate, you have 100 epithelial cells. In the kidney, you have 200 epithelial cells. and in the prostate you have another 300 epithelial cells. So there are multiple organs basically. Then we would also take their distances between them, right? These 100 cells, they have very close distances, but in the kidney, these 200 cells, they have separated distances. They are distant from each other, right? Then by using this two information, you would try to create a comparative analysis using a latent space model, right? That we just discussed, the metabolite and protein data that I mentioned, right? One of them has more markers than the other. But then what you do is in the deep learning space, if you convert them to a latent variable, right? Latent variable, then that latent variable will be able to combine them all in a single map, right? Then you don't need any previous knowledge, right? You don't need any specific training. But all you do is when you do the latent space combination, then in that combination, you can actually then project them on a high dimensional visual or cluster that will tell you the organ specific features on a heat map, right? It can tell you number of cells, it can tell you the distances in between them. But again, it's important to compare them in the latent space, but not originally. If you just take this data set, put it in a heat map, it will be difficult to combine because there are different cell types, there are different distances and all that, right? So therefore, you need to be able to distinguish the two. But this is a great question. I don't want to, of course, take too much of your time, but I would suggest you think about latent space combination for this.
Thank you for your answer. And next question is, in spatial, technologies now enable the characterization of heterogeneity of tissue architecture and the opposite of the novel data can detect spatial information. With this limited information, how could it be made clear the relationship between cell-to-cell interactions?
Yes, I think this is a good question. So I think this refers to the fact that most of the spatial technologies, they choose the genes to measure. Let's say you measure 100 genes, but the genome-wide information is 20,000 genes, right? But 20,000 genes RNA sequencing, for instance, can discover new information. So what we are doing currently is to integrate both worlds, right? By using, for instance, the information from 1,000 genes, you can define the cell-cell communication interactions, but then look at certain interactions of, let's say, subset of 20 genes. Those 20 genes go back to the original discovery data, RNA-seq data, and find, similar to these 20 genes, what are the other genes doing similar functions, right? Then if you integrate these two data sets, then you can infer that, oh, these 20 other genes that I didn't measure in my spatial could also be related to my cell-cell interactions. And as a result, you can fill the gap in your information. I think the key is the hybrid analysis between the spatial and non-spatial data together. There are actually very nice works recently published by our colleagues I can refer to. If you're interested, just send me an email. I'll send you some of these recent publications. And that's what we were also doing in our case. If you look at, for instance, 100 genes in stem cells, and then look at the databases measuring 20 genes for stem cells, then can you actually find the differentiation of stem cells in the differentiation path, right? Much better than, right, just the spatial only. Because spatial is really limited in terms of number of genes, right? But then using the non-spatial data, you can find associations of those non-measured genes, and that can tell you this differentiation path much better than only looking at the spatial data.
I appreciate your answer, sir. Our next question coming from YouTube participants. Was clonal heterogeneity in samples evaluated in your SPA-GNN study? Did you decipher the genetic purity of MSC colonies? Thanks.
Thank you. This is a very interesting question, Bedish. And so in terms of clonal heterogeneity, so currently we have not compared this for MSC or mesenchymal stem cell colonies for how a genetic purity of a colony compared to another genetic with a different clone, right? How they compare with each other. I think that's our next step. And we would like to compare with, for instance, take an MSC and start mapping their colonies, right? You have this colony with this clonal architecture, another clonal, and another clonal, then fix the sample and continue, right? And then continue by using SPA-GNN. I think that's going to be our next step. And that will be very exciting because then you can look at how clonal information generates different subcellular networks. But thank you for the suggestion.
Thank you for your answer. And our last question is, which advices Dr. Coşkun may give for students studying for scientific fields?
Thank you. I think you are doing very great work here with Bioinforange, integrating bioinformatics to research programs, big data analysis, machine learning, AI to biology is the future of medicine. I highly recommend students to learn how to code as early as possible, right, in their careers. And also I highly recommend them to reach out and find networking opportunities within this community and do internships, right, summer studies, as well as pursue research opportunities as early as possible in these fields. And I hope in the near future, AI combined with biological, problems will really take over all the possible strategies that can help us solve the challenges of diseases, right, and many others. I think this is what I would recommend. And I think that you're doing really, again, an important key piece here, which is bringing the people internationally all into one online platform, right? And I highly recommend that your students here in this platform should reach out to these highly, interesting, research groups, and then start working with them together, right? And that will, of course, increase the impact of our students' future studies.
Thank you so much for your advice.
Yeah, thank you for the kind invitation. And sorry, I think we're a few minutes late, but yeah, I'm happy to answer any questions if you may have separately on offline. And my email should be online, or it should be also with you guys in the Bioinforange. They can get my email from you guys, and I'm happy to answer offline questions. And thank you again, everyone, for organizing this fantastic event.
On behalf of myself and all our participants, I would like to thank you for spending your valuable time, transferring your knowledge to us, and for this informative presentation, sir.
Thank you. Have a nice day.
I wish you healthy days.
Thank you. Teşekkürler. Bye-bye.
At 7:15 p.m., Dr. Ming-Tommy Tang will be with us to give a speech on Learn Computational Biology the Right Way. Thank you.
Bioinfocongress IV. Dr. Ming-Tommy Tang. Learn Computational Biology the Right Way. Dr. Ming-Tommy Tang is now the Director of Computational Biology at Immunitas Therapeutics. At Immunitas, they employ a single-cell sequencing platform to dissect the biology of immune cells in human tumors. Dr. Tang is a computational biologist working on genomics, epigenomics, and single-cell transcriptomics. Together, Dr. Tang has over 10 years computational biology experience and 6 years wet lab experience. He uses R primarily for data wrangling and visualization in the tidyverse ecosystem. Dr. Tang uses Python for writing snake-make workflows and reformatting data. He is a Unix geek learning shell tricks almost every month. He cares about reproducible research and open science.
Dear Dr. Ming Tommy Tang and our participants, welcome all of you to our Bioinfocongress IV events. Dr. Ming will give us a talk today on Learn Computational Biology the Right Way. At the end of the presentation, we will have a question and answer section prepared by our team and consisting of questions from our participants. Dr. Tang, welcome back.
Thank you very much for the invitation and the introduction.
You're welcome. If you wish, you can start your presentation.
Sure, I will do full screen share slideshow. Can you see it? Okay.
No, right now. So we can see it.
Okay.
If you wish, you can start.
How about now?
Yes, we can see it.
Okay, so I will go ahead. First of all, thanks for the invitation. It's my honor to present at the Bioinfocongress 2023. My name is Ming Tang. You can call me Tommy. So I'm currently the Director of Computational Biology at Immunitas, a single-cell genomics company committed to develop novel therapeutics for cancer patients. So today I'm going to talk about learning computational biology the right way. I think my talk is right at the right timing because our last speaker, Dr. Ahmed, also said learn how to code as soon as possible. So I hope this talk will give you some inspiration so you can learn how to coding as early as you can. And I hope my experience can, so I'm going to talk about how I learned it the hard way, but I want to show you the right way. And I hope my experience can help you to avoid the mistakes that I made. And this is my Twitter. I tweet mostly about bioinformatics, the tools, the papers, and some tips. Sometimes I also complain about it, about bioinformatics. And this is my blog, and I post some of my learnings there too. So in this slide is a schematic view of my career trajectory. So I graduated from Shanghai Jiao Tong University in 2008. And I started my PhD at the University of Florida. And then I was trained in a wet lab. So everything was fine. It was great. I had my 2 first author publications in well-respected journal at 4th years of my PhD, not until one day when my advisor asked me to analyze a public ChIP-seq data back in 2012. I couldn't open it with Excel, so I was forced to teach myself Linux, R, and Python. So I then did a 3-year computational biology postdoc with Ro Vahak, who was leading the GCGA glioblastoma project. And after my postdoc, I moved to Harvard FAS Informatics to work on single-cell RNA and single-cell ATAC sequencing data. So in 2020, I joined Dana-Farber to lead an NIH-funded consortium project called Cancer Immunologic Data Commons, in which 4 cancer centers carry out immunotherapy clinical trials, and they profiled patients using whole exome sequencing, TCR sequencing, RNA sequencing, and ATAC sequencing. So our job was actually to process all data securely over the cloud and give them back for downstream analysis. So I stayed in Dana-Farber for almost 2 years, and now is leading the computational biology team at Immunitas. So I have a great interest in promoting open science, transform biological data science education by teaching. So I consider myself as a genomic data scientist or a computational biologist. So I have extensive experience in analyzing sequencing data for genomics, epigenomics, and transcriptomics. So I use R primarily for data wrangling and visualization in the tidyverse ecosystem. I use Python for writing SNAKEMake workflows, reformatting data, and Laravel for the Unix commands. I share my experience of sequencing data analysis in my GitHub repository. So you can go there and see, find those resources. It will be helpful for you. And the reproducible research topic is always near and dear to my heart, and I will touch on this topic later. So I just want to show you the transformation using these 2 pictures. So in 2013, I was pipetting on the bench, doing qPCR, cell culture, and Western blot. And in 2023, I am a computational biologist. So if I can do it, you can do it too. So it takes me almost 10 years, but you can do it shorter with a clear roadmap. So we are in the age of data exploding, right? This is an old stats from Sean Davis. So this is how much basis we have back in 2018, 1.8 times 10 to the 16 basis in the NCBI sequence read archive. This is equivalent to 6 million human genomes, which is 9 billion bases. 3 billion bases. And every day, they process roughly 3 TB data. And this is really old, like 5 years ago. And nowadays, you can imagine the data is exploding even more quickly. Now we have single-cell data, image data, spatial transcriptome data, and it can be overwhelming without any data skills. So I believe many of you work with data, but which category are you in? So I really like this cartoon borrowed from Thorson, who is a bioinformatics professor in Australia. So if you know computer, and stats, you are a data scientist. But if you happen to have the domain knowledge, in this case, biology, you are a Superman or Wonder Woman. I would argue actually a bioinformatician or computational biologist should know all the 3 fields. So I will use bioinformatician and a computational biologist interchangeably. There are some differences, but I'm not going into the nuances. So what should you learn to tame the data? So my first tip here is you need to learn Unix command line. So humans and computers commonly interact in many different ways, such as through a keyboard, a mouse, a touch screen, or using speech recognition systems. And the most widely used way to interact with personal computer is called a graphic user interface, or GUI, if you happen to heard that before, GUI. So with a GUI, we can give instructions by clicking a mouse and using manual-driven interactions. The Unix command interface, or the CLI, is a scripting language allowing such repetitive tasks to be done automatically and fast. So really, why we need to use command line? So remember, the text form, a file, is still the key format of bioinformatics. And most of your work will be actually wrangling the file formats in order to feed into different bioinformatics tools. And Unix commands are perfect for wrangling the files. And most bioinformatics tools are run by command line. So if you don't know how to use command line, you can't use them, right? And it's more efficient and powerful with a single command, like here, copy all the PNG files into this picture folder, for example, this simple example. Instead of clicking all the PNG files and select them all and drag them into this picture folder. This is a simple example. And the Unix command line is omnipresent in many settings. For example, the high-performance computing cluster and cloud computing, they don't have the GUI interface. So you have to learn how to use Unix command line. So this is how it looks like a terminal. So essentially, a terminal is a command line interface, and this is the GUI interface that we are familiar with all the folders in here in my home directory. And this terminal, it's like a black box, right? You type in the commands, you press enter, and it's executing that command. But you will see the same file system represented in the terminal. So all the folders here will be the same as here in your files in your GUI file system. So I recommend either using a Mac or you can have a dual booting system for Ubuntu to have this unique command line interface. And this software carpentry has a lot of nice beginner courses to teach you how to use this Unix command line. The next tip is to learn some Python. Python is the most popular language in data science. It is easy to learn and is really powerful, right? So why in this cartoon it says, well, you are flying how? This guy said, oh, I'm using Python, right? And the good thing about all those languages, there are so many libraries of packages that you can use. So in this case, you just type import anti-gravity, and then this guy is flying. So take advantage of those nicely made libraries. So next is to learn some R. So R is a statistical programming language, which is free, open source, and powerful. And R has its strength in statistics and plotting. So for IDE, I really like RStudio. And for R packages, don't try to reinvent the wheels. So use the tidyverse and ggplot packages for data wrangling and do this and data visualization. So Bioconductor essentially contains many different R packages specifically for bioinformatics. So if you search an assay name, then add a bioconductor, you most likely you will find a package that is written by someone else. So this is the data analysis workflow from this R for Data Science book. So first you import the data, you tidy the data, clean the data, then you explore the data, then you communicate. I want to emphasize tidying the data is one of the key skill sets actually in data science. I would say 80% of the time I spend is to clean up the messy data. So again, this book is also free to read, so go there. And investing your time to learn a tidyverse is a good investment. Data visualization is very important in exploratory data analysis. So you get an idea of how the data look like. You can review the structure of the data, identify if there are any batch effects or sample swaps in your experiments. For example, in this example, like all those points, they all have the same mean, the same standard deviation, and also the same correlation. But if you visualize data, they look drastically different. So learn how to use ggplot2 or this R package, and then you can do a better job in exploratory data analysis. what people think we do usually. So they think we have a magic push button bioinformatics keyboard. So I borrow it from Thorsten again. And we have just, assemble the genome, I just press this key, and covariance, I just press the key. But in reality, what we do, so this is a typical day of my life as a computational biologist. The first thing is to install software. Second is Google how-tos and the error messages. Then read the manuals of bioinformatics tools, converting file formats, as I mentioned before, typing the data 80% of your time. The real analysis is like only 20%. So really, Google is how we learn and do things. So in this example, it's very funny, actually, cartoon here. So Rob, you use Unix, there's a bomb that's going to explode. To disarm this bomb, you simply type a valid tar command and in your first try, and no Google around. But then this guy failed. I'm so sorry. So this really shows that we use Google a lot. Even at some commonly used command, we probably cannot remember the exact syntax. So it's okay to Google. And it's even better to know some of the Google tricks. So I will share some of those with you. For example, if in Google, if you add this brackets and the small letter R, it will search all R programming related pages. And if you use like bracket and use Python, and it will actually find you the Python related web pages. So you can use this double quote to search for exact phrase. Use this tilde sign in front of word to find synonyms. And if you can use this minus sign to exclude the terms that you don't want to search. And you can also even search within a specific website. For example, you want to search heat map. You want to know how to make a heat map, but only within the site of this bioconductor supporting page. So this will search a heat map inside this website alone. You can also define a file type that you want to search. For example, heat map, then you say file type colon PDF. It will only give you PDF files in the searching results. So I hope those little tricks can help you. And again, so ask for help. So no one knows everything. And those are like 4 really popular forums to find help. Seek Answer, BioStars, Stack Overflow. Those 2 are more like sequencing data related. Stack Overflow is general programming language, and Bioconductor helps support, the help page is for bioconductor related questions. And by the way, so when you ask a question, make an effort to ask good questions. So you have to define problem, what you have tried, what did not work, and it's even better to include code. So many times I see people asking questions online like, this didn't work, and that's it. So if you don't give enough context, people cannot help you. So again, if you do not make an effort to ask good questions, don't expect other people to make an effort to answer you. That's fair, right? And I'm not lying, right? And this professor, I think is from Stanford, he was trying to find out something, Google something about snake make. And actually, he found an old post that he actually was asking the question. And then he got an answer again, for sure. So now I will switch the gear a little bit to talk about Excel. So we really want to use Excel with precaution. I know Excel is still the most primary tool that people use for data analysis, but we need to be aware of its weakness. For example, in this news, it says some of the data are lost, COVID-19 data are lost because the Excel, this old version of Excel can only like contain like 1,400, for example, rows. And in this example, actually, an Excel screw up leads to a retraction. Actually, it causes the rows to shift the data from different groups. And that's actually tell you something, because if you use Excel manually, it's really easy to make those type of mistakes. What's even worse is that Excel converts gene names to dates. And some examples here, for example, APR1 will be converted to April1, OCT4, OCT4, which is a really well-known transcription factor, right? But it got converted into October 4th. So really be aware of this problem. And it's prevailing in all those published papers in their supplementary file. So you really need to learn, for example, how to use R packages, read XL to read in the Excel file, and use R packages such as Janitor to actually clean the Excel file. So now let's dive into my favorite topic, reproducible research. So we all know how science should be done. However, you first make an observation, right? And then you form a hypothesis, perform the experiment, analyze data, and report your findings. And then you invite others to reproduce the results. But however, we often hear that one cannot replicate a published study. So there is a reproducibility crisis going on here. So according to Titus Brown, most computational research is not reproducible. So you may think experimental studies may be hard to reproduce because the experiments were performed under different environments, different people handled the mice, or different reagents were used. You may think computational studies should be easier to reproduce. However, according to Titus, a bioinformatics professor at UC Davis, 95% of the studies fail to include details necessary for replication. So it's really hard to build off research like this. So let me give you an example. So there was a scandal in Science that the researcher at Duke faked the data and built the results on a flawed analysis. And Dr. Kate's group at MD Anderson and downloaded their microarray data and could not reproduce the results claimed in the paper. So through a detective investigation, he finally found original authors had one row shifted in Excel and got all the gene names wrong. Unfortunately, the clinical trial actually built upon the results has to be stopped. This speaks to the fact that how important we should make our analysis reproducible, both for your own sake and for the patient's sake. So I highly recommend actually you watch this video, and it's very fun. I will actually share this slides to the organizer, and then you can get the slide deck. So here's another example. In this paper published in Science, rearrangement bursts generate canonical gene fusion in bone and soft tissue tumors. And in their method section, they say we detect gene fusion in regions of genomic complexity using an approach that integrates multiple independent fusion algorithms. And that's it. So without knowing the details, I don't think I can reproduce their results. Again, method matters. So why reproducibility is so hard? So there's several reasons that I come up with. But you think about on your side. The first is no raw data available. So they didn't give you the raw data. And second, scripts and data available upon reasonable request. And then you still see this actually in the published papers, which in my opinion is unacceptable. Third is a lack of method description, just like the Science paper. The version of the tools may be different. So because if you use R or Python, different bioinformatics tools, they may be built in different versions. And they can generate different results. And lastly, you may run in a different machine. The Unix machine versus a Windows machine may give you some different results. So if reproducibility is so hard, should you care about it, right? But keep this in mind. You are going to do the same analysis for sure in the future yourself, especially during the journey of submitting a paper, right? You're going to get the comments from the reviewers, and you're going to actually redo some of the analysis. And this is really for your own benefit. And now I'm in a biotech company. Our mission is to develop life-saving drugs for patients. I certainly want my analysis to be reproducible. Then how to ensure reproducibility? There are several points I wrote it down here. So first, Git version control. Second, using a notebook, Jupyter or R notebook, document every step. And you can use containers, like Docker containers, Singularity containers, which is used to containerize your computing environment so others can reproduce your analysis using the same environment. If you are doing software engineering, you want to do unit test and continuous integration of development, CI/CD. So I will talk about some of those points in detail in the coming slides. So I'm sure we all experience this on versioning our Word documents. We just keep adding version numbers after final, like final revision 2, 3, 6, and then it becomes longer and longer. And Git is a software for tracking changes in any sets of files, usually used for coordinating work among programmers. So Git records all the history of your files, so you can easily revert back to any previous version. And then GitHub and GitLab are websites that host your Git repositories. I highly recommend putting your code for the published paper on GitHub so others can replicate your analysis. And there's some learning resources here in this link. And let me talk about notebooks. So a wet lab member has a notebook to document his daily experiments. So what, when, and how he or she performed the experiments. Right? Similarly, a computational biologist should have a digital notebook to document all the analysis. And Jupyter Notebook is a Python-flavored notebook, although it supports other kernels such as R. The nice thing about notebooks is that one can put the documentation together with the code. And the code is evaluated in line. So I usually have a notebook for every analysis that I did. So how I download the data, where I download data, what command did I use? So all put them in the same notebook. So I use actually R more often. So I love R Markdown or R Notebook. It's similar with Jupyter Notebook, but it's more like R focused. Again, you can evaluate those commands in line. For example, there's a figure that is generated included in this notebook. So here I want to talk about automation. So if you want to make your analysis more reproducible, you want to automate your analysis as much as possible. Not only it helps you to make your analysis more reproducible, it also saves you time in the long run. So in this figure, I'm showing you there are two ways, the geek way and non-geek way to approach certain tasks. So when the task size increases, the geek way is that, oh, I get annoyed. Let me stop and write some scripts to automate it. So it takes some time. But then after he has it, he can run the scripts. And when the task size increases, and it actually is better than the non-geek way, which does all the tasks manually. And remember, computers are really good at repetitive work. And the good side effect of automation is you don't need to actually document it. The best documentation is automation. So write scripts for everything, unless it's not possible. If you have to do manual editing, document it in a notebook. Say, oh, I did this, did that. Document, document, document. So important things I say 3 times. And we also use this, write all this document in Markdown files. And these two are called mkdocs, which translate all your Markdown files into a really nice website. And there's some tips for automation. For example, if you have a repetitive simple task, put them into a shell script, for example, myroutine.sh. So instead of typing each individual command every time, you can just run myroutine.sh. It can save your time. And you can also use the old GNU Make. Once was designed for compiling software, but people use it for data analysis too. So more recent workflow language such as SnakeMake, Nextflow, and Widow are your friends too. And in this repo, it collects hundreds of tools that can be used to write pipelines. I would suggest you just start with one of them, I highly recommend SnakeMake, and I know Nextflow is drawing attentions to. SnakeMake is just a Python extension, so if you know Python, it will be very easy for you. So let's talk about containers. So why we need containers, right? So imagine you are working on an analysis in R, and you send your code to a friend. Your friend runs exactly the same code, exactly the same data set, but it gets slightly different results. So this can have various reasons, such as different operating system, a different version of an R package. Docker is trying to solve problems like that. So you might have used a virtual box. Think it as a virtual machine that encapsulates all the software, libraries, and configuration files. Then you can ship this virtual computer to your friend, and you can use the same computing environment to do the same analysis. And this in part solves the reproducibility issue. And I also put two links there for you to learn. So as I mentioned, the number one actual problem of bioinformatics is converting formats of input and output between different tools. But the number zero problem is installing tools. So there's so many bioinformatics tools, and many of them have different dependencies on other software. For example, a particular tool is Python 2, and another tool is Python 3. How can you do the analysis if the analysis is dependent on both tools? So Conda actually is a Python, is a package management tool for any language. And you can set up two different Conda environments, for example, one for Python 2, the other one for Python 3. And you can activate different environments for different tools. And BioConda is a life science-centered channel in Conda. And so my message here is to use Conda to manage your computing environment to help with the reproducibility. So project organization is really underrated, and not many schools teach how to organize a computational biology project. So I recommend you to read this paper, A Quick Guide to Organizing Computational Biology Projects. And I follow a similar folder structure. So on the top level is the name of the project. Then you have a documentation folder, a data folder, a source file folder, and the bin folder, and the results folder. And you prefix all the results and the data folders by date. So you don't need to actually follow exactly the same folder structure, but as long as you have a consistent folder structure, that's fine. Having a fixed project structure can help you to reproduce your results. There are two more papers that I recommend you to read. It's Good Enough Practices in Scientific Computing and the Best Practice for Scientific Computing. So they were written by the software carpentry community. I highly recommend you to read them. So one can take even one step further. So this tool called RPack called Workflow R. So it's an RPack to translate your analysis R Markdown files to a web page. And one can easily browse through the code, the documentation, and the figures on a website. And you can share this website to everybody. It's very, very neat. This is an old post that I wrote back in 2015. Some tips for coding from a bioinformatics beginner's point of view. So first, computers make mistakes. They can give you nonsense results and exit without error. So you have to actually make extensive tests before running your code. Also, do sanity check with the results, right? Check with whether the results makes any biological sense. Talk to the experts in the biology field too, whether this makes sense or not. Second, share your code, right? Even your code is correct, you want to share them so other people can look at them, and they also may improve the code. So you want to make your code reusable. So you do not want to hard code your scripts. If it takes a file path as input, make it as an argument in your script. So you can use the same script for different analysis. You also want to modularize your scripts. So usually the data could come, for example, come in different formats. Take chip sequencing as an example. You may have a script that starts processing the data from FASTQ to the final peaks. And you want to modularize the scripts into two modules, for example. One for mapping the FASTQs to band, and the other is from the band to peaks. So modularized scripts can help you because if the data come in the band format, you can directly use in your second module to call the peaks. 5, heavily comment your scripts. So it will not only make other people to understand your code better, but also make the future you to understand what you did. To trust me. So your future self will thank you if you comment your scripts. And lastly, you need to make your analysis reproducible. And how to make them reproducible, I have actually mentioned in the previous slides. I think this is my second last slide. There's some key takeaways from this presentation. So if you want to learn computational biology, first, you need to learn Unix commands, Python, and R. Second, learn how to use Google effectively. And even recently, we have this ChatGPT. I'm not sure whether you're aware of this. It's an artificial intelligence language model that can understand and generate human-like language, making it useful for tasks such as conversational AI, language translation, and text generation. And you can take advantage of this to learn computational biology. You can ask him questions. Third, be cautious with Excel. So you still use it, but remember the weakness of Excel. It may actually convert your gene names. It may actually also truncate your data, for example. Git version control your code. So you can always track how your code is evolving, and you can quickly revert back to your old code if the new code didn't work anymore. You need to have a consistent folder structure for projects, reproducible computing. And learn by doing, I didn't actually talk much about it, but the way I learn computational biology is really learn on the job, which means if you're in a wet lab, but the lab is doing some sequencing experiment, take that project and then learn how to analyze the sequencing data by yourself. And I learned how to analyze chip sequencing data because I had to analyze the public available chip sequencing data. So learn by doing. And lastly, I want you to focus on your strength. I know we are not a computer scientist, we are not a statistician, but really biology, the domain knowledge is our strength. So I will focus on your strength, but gradually learn other skill sets, for example, the computer languages, programming languages, and the stats. And this is my last slide. I want to thank Samir Emming, my colleague during my postdoc in the Hack Lab. He taught me a lot of new things when I was a beginner. Titus Braun, a professor in UC Davis. I attended his next generation sequencing workshop back in 2015. And Data Carpentry actually has a lot of educational free courses for you to learn data skills. And I also want to thank all the people who share their wisdom on the web. Thank you very much. And that concludes my presentation. So what questions do you have? I will.
First of all, thank you very much for your very informative speech. I have questions from our teams and from YouTube participants.
Okay.
And my first question is, how can we measure whether we have enough data sets for an ideal analysis where we can use computational biology mechanisms?
How to measure the size of the data sets?
How can we measure whether we have enough data sets for an ideal analysis where we can use computational biology mechanisms?
Okay, I think, thanks for the question. I think it really depends on which field exactly you are in. For example, like if you are in for sequencing data, And I would suggest you at least have, for example, for RNA sequencing experiment, you want to at least have duplicates. Better to have triplicates. I mean, the more the better. But I mean, of course, it's expensive to generate those data, but you need to have like enough samples to have statistical power. And you can always calculate the power of your experiments beforehand. So always talk to a bioinformatician or statistician before you carry out any experiments. So in order for your data to be usable.
Thank you very much for your response. Our question is from YouTube. There are many software available online or as a package for data analysis. What is the advantage of learning a programming language at this point?
So I guess you are referring to the GUI, like the click and point and click softwares. But as I mentioned, it's very nice. Some of the tools, they are very nice, but they're designed specifically for certain tasks, right? Of course, if it can meet your needs, that's fine. But if you learn how to use a programming language, it's much more flexible and much more powerful. You can do any type of analysis that you can, because the software may only has certain functions that you want, and it may take them a very long time to update the software for you to use the other function that you want to use. But if you learn computational biology, you know the programming languages, you can do the analysis anytime. I think that's an advantage.
Thank you for your answer. You can close your presentation, and actually you can see us and also screen if you don't see.
Okay, so let me. Am I still sharing my screen?
Yes, you are
okay.
No. Okay.
I just stopped.
So can you see us?
Yeah, I can see you.
Okay. So I move on with next question. There are now without coding programs coming for future statistics. Is this adequate and permanent to work with?
Oh, I think this is a similar question with the previous one, right? So yes. I would encourage you to actually learn the basics of stats, and sometimes they have like those black boxes, like those softwares you can click, do this stats test, do that stat test. But if you don't really understand it, you may use the wrong test. So I encourage actually you to learn stats, basic concepts, but also if you use a programming language, for example, R, it's really good for stats. So I think still like learning an R package is still R language is very valuable. Yeah.
Thank you for your response. Next question is, how soon can we evolve from a wet lab novel biologist to a computational biologist? Or do we really need to computational biologists for computational analysis?
Oh yeah, that's a good question. Like I was a pure actually wet lab bench scientist. It took me 10 years, but really after the first 1 or 2 years, now you get comfortable with the command line, the R stuff. You can already do quite a lot. But of course, it takes time to be really better, to be good, right? So as the time goes by, you accumulate the experience. I think time is just the currency you have to pay to gain experience. But I would say if you start now, the best time to start is like 10 years ago, right? But the second best time is now. If you start learning it now, I'm sure you'll benefit like sooner, right? So I think if you in my GitHub repository, there are some links for courses that you can take for free. Go there and commit yourself. Every day, do 1 hour, exercise, watch the videos. I'm sure you will get there. So that actually reminds me when I was studying. Yeah.
But wet lab is so time consuming. There is no time to. I'm also a wet lab scientist, but I don't have any time for studying computational things.
Yes, that's also a common actually So you have to actually better do a time management. So sometimes, for example, if you need to weight something, you're doing the centrifuging, you're doing Western block, you can always have those time to learn something. And what I did actually during my PhD is that after work, actually, I would actually watch those Coursera, edX courses at night, and learn the computation and do the exercise. Yeah.
So there is no excuse.
No excuse, yes.
Next question from YouTube. Scalable and reproducible scientific workflows like Nextflow are proliferating day by day. Can we say that computational science is stepping into a new era, or would you recommend sticking to the roots?
Okay, thanks. That's a very good question. Actually, I haven't touched that point. Like workflow language, for example, Nextflow, SnakeMake. I use SnakeMake a lot. Those workflow languages is really useful for pre-processing data. Those steps has like fixed step. For example, for RNA sequencing, align the FASTQ. I mean, you do quality control, then align FASTQ to band files. From the band file, you get this count matrix. Then you go to the D62 for differential expression. For example, those are fixed sets, right? You can easily reproduce, use this workflow language to actually encapsulate. But really, the downstream analysis require your domain knowledge, your biology knowledge. So that I don't think it can be replaced by any workflow language. You need to, if you're in a PhD program, you know that you always do this analysis. Oh, this is not working. Let me change the analysis. You use a different tool. It happens all the time. So that cannot be automated, and that's why we still have a job, because if that can be automated, I'm out of job. Yeah.
Thank you for your answer. Next question is from YouTube. Data sharing is a big problem. At this point, where can we obtain the data that we analyze to improve ourselves? Does repositories such as GitHub can enough?
Okay, so that two questions here. Actually, the GitHub actually is mainly the repository for code. And if you want to fetch some data, for example, for your own analysis, or maybe even just for practice, go to GEO, and then we'll also have ENA, those public available. So when you publish paper, you deposit your data there, right? So in my GitHub repository, there are some links there showing, okay, what are the available repositories for data repositories that you can fetch the data. For example, also TCGA, the Cancer Genome Atlas, it includes all the cancer data that you can use for your own project. Yeah, so you go to my GitHub link and you will find some of the links for data repository. Yeah.
Thank you for your answer. Next question. Despite the developments in computational sciences, it is necessary to verify the NGS data with wet lab methods. Should this dependency continue, or are computational methods alone sufficient now? Thanks.
No, I don't think computational methods alone is sufficient. So we always need to do wet lab experiments to verify the finding. But computational biology is very good when you have a large amount of data. You can quickly narrow down the targets that you want to focus on, right? You can quickly generate insights from a large amount of data. Maybe you find the target, then the wet lab scientists need to verify that.
Okay, thank you for your answer. I move on with next question. When I tried to use Dakika, it was challenging both in terms of computer hardware and usage. Are there any more user-friendly programs?
Okay, so I think Docker can be clunky sometimes. I understand it, because you have to actually encapsulate all the operating systems. It can be several gigabytes, can be really big. So I would say, you can use, so nowadays cloud computing is it's so easy, like AWS or Google Cloud. You can just increase the amount, disk size, the CPU, the memory, you can always increase. So if your desktop cannot handle Docker, maybe use those cloud computing facilities.
Next question is, if we think of data analysis as a language, would it be more effective to focus on the alphabet or words for learning?
Oh, that's a good question, actually. Yeah. So let me take R for an example, because I use R most of the time, right? So you can learn base R, which are the really basic functions, right? I will refer that as the alphabet. But you can also learn tidyverse. It's a group of packages, for data visualization, data analysis, data wrangling. And it's a higher level abstraction of those base R functions. So I would say if you want to do something quickly, you want to actually learn the tidyverse first, because it can quickly get you some results. But I would say understand the alphabet is also critical, right? Because you want to understand how that high-level function works eventually. And yeah, and there's actually some argument whether you want to learn base R first or tidyverse. But I would say if you really want to get the results fast, you can start with the words. And the other analogy I had is that I want to get from A to point B. I need to drive from A to B, but I don't need to understand how the engine works. I don't need to understand how each of those elements, they all work with each other, right? I just need to know how to drive.
Yeah. Thank you for your answer. Next question is again from YouTube. Tommy, thanks for the talk. Data cleaning part is an important step for analysis. Are you losing any crucial information during cleaning up process? Is there any information showing how to clean data?
Yeah, thanks for this great question. Yeah, data cleaning is an important step, but you are afraid of losing some crucial information, right? So that's why you always need to compare, actually. Before cleaning, after cleaning, you need to compare. Is there any leakage, any information lost during this step? And I will say you also need to actually compare the final results, for example. The data without cleaning, the data cleaned, and whether that results make sense or not. So every step, I would say now the domain knowledge actually really actually important, because okay, I clean the data, but now my results doesn't make any sense. Like whether I need to do that or not. So I need to go back to the original step. Okay, maybe let me relax the cleaning criteria. Okay, to see whether the results make sense. So you need to always talk to the expert in, if you're working in biology, talk to them, right?
Thank you for your response. Next question. Nowadays, data analyses are made with machine learning methods, especially for cancer-like heterogeneous disease.
Mm-hmm.
I would like to ask how I need to start to learn them with sufficient R or Python languages.
Okay, thanks. That's a good question. So machine learning is very hot, right? Everyone wants to learn it. So my talk actually didn't touch about machine learning that much, but I want you to start with basic data skills first. And to be honest, most of the biology questions, you do not need deep learning. You may not even need like machine learning. But for your next step, if you want to really learn, there are some areas machine learning is great. And if you want to learn, I would start with, for example, some YouTube channels. Like there's this Josh Stammer has this StatQuest. I'm not sure whether you know this. It's a really good YouTube channel explaining all those machine learning concepts. And there's several other actually courses actually on edX, Coursera that you can take free, and you can just go there and search machine learning in those websites, and you will find courses that you can take for free. So, and I would actually suggest you to use a real data sets, and maybe you can even just read a machine learning paper, like a biological paper, to see how they use machine learning to address that question, and you want to reproduce their finding. And I think that's the best way to learn machine learning and how to use it to address biological questions. So get your hands wet.
Thank you for your answer. Next question is, if a computational analysis in a published article, which is not reproducible, it is really reliable for the science?
No, if it's not reliable, it's not, it's not good for science. To be honest, especially if those papers published in those glamour journals like Nature. I think I saw a paper saying there is a high probability that those papers are wrong in those high-profile journals. So of course you want to see the data, you want to actually see the code, you want to reproduce some of the analysis.
Thank you for your answer. Next question from YouTube. What do you think about what should a computer science student should do to become a computational biologist?
So you're from the other side of the river.
Yes,
okay.
Other side.
So if you're a computer scientist, so you know programming language really well, but you don't know biology that well. So I would actually suggest you to start learning the basic of biology. There are some courses. You need to understand what's DNA, what's mRNA, what's the protein, how they, and if you want to analyze a type of sequencing data, you need to understand how the sequencing technology works, what this technology is measuring, right? So I would suggest you to understand some of the biology first, and the same advice for people from wet lab. Go find a paper, and download their data. Use this machine learning approach or computational biology approach to reproduce their results, and that will give you some sense how the field. How it's working in this field.
I think computer scientists have more advantages from biologists.
No, not necessarily. I think both have challenges, but I would say learning biology is also not easy. I would say if you're a computer scientist, because biology is just so weird. Like sometimes it doesn't make any logical sense. It's like why some RNA and protein are not correlated. Why?
Many of us know this.
Many of you know it, right? So okay, like for example, HIF, like the hypoxia-induced effect. mRNA is so high, but why? When there's oxygen, that the protein gets degraded. When you have no oxygen, now it's stabilized. There's so many of those things that biology can explain, but not necessarily the computer scientists. I don't know that. So
Thank you. Last question. Except from computational sites, what advice would you give to students studying in scientific fields?
Computer. What? So can you repeat the question again?
Except from computational things, what advice would you give to students studying in scientific fields?
I think
not related about your presentation.
Okay.
So for academic career.
No matter what you're learning, so it's all about learning, keep learning. So in this case, in this presentation, I show you we need to learn computational biology, learn programming. But if you in other fields, the same thing, you want to keep learning, because the day you stop learning, you start dying, because the knowledge are exploding. If you don't keep yourself updated, So my suggestion is keep learning. Different fields, no matter what kind of fields you are in.
Thank you for your advice, and also on behalf of myself and all our participants, I would like to thank you for spending your valuable time transferring your knowledge to us and for this very informative presentation.
Thank you very much for the invitation, and I hope like my presentation is educational and can help some of you to start your journey as a bioinformatician. Bye.
It was really necessary for us. I wish you healthy days.
Sure. Thank you very much.
Bye bye.
Bye.
At 2030, Dr. Pukibre will be with us to give a speech on in vitro modeling of human cortical assembly in health and disease. Thank you.
Bioinforange platformunun kuruluşunun 2. yıl dönümünde düzenlediğimiz Bioinfocongress II etkinliğimize hepiniz hoş geldiniz.
Merhabalar, hoş geldiniz laboratuvarımıza. Merhaba hocam, hoş geldiniz.
Merhaba, hoş bulduk. Sağ olun. Teşekkürler davetiniz için, organizasyon için.
Davet için tekrar çok teşekkürler. Organizasyon da çok başarılı gözüküyor. Çok teşekkür ederim.
Tekrar teşekkürler davetiniz için. Bugün bilgisayar teknolojisiyle biyomoleküler sistemlerin simülasyonu ve ilaç tasarımı konusunda konuşacağım.
Muhteşem. Çok teşekkür ederim. Evet, hepinize merhabalar. Organize komitesindeki arkadaşlara da çok teşekkür ederim beni davet ettiğiniz için.
Herkese merhabalar. İlk önce davetiniz için çok teşekkür ederim. Buradaki diğer konuşmacılarla beraber sizlerle beraber olmak gerçekten gurur verici.
Bioinfocongress'te sunum yapmaktan beni davet ettiğiniz için çok memnunum ve sanıyorum tamamen insan hücre fonksiyonlarına yönelik olmayan daha basit canlılarla ilgili olan tek konuşma belki. Ama böyle bir değişiklik getirmekten de çok memnunum.
Öncelikle hepinize teşekkür etmek istiyorum. Bioinfocongress oldukça güzel bir organizasyon yaptınız. Tüm Bioinforange çalışanlarına ya da gönüllülerine teşekkür ediyorum. Beni davet ettiğiniz, zamanınızı ayırdınız.
Merhaba, davetiniz için çok teşekkür ederim. Bu aktiviteyi organize ettiğiniz için de teşekkür ederim. Gerçekten çok büyük bir çalışma gerektiriyor.
Thank you, it's a pleasure. Thank you for the invitation.
İyi akşamlar. Başlamadan önce bu güzel organizasyonu düzenleyen arkadaşlara gerçekten teşekkür etmek istiyorum. Bu kadar büyük bir topluluğu bir araya getirmek, böyle bir organizasyon gerçekleştirmek gerçekten büyük bir başarı.
It's a really fantastic program that you've put together over these last few days. So it's been some brilliant science and some brilliant scientists speaking. So thank you very much again for the kind invitation.
Welcome everyone, I'm Neva Çalışkan. I'm a group leader at the Helmholtz Institute for RNA-based infection research. And today my talk will be about how genome, meaning of the genome can be altered by frameshift RNAs and how this response can be regulated by host and viral proteins.
Teşekkür ederim davetiniz için. Bugün mRNA aşılarından bahsedeceğim sizlere.
Merhaba arkadaşlar, bugün sizlere katılmaktan çok hoşnutum. İlk önce Bioinforange öğrencilerine bu güzel organizasyonu bir araya geldikleri için teşekkür ediyorum. Gerçekten benden önceki videoları izlemekten çok hoşlandım.
Merhaba herkese, buraya geldiğiniz için teşekkürler. Bugün ökaryotik genomların organizasyonu hakkında bir konuşma yapacağım.
Merhaba herkese. Katıldığınız için ve dinlediğiniz için teşekkürler.
Merhabalar, hoş bulduk. Başlamadan önce böyle güzel bir organizasyonda emeği geçen arkadaşlara teşekkür ediyorum. Ayrıca davetiniz için de teşekkürler.
It's a tremendous honor and pleasure for me to be here after starting my lab just over 2 months ago.
Gün boyunca 30 saat bir yayın süremiz oldu. 3 eğitim, 17 konuşma, toplamda 20 konuşmadan süren, görüyorsunuz yorgunluğumuzu da arka planda. Öncelikle bizlere konuşmalar, eğitimler veren 20 değerli hocamıza çok teşekkür ederiz. Açıkçası davet ettiğimiz hocalardan çok yüksek oranda kabuller aldığımız için bu kadar çok yoğun bir programımız oldu. O yüzden hocalarımıza çok teşekkür ederiz. Onların bize davetleri bu kadar çabuk bir şekilde, hızlı bir şekilde bizleri kabul etmesi, konuşmalar yapması bizler için çok güzel ve bizleri gerçekten çok motive ediyor. Hocalarımızın haricinde siz şu anda 277 kişi var bu saatte. Çok teşekkür ederiz. Değerli katılımcılarımıza çok teşekkür ediyoruz. Ancak büyük bir teşekkürü de ben buradaki ekip arkadaşlarıma yapmak istiyorum. Bioinforange'ın 2. yıl dönümü, doğum günü. Buradan Bioinforange'a emek veren, gönül veren, Bioinforange'ı seven tüm bilim severler, katkı sağlayanlar, öğrenciler, insanlar adına iyi ki doğdun Bioinforange diyorum. Bilim paylaştıkça güzel, bilim paylaştıkça ilerler. Bilimle kalın. Bioinforange platformunun bilim yolculuğunda 3.sünü düzenlediğimiz Bioinfocongress ilk etkinliğimize hepiniz hoş geldiniz.
Çok teşekkür ediyorum. Mehmet, senin şeyin nezdinde bütün organizasyonu yapan arkadaşları da tebrik ediyorum. Emeğinize sağlık. İyi bakın kendinize, görüşürüz.
Merhaba İvan Hocam, tekrardan hoş geldiniz.
Merhaba, teşekkürler davet için.
The first time we go very little time, transferring your knowledge to us and for this informative presentation. Kendim ve tüm katılımcılarımız adına çok teşekkür ediyorum hocam.
Ben teşekkür ederim.
Çok memnun oldum hocam. Sağlıklı günler diliyorum.
Sizlere de iyi akşamlar.
Yep, thank you guys so much. I really enjoyed it. Great questions from everyone.
Merhaba Tuğba Hocam, tekrardan hoş geldiniz.
Hoş bulduk. Çok teşekkür ediyorum davetiniz için. Harika bir organizasyondu. Herkese selamlar.
Değerli vaktinizi ayırdığınız, bilgilerinizi bize aktardığınız ve bilgi dolu bu konuşma için kendim ve tüm katılımcılarımız adına çok teşekkür ediyorum.
Ben teşekkür ederim davetiniz için.
Merhaba Nilüfer Hocam, tekrardan hoş geldiniz.
Merhaba, hoş buldum. Beni iyi duyup görebiliyor musunuz?
Evet, görüyorsunuz.
Çok teşekkür ederim davetiniz için. Çok öğrencilerle böyle bu sanal ortamda da olsa bir araya gelmekten.
Merhabalar. Öncelikle davetiniz için çok teşekkür ederim.
Teşekkür ederim hocam.
Çok güzel bir event ortaya koymuşsunuz. Bu kadar değerli bilim insanı arasında yer almak gurur verici. Teşekkür ederim.
Çok teşekkür ederim. Nazik davetiniz için çok mutlu oldum. Bu arada bu hani YouTube'da olan sayın seyirciler, bu saatte cumartesi akşamı gidip başka bir şey yapmayıp bu konuşmayı dinlemeye geldiğiniz için size de hürmetlerimi sunuyorum.
Çok teşekkür ediyoruz. Hem kendi adıma hem de bütün katılımcılarımız adına.
Rica ederim. Ben çok memnun oldum. Beni onure ettiniz. Çok teşekkür ederim.
Biz onur olduk hocam. Çok sağ olun.
Size çok teşekkür ediyorum. Katılımcılarımız ve kendim adına. Ben çok teşekkür ederim. Benim için de güzel bir deneyim oldu.
Bu harika sunum için katılımcılarımız ve bütün ekibimiz adına çok teşekkür ederim hocam.
Ben çok teşekkür ederim beni misafir ettiğiniz için.
Thank you. Thank you so much for this space. Thank you for having me here and for this opportunity.
Bilgi dolu konuşma için kendim ve tüm katılımcılarınız adına çok teşekkür ederim.
Ben teşekkür ederim davetiniz için.
Tekrardan çok teşekkür ediyoruz. Katıldığınız için çok mutluluk duyduk.
Ben çok teşekkür ederim. Çok güzel bir organizasyon gerçekleştirmişsiniz. Beni davet ettiğiniz için tekrar teşekkürler. İyi günler dilerim.
Biz teşekkür ederiz hocam. Bioinforange platformu olarak düzenlediğimiz Bioinfocongress 3 etkinliğimizde çalışmalarına yurt içinde ve yurt dışında devam eden değerli bilim insanları katılımıyla toplamda 15 seminer gerçekleştirmiştik. İzninizle burada organizasyonda emeği geçen arkadaşlarımı davet etmek istiyorum. Şimdi burada ekranda olan arkadaşlarımız haricinde kongremizin düzenlenmesinde organizasyonda emeği geçen tüm ekip arkadaşlarımıza teşekkür ediyoruz. Ve sizler gibi yani burada hepimiz öğrenciyiz biz ekip olarak. Özellikle öğrencilere bir katkımız oluyorsa akademik hayatı ve işte gelecek idealleri konusunda bir fikir verebiliyorsak buradaki konuşmalar, işte bizler bir katkı sunabiliyorsak açıkçası biz bundan çok mutluluk. Tekrardan etkinliklerimize devam edebilmeyi umuyorum. Bizi sosyal medya adreslerimizden takip edebilirsiniz. Sizlerin desteğiyle biz motivasyon buluyoruz. Bizler motive olunca da böyle güzel etkinlikleri sizlere sunmaya çalışıyoruz. O yüzden karşılıklı etkileşim ve destekleşmeyi umuyorum. Tekrardan hepinize çok teşekkür ediyorum. Katıldığınız için de çok teşekkürler tekrardan ve herkese bol başarılı akademik kariyerler, güzel bir hayat, sağlıklı bir hayat diliyorum ve sağlıklı günler diliyorum. Görüşmek dileğiyle tekrardan. Hoşçakalın.
Bioinfocongress IV. Assistant Professor Dr. Fikri Birey. In vitro modeling of human cortical assembly in health disease. Assistant Professor Dr. Fikri Birey, as a Fulbright scholar, earned his Bachelor of Biology degree in Biology and Philosophy from the University of Kansas in 2008. He earned his PhD in Genetics on neuroglial interactions in stress-related disorders at Stony Brook University in 2014. He completed his postdoctoral studies at Stanford University in Sergio Pasque's laboratory between 2015 and 2021. Furthermore, he developed the Assembloid platform, a stem cell-based developmental forebrain model, and used it to understand how neurodevelopmental disorders occur during human cortical development. In early 2022, Dr. Bray established his center in the Department of Human Genetics at Emory University, where he continues to produce and use stem cell-based models to understand the guiding principles of how the human brain assembles.
Dear Dr. Fikri Bey and participants, welcome all of you to our Bioinfocongress IV event. Dr. Fikri Bey will give us a talk today on in vitro modeling of human cortical assembly in health and disease. At the end of the presentation, we will have a question-answer section prepared by our team and consisting of questions from our participants. Dear Dr. Fikri Bey, welcome back.
Hello.
If you wish, you can start your presentation.
Sounds great. Thank you so much for that kind intro and inviting me. This is such a fascinating, well-organized initiative, and kind of astounded by the fact that it's all organized by you trainees, students. So kudos to all of you, and again, thank you so much for inviting me. To the end, I also want to thank the people that stuck around all day until this last talk of the day or the night, I guess. But I'll try to keep it short and short and sweet. But what I want to talk to you about today is about our efforts in developing in vitro stem cell based models of the human brain assembly and how we're using such models to better understand how brain disorders emerge during neurodevelopment. And broadly speaking, I'm interested in the question of how the human brain is constructed during fetal development. And just like many other species, the development of the human brain starts with the formation and closure of the human neural tube, shown here, which in humans happens around 4 weeks post-conception. But unlike many other species, the processes that are triggered afterwards are orchestrated and very tightly orchestrated over very long periods of time. And you can see here in the schematic that some of them really do take place well after birth. For example, things like neurogenesis and migration can take place largely in utero. Prenatally, things like myelination, synaptic pruning, etc., take place largely after birth. Really resulting in this immensely sophisticated and functionally compartmentalized piece of tissue. And this really is an astonishingly complex project. And despite it involving generation and coordination of a stunning number of cells and connections over months, it occurs with an incredible precision and accuracy from individual to individual, which really, I think, highlights how robust the assembly instructions that are encoded in the system are. One of my specific interests lies in the question of how a particular region of the human brain, the cerebral cortex, is assembled. The cerebral cortex arises from these forebrain structures during development, and in adult really performs as higher order cognitive functions. And very interestingly, is significantly expanded in volume in the human lineage compared to our primate ancestors. The period during which the first cortical circuits are assembled, which really spans mid to late gestational periods, is thought to be a very critical period during which these principles of mature circuit structure and function are established. And at the cellular level, the assembly of this early cortical circuits really involves two major neuronal subtypes: glutamatergic neurons, as shown in red here, and GABAergic neurons, GABAergic interneurons. And while the glutamatergic neurons are born locally in this dorsal side of the forebrain, which is the part that later gives rise to the actual cerebral cortex, the GABAergic interneurons, on the other hand, born a little bit remotely ventral to this dorsal forebrain side, in the ventral forebrain structures, and in the structures that are sort of transient in development. They're called ganglionic eminences. And one of them is shown here, the MG, the medial ganglionic eminence. So once these interneurons are born in these regions, they really have to migrate, especially in humans, really long distances towards the dorsal forebrain, where they meet their glutamatergic counterparts and they really start forming these early connections. And that's what I refer to as this early assembly of the cortical circuits. And really the genetic or environmental perturbations to any number of these processes that I just outlined are thought to be linked, very closely linked, to several neurodevelopmental and neuropsychiatric disorders, like autism, schizophrenia, epilepsies. Yet a mechanistic and dynamic understanding of how these processes really unfold in humans and go awry in patients remain virtually non-existent. And this is largely due to the fact that we really lack access to the functional brain tissues, at a level where we can really study these processes dynamically and mechanistically. And this really has been the primary roadblock that my work over the last several years has aimed at address through developing these personalized stem cell-based platforms that can relatively faithfully model these processes in a dish, in a cell culture dish, so that we can really study them directly in patients. But before I go into details about the model, I want to take a step back and really explain the core technology that has enabled us to develop these models in the first place. The starting material for these models that we have developed are called human-induced pluripotent stem cells, or what I'm going to be referring as HIPSCs. These are pluripotent stem cells, and what I mean by pluripotent stem cells is that these are cells that can really differentiate into any kind of body, any kind of cells found in your body. But in this case, they're induced from post-mitotic somatic cells, things like skin cells or blood cells. And this technology was first described by Shinya Yamanaka and colleagues over a decade ago, won a Nobel Prize, I guess, in 2008 or so, if I'm remembering correctly, and really unlocked this huge potential for personalized medicine, really enabling one to have access to any kind of cell type of interest directly from individuals. And I'm going to outline a typical workflow here, which starts by harvesting the somatic cells. These can be skin cells, blood cells, and then through the overexpression of what's called Yamanaka factors, which are these transcription factors that Shinya Yamanaka described, we're able to reprogram these cells so that they assume a pluripotent state. And at this stage, they really look and act like real stem cells. They form these colonies, and given the right cues, chemical cues, they can make cells that come from all three germ layers. And it's in that sense, they're very much like human embryonic stem cells, which are sort of naturally found in the inner cell mass of a blastocyst. And then these cells can actually be dissociated and then reaggregated into these three-dimensional spheroids. And using various patterning molecules, small molecules, growth factors, specific cocktails of these, they can be differentiated into region-specific brain organoids that sort of resemble specific parts of the developing brain. And I'm going to just give you an early example of this kind of work where this human-induced pluripotent stem cells, iPSCs, were aggregated in 3D and really pushed towards certain regional fates of the human brain. And this early work really focused on trying to model this part of the developing human brain, was the dorsal forebrain. As I mentioned to you, this is where the glutamatergic neurons really arise during development. And the work from my postdoctoral lab coined this term human cortical spheroids at the time. And this model really involved generating these 3D structures that were a few millimeters in size, kind of in terms of its size resembled an E12.5 mouse brain, for those of you that come from this kind of work. And when we mapped them transcriptionally to the primary data sets, primary tissue data sets, they really mapped onto the primary fetal cortex that were around mid to late gestational stages. So that was very encouraging at the time. And then we did a lot of characterization showing that they had these cellular features that was reminiscent of the human of the developing cortex, the ventricular zone like structures that had these progenitor pools. They had this sort of lamination like structure to them, where they're the deep layers and the superficial layers, just like how it is in the real cortex. And then they also at later stages went through gliogenic switches, where they would start making this non-neuronal subtypes like astrocytes. But this protocol really lacked a critical subtype. It was the cortical GABAergic interneurons. And this made sense that they were missing this subtype because the patterning strategy really targeted to generate the dorsal forebrain. And you might remember from a few slides back, the cortical interneurons are generated instead in these ventral regions, in the ventral forebrain regions. And then they have to migrate dorsally. So when we started thinking about how to build sort of a fuller model of the developing cortex that to incorporate these interneurons, the strategy we took was to separately generate the ventral forebrain spheroids, which we called human subpallispheroids. Subpallium is another name for the ventral forebrain, and sort of assemble these subpallispheroids with the cortical spheroids so that we can start modeling some of these processes like interneural migration and integration onto the excitatory circuits. And we ended up calling these models forebrain assembloids. And we described the first assembloid model in 2017. And after that, several novel assembloid models really followed, modeling various interregional interactions that arise during human brain development. And I'm highlighting a few of them here. And actually going to highlight the latest one. This is a preprint that came out a day or two ago, where we built an assembloid model of the thalamocortical connectivity. This is a hugely essential and fundamental interregional interaction that really drive this early corticogenesis and sort of like this cell extrinsic aspects that depend on the early presensory activity that comes from the thalamus. So this is a very exciting model that we had worked on for a while and just came out. But that really, this is just one example out of many that is out there. So this is like a hugely versatile toolkit now that we have to decipher these principles of human brain assembly. And if you ask me what really makes these models stand out as these robust modeling tools, and likely why we have so many of them out there that came out over the last few years, I would list probably four things. That one, that they're modular, that you can make these region-specific organoids separately, and then assemble them at will, giving you really that tight control over when to introduce that connectivity between regions. They're very tractable, unlike an embryonic mouse brain, for example. They're in a dish, and you can really at ease do these longitudinal studies where you can really follow any number of processes over time within a single assembloid, right? So that's a huge advantage. They're tunable. Just because they're modular, we're able to sort of introduce various tools pre-assembly to a given region-specific spheroid. And for example, we're able to introduce an opsin only, let's say, in the subpallial derived interneurons, and then perform the assembly so that you sort of limit the expression of a certain viral vector to a particular region without having to go through like complicated infection schemes or anything like that. So it makes it sort of a tunable model when you think about this aspect. And as I'm going to be showing to you and spending the end part of my talk a lot on this, that they're very amenable to disease modeling. And I particularly worked on neurodevelopmental disorders of monogenic origin, and they're extremely robust models when it comes to understanding those kinds of diseases. But then now people are sort of starting to study neurodegeneration using these other kinds of assembloid models, for example, corticospinal muscle assembloids. But for the rest of my talk, what I would like to do is to take you back and walk you through the steps of how we built the first assembloid model, the forebrain assembloids, and basically going from the parts of the assembly and how we validated it using primary fetal tissue, which is a hugely important part of any in vitro model. And as I said, just at the end, going a little bit deeper into how we applied this model to really better understand and cover novel insights about a particular neurodevelopmental disorders called Timothy syndrome using patient-derived assembloids. All right. So the first question, as I alluded to, was how do we go about developing the subpallial spheroids? I'm going to be referring to subpallial spheroids as HSS and cortical spheroids HCS. And for that, we started by running a small molecule screen. We took the HCS recipe as our base recipe, and then we basically asked the questions: what are some of the compounds and small molecules that we can use to modulate signaling pathways in a way that we go from the dorsal forebrain to ventral forebrain identity? And we, of course, looked at classical developmental studies that really teased out the interactions between these signaling pathways that really drive specifications of those regions. And we basically focused on the sonic hedgehog signaling, which is very enriched in the ventral side, and the wind signaling, which is sort of enriched at the dorsal sides. So the strategy was very simple. We said, okay, why don't we then block the wind signaling, which gives rise to dorsal identities, and enhance the sonic hedgehog signaling, which is sort of enriched in these ventral regions. And that turned out to be good enough of a strategy. So when we started looking, we saw these progenitor pools that were related to ventral progenitors. And then when we waited a little bit longer, we were able to see these GABAergic interneurons arising. And we wanted to get a full picture of the cellular diversity. And as everybody under the sun does, we did single-cell RNA-seq. And we basically were pleasantly surprised to see initially that our HCS and HSS recipes really segregated. They were the primary clusters that segregated. And within those, we were again happy to see that each of these had their subtypes that we expected to see. And if you focus on the subpallium, for example, you'll see that there's the GABAergic neurons and the ventral progenitors that give rise to these neurons, as well as some oligodendrocyte progenitors and astroglia as well. And I'm skipping a lot of the characterization here, but once we were convinced that we had a good subpallial spheroid recipe, we went on to assemble these together. And the assembly is quite simple. We just sort of restrict them in space, and then they fuse after a few days. And then we started tracking these intraneurons. That was the first thing we started looking at, the migration of intraneurons from the subpallial to the cortical side. And then we use this virus that is specifically reported that is specifically expressed in intraneurons, DLX12B, GFP vector here. And then in green in GFP, you can see the intraneurons sort of slowly moving in. And after about 24 days, there's like heavy infiltration. And if you sort of 3D construct an assembloid 30 days after fusion, you can see this heavy infiltration of intraneurons. And I just want to put a side note here and explain the way that these intraneurons move, which is quite stereotypical, that has been described in mouse in great detail. where based on the directional cues that they receive from the environment, this leading branch stabilizes towards the direction of the forward movement. And there's very interesting this cytoskeletal rearrangements and crosstalks that are triggered, creating a pulling force by the leading branch and a pushing force by the cell rear. And this whole coordination and then this saltatory mode, so it basically looks like a cell jumping, and this mode of movement is called nucleokinesis. And it's very stereotypical. You recognize it when you see it. So when we started watching these things live, we were happy to see that this is indeed the way that they were moving. And if you take a I'll give it a second for you to sort of appreciate this. But then I talked about validation being an important step. We really wanted to see if we can compare the kinetics of the movement in the assembloids to the kinetics of the movement from the primary tissue. And for that, we acquired there was some primary fetal cortical sections. We infected with the same reporter, and then we did a very similar kind of live imaging. And then you can see that this is very similar to what we saw in the assembloids. Again, they're moving place to place, spot to spot using this nucleokinetic events. And if you're not convinced, here's a side-by-side comparison. On the left is an assembloid interneuron moving, and on the right is a real, quote-unquote, human cortical interneuron moving. So at that point, we were like, okay, we're able to model this migration phenotype quite well, but are they really sort of following their developmental trajectory, and after migration, stopping, branching out, and really integrating with their dorsal forebrain counterparts, which are the glutamatergic neurons? So to really address these sets of questions, we first started by looking at morphology. And it's sort of a classical thing to look at if you're looking at the maturation of interneurons, that they go from this bipolar morphology to a more complex dendritic branching. And this is what we quantified first, and there was indeed a significant increase in the complexity, branch complexity of the interneurons that had migrated and then stopped. And then we asked the really critical question of whether these interneurons are synaptically integrating to these into these excitatory networks. And then for that, we perform this wholesale patch clamping experiments on interneurons in 3 different experimental groups. If you look at the little schemes under the bar plots, these are the groups either in subpallial spheroids, interneurons in subpallial spheroids unfused. So this is our unfused control. Interneurons unfused in an assembloid. This is our assembly negative control. And then the actual experimental group is the interneurons that had migrated over. And what we saw in the first bar plot here, you can see that only the cells that have moved over and stopped and integrated are really receiving this excitatory postsynaptic currents from the glutamatergic network around them. So that was really key for us to be able to see that. But then our collaborator did the reverse experiment and asked, okay, are the excitatory glutamatergic cells receiving inhibitory, the GABAergic input? And that wasn't the case. When he patched unfused glutamatergic cells from an HCS, he didn't see any inhibitory this time, inhibitory postsynaptic currents. But then he saw a sizable percentage of the excitatory neurons in an assembloid receiving this inhibitory postsynaptic currents. So there was this in sort of this bidirectional crosstalk synaptically that we observed that told us that, okay, these are probably really sort of integrating and synaptically connecting, which is sort of what you would expect as the next step in their developmental journey. And then at that point on, interneurons really go through this activity-dependent activity-dependent maturation and subtype specification, a lot of interesting things that they do once they start receiving this excitatory input. And one of the questions we asked is whether they are more mature, and as a proxy to that, we looked at the action potential generation. And I mean, lo and behold, that's what we saw, that the cells that actually migrated over and integrated, they're firing twice as many action potentials as their non-migrated counterparts. Okay, so at this point, I'm skipping a lot of the control experiments and some of the other experiments, but after seeing like the results of these key experiments, we thought, okay, we do have a model that we could model various aspects of this human cortical development with, and this is the migration, maturation, network integration. And we thought, okay, maybe we can now use this model really to uncover some new insights about disease, and really sort of fulfill the promise of a model like this. And we sort of got really interested in this particular syndrome when I was in the Pascal lab. Is this Timothy syndrome? So I'll give you a brief overview of what the syndrome really is. This is a rare, highly penetrant neurodevelopmental disorder, and penetrant meaning once you have the mutation, the percentage of the patients individuals that have the mutation, an overwhelming majority of them will actually present with clinical symptoms. That's what highly penetrant means here. And it's very rare. It's a very deleterious sort of mutation, so it's not found all over the place, but it's a very sort of rare, very sort of severe neurodevelopmental disorder that causes various developmental defects, cardiac defects. But sort of molecularly speaking, it's caused by this gain of function mutation in the gene called CANC1C, which encodes for particular subunit of a calcium channel. And what it does once you have this mutation, it delays the inactivation of this calcium channel. And this calcium channel is a voltage-gated channel. So it means that the cell depolarizes by incoming activity, it opens, but calcium levels are very tightly regulated in the cells. So it needs to really shut down really quickly so that it can buffer the incoming influx of calcium in healthy cells. Once you have this mutation, it takes the channel really long to really close, and we can really demonstrate this core phenotype using two different strategies. We can do calcium current recordings, and then you can see on the left side that in cyan, the delayed inactivation phenotype is very obvious. That this delay, once we're recording the calcium currents, you can see that delay in that closing, and then we can also look at that by depolarizing the cells using high potassium chloride and doing calcium imaging with that. So we thought this is a good candidate for this kind of model because calcium, among many things that it does, it's been shown to be important for motility, axon pathfinding, these early developmental things that really is related to cells finding their way in the developing brain. So basically, we had human iPSCs from 3 different Timothy syndrome subjects, and along with 8 control subjects, we generated this set of assembloids, and looked at how these cells were moving basically. And initially, what we noticed that the Timothy syndrome intraneurons were moving very inefficiently. And when we try to tease apart what this phenotype might be, sort of in terms of its components, we noticed that the Timothy syndrome intraneurons were moving more, they were jumping more often, but the length of their jumps were smaller. And one thing that is sort of cool and is related to one of the advantages that I listed, the modularity, one thing that you can do, you can mix and match genotypes. So you can put, in this case, TS subpallium with a control, a TS HSS with a control HCS. And what that enables you to do is ask this cell autonomous or regional autonomous versus non-autonomous question. And in this case, what we show that even though we had a control cortex, having a TS HSS was enough for us to see the phenotype. And then we sort of thought maybe we could pharmacologically rescue this phenotype. And for that, we used a drug called nimodipine, which blocks the L-type, this kinds of calcium channels. And the rationale was if the delayed inactivation is letting a lot of calcium in, maybe by blocking a little bit this channel, we can ease the burden on the cells coming from that extra calcium. And that was a very robust rescue for a part of the phenotype. So the saltation length phenotype was really robustly rescued, but astonishingly, it didn't do anything to the saltation frequency phenotype. So this was such a puzzling thing for me that it made, basically I spent the second half of my postdoc trying to figure out why this is the case. And it really took us down a rabbit hole where we ended up really teasing apart the molecular drivers of these two components of this migration phenotype. And I want to spend some time going through the details of that. And initially, when we were thinking how to go about really understanding what is going on, we thought, okay, one thing we could do is really watch these cells closely, really look sort of phenomenologically at the movements, at the morphological changes as they were migrating, and to be able to really track subcellular compartments. We use this algorithm called deep lab cut that people use for actually tracking, markerless tracking of animal body parts in behavioral studies. But we sort of adapted it for interneuron live imaging, and we were able to reliably track different sections of a migrating interneuron, and just focusing on the front and the rear of the soma, which are as a readout of the nucleokinesis event itself, and as a readout of the pushing and the pulling forces, we were able to identify this very interesting phenotype that although in controls, the front and the rear move together, in the Thymicis syndrome, the front moved first, and then minutes after, in some cells even, the rear followed. So almost there was a uncoupling of the coordination between the pushing and the pulling forces, and then we quantified that by the correlation coefficient between the rear and the front movement. And once we knew that there was a cell contractility phenotype, we immediately honed in on this signaling pathway, which is the actomyosin contractility pathway. This is the pathway that is involved in making your heart contract, and it is heavily calcium dependent. So it really made sense for us that this pathway was probably overwhelmed in TS by this extra calcium that was being brought into the cells due to the mutation in the channel. So we did a very similar thing in terms of sort of pharmacological rescue of this phenotype, and then we thought, okay, what if we block some of this pathway again that is phosphorylated through calcium, and if we sort of block the kinase that does the phosphorylation, maybe again we ease the burden on this pathway a little bit. And again, this worked really well in the saltation length. Again, it did nothing with the saltation frequency. And this is kind of like a gnawing puzzle for us. It's just like, why? Okay, we sort of figured out the contractility deficit is completely separated from overall motility of the cells. And basically, to be able to figure out what is going on, we went back and did an unbiased RNA sequencing experiment from various different time points, from the HSS, from different patients, controls. And I'll cut a long story short here and just put a one plot to really summarize the relevant finding. Was that we in this age where we're doing a lot of our migration assays, we saw the top two terms that were so upregulated in Timothy syndrome HSS were the regulation of membrane potential and the synaptic transmission, GABAergic synaptic transmission. And here I'm going to pull up some of the subunits that are upregulated in TS that are sort of contributing to this term. So that was interesting. This is the GABA receptors expressed by a cell type that is actually synthesizing GABA itself. So this pointed to a potential autocrine signaling that might be that might sort of potentially be related to this saltation frequency phenotype through the action of the GABA, which was shown to be pro-motility before in other various studies. But before that, we really wanted to sort of functionally validate some of these findings that we saw in RNA-seq. And for that, we went and actually in migrating interneurons, put in a patch clamping pipette and really wanted to record this the membrane potential in actively migrating interneurons. And we really did see a robust depolarized resting membrane potential phenotype in the interneurons. And then that sort of like made us ask whether, this maybe is making the cells a little bit more prone to be active. And then to assay that with the calcium imaging in interneurons, TS versus control. And we indeed see that Timothy syndrome interneurons were sort of had an increased number of calcium transients. So this was very interesting, but then we were scratching our head about what might be really the reason why we're getting this increased this hyperactivity. But then it occurred to us that it really could be GABA itself and nothing else, because GABA is depolarizing. As you might know GABA as an inhibitory signal, but GABA is really during development due to the fact that these two chloride transporters are expressed relative to each other. It makes the relative expression of these transporters makes GABA excitatory mostly during development, and that was also the case for our interneurons when we did calcium imaging in these interneurons and added some GABA. We really saw a response. But to really sort of pinpoint the fact that yes, adding GABA in these interneurons does create an inward current, but not only that, the TS interneurons are more sensitive to such a GABA treatment. We did this thing called the GABA puffing coupled with GABA current measurements. And to cut a long story short, in Timothy syndrome interneurons, we really indeed saw this enriched inward GABA current as a response to GABA puffing. This sort of like tied in the loop for us a little bit that, so there's probably this GABAergic signaling deficit due to the fact that there's GABA receptor upregulation in these cells. So once they're secreting GABA, just as they do, as they're migrating, it is going back and really signaling through these receptors that are upregulated in the cells. And whether this could be really the reason why these cells are sort of like higher in their motility as sort of assayed by the saltation frequency. And when we use a similar pharmacological technique where we sort of antagonize some of these GABA receptors, and then we saw that we're indeed getting this rescue of the saltation frequency, but this time nothing was happening with the saltation length. So, I went through this sort of detailed molecular mechanisms related to this interneural migration phenotype, and I'll just summarize what we found here. Was that in interneurons that are migrating, there are two independent pathways that are dysregulated that we think really drive different aspects of this migration phenotype. One is the actomyosin complex that we think are more acutely dysfunctional, and then we have this autocrine dysfunction that is more indirectly dysfunctional because we think the GABA upregulation of the GABA receptors really is the result of this activity-dependent pathways that are being sort of hyper-driven by this incoming calcium influx in Timothy syndrome. And I just want to, wrap up this story here real quick, and just to point out that, I feel like this whole story, not only points to the fact that these L-type calcium channels are really central to many aspects of cortical assembly, but I really want to underscore the nuance and the breadth of new biological insights one can gather about disease using this patient-derived assembloid models. If one were to just do a simple migration assay, for example, maybe you might be able to get an elementary level insights into what might be going on, but then this model really allows you to really tease apart at that molecular level what the drivers might be, and that could be really essential if you really want to target very specific pathways towards therapies that, for example, this sort of underscores how important GABA signaling is pre-circuit assembly, and if you want to sort of avoid this actomyosin pathway towards restoration of the migration in these patients, you might instead go for the GABA signaling modulators instead of touching the actomyosin signaling, which is again important for the heart. And I'm not gonna have time to really go into the details of some of the follow-up studies that we're doing. There's a very exciting study now that, based on the insights that we've gathered, we're moving towards more of a therapeutic study where we are thinking of ways of genetically modulating this channel in an in vivo setting, whether that could sort of be more sort of a clinically relevant going forward for this particular disease. But in my lab, what we're really interested in is really understanding what the molecular drivers are in the spirit of what I described in Timothy syndrome in the second part. We're sort of moving towards this idea that what are the molecular drivers that really drive this assembly of the human cortex, and how can we study these using these assembloid models? And I'll just highlight a few directions now that we're actively pursuing. Again, we're really interested in activity and how it really drives this cell extrinsic mechanisms. We are fascinated by interneurons. If it has not been clear so far, we are really thinking a lot about their developmental trajectories and how the many interneuron subtypes now that have been described, how these diversifications come to be, and what role activity, this presensory activity, plays in their development. One thing I didn't talk about was isoform shifting. The CACNA1C, the gene that causes the TS, once mutated, goes through these really interesting isoform changes in development. And there's a lot of genes that really do this alternative splicing dynamics. That is a completely black box, and we feel like that our assembloid models are really sort of uniquely well-suited models to be able to study these kinds of dynamic, temporally resolved dynamics that like the shifting isoform landscapes. And we're developing a lot of new assembloid models to study various different interregional interactions. One of them is one thing that I'm really interested in this early neuromodulatory inputs to the cortex, like, for example, serotonin, norepinephrine. These neuromodulators come in very early development but not much is known about what they really do. And given the nature and the advantages of the models like these, we feel like we can build these models where we have a region-specific an organoid where the serotonin neurons are born, and then we have this, at the cortical, and then we can really control when the serotonergic input arrives, and then we can sort of modulate that input. And we're really interested in that world of neuromodulation and its effects in early development. We're developing tools to study, again, how this early, in the spirit of looking at specification and regionalization, we're building tools and implementing tools to really understand how regional patterning happens during human brain morphogenesis. This is sort of like taking a step back and moving away from the cortex. So we're building a lot of CRISPR-related tools, implementing these tools, and what I'm showing you here is a particularly interesting one where we're hoping to record temporally resolved dynamics of signaling as the cells are maturing and acquiring certain fates. So we're very excited about this particularly new direction in the lab. And again, all of this in service to understanding disease better. And one particular direction now that we're heading towards is this idea of convergence, and really trying to understand how genes that are of various functions, for example, chromatin modelers, ion channels, transcription factors, how all of these genes can really converge to confer risk for various disorders like autism, schizophrenia, etc. And then on the flip side, we're also interested in this question of how can a particular gene, which is what I'm showing here, is a TCF4, depending on the mutations that it accrues, depending on where those mutations land, can really give rise to wildly different disorders. And with that, I'm going to close. Thank you so much for listening, and I would like to thank the former and current members of the Pascal Lab, where I completed most of this work. My lab opened at Emory in early 2022, and we're finally up and running. This is from a picture from late last year. And one last thing that I'm gonna highlight is the Brain Organoid Hub. Please visit this website that will tell you more about this initiative that we're building at Emory, that we sort of put our minds and hands together. My lab, the Anderson Lab and Sloan Lab, which are both organoid experts, and we're sort of building this initiative to standardize and optimize these organoid models. And if you're at all in this space and you have like these burning questions in your mind, we're running experiments, we're forming collaborations, and we're sort of sharing our data with the field in the hopes that we get to a place where this is a widely accepted model system. So please do visit our website if you're interested. And I'll close with saying that we're recruiting at all levels, and if any of the things that I mentioned to you really grabs your interest, please do reach out, and you can find more information on my lab website. I'll end with that. Thank you so much, and happy to take any questions.
Thank you very much for your presentation, Dr. Fikri Bey. If you wish, we can go to the question and answer section. I'm starting with the first question. Could assembling brain organoids that resemble different areas into assembloids be used to model aspects of the interactions that occur between regions in the human brain?
Assembling that resemble be used to model. Yes, I mean that is our ultimate goal, to be able to model exactly this, the interactions that arise So we are sort of limited at the moment to these interactions that arise during development. The more sort of nuanced and complicated interactions that the circuits have during the adult, sort of like this postnatal to adult development, the end stages of development, I would say we're not there yet, but we are getting more and more proficient in really building whatever interregional interaction that you might be interested in, and really sort of honing in on like different region, different thalamus and this and the striatum, basal ganglia, and really building these region-specific organoids. We're really getting good at that. So and then you're just left with assembling them, and now we have a lot of tools in our toolkit that we can use to molecularly, cellularly, functionally really probe what's going on.
Thank you for your answer. I continue with the next question. This comes from YouTube. Neural organoids has lack of circuit connectivity that exists in vivo. So how can we build wiring?
Yes, that's a good point. As I said, we are sort of limited, and the way that I refer to wiring is very rudimentary. So it's this early connectivity that involves this level of synaptic integration, but there's also gap junctions, there's extra synaptic communications, and there's no spatial organization to how these things are connected, right? So if you compare it to an adult cortex that is sort of heavily integrated, well, both locally and remotely from the periphery, it looks nothing like that, right? The connectivity that we get. And people have sort of been thinking about this problem, and one solution that is sort of some people are moving towards is transplantation studies, where you can take these organoids and as a hybrid model, put them in a mouse, developing a mouse brain, and they sort of thrive and survive, and they really get integrated into the mouse circuitry. So that's as close as we have been able to get to be able to model real sort of a human-like circuitry, I would say.
Thank you for your answer. Next question is, what are the temporal, spatial, and cross-species limitations that preclude the formation of human neural circuits with high fidelity even after transplantation?
Yeah, that's a great question.
Human cortical organoids at early stage in development.
I mean, this is one that I would have to, because there are certain things, there are certain features that are unraveled post-transplantation. A striking one is how mature these neurons get, and morphologically, electrophysiologically. I think there's still roadblocks when it comes to specification. When we look at cell type specification and how granular, how specified, let's say, interneurons might get, or cell states even, like the microglia and astrocytes are very context dependent when it comes to their cell states. And whether we can faithfully recapitulate that remains to be seen. And yes, the question of species dependency is a good and complicated one. And it's I would venture to say that there are certain things that will probably be out of our reach for a long time, and we probably won't be able to even get there ever. And this in vivo hybrids is putting it in a sort of like another mammalian species probably is as close as we're gonna get in terms of introducing an in vivo environment. to these things? These are fantastic questions, and I'm sorry I don't have a more convincing answer.
Thank you for your answer. I'm moving with the next question. Are standardized electrophysiological assays sufficient to improve the reproducibility of electrical recording of neural networks in cortical organoids and assembloids?
I'll just read the question 1 second. Yes. Yeah. We have been doing now standard classic patch clamping experiments for a while now, and they've been sort of our go-to when it comes to this physiological characterization of these neurons. They're sufficient to improve reproducibility on neural networks. I'm not sure if I understand the question of reproducibility. There is a level of variability when it comes to maturation states within a given organoid. I mean, if you patch 10 neurons there, you're going to see a gradient of maturation states. And we sometimes use viral vectors to really hone in on a particularly mature population, for example. So we have tools that really can help us with this kind of variability. And then we have tools that we assay networks with, like multi-electrode arrays, extracellular recording tools that maybe don't really have that cellular resolution, but you have the network level resolution. And there's a level of variability there, but if there's a robust phenotype, let's say when you're disease modeling, you're usually able to see a sort of reproducible enough of a phenotype that if your numbers are high enough, then statistically speaking, you'll be able to find your phenotype. But I think this question of reproducibility is a larger one. It goes beyond the question of electrophysiology, and it's something that the field has been sort of struggling with, for lack of a better term. And it is at the moment our best tool toward combating this variability. It has been numbers. So just like a lot of differentiations, a lot of lines, compared to a mouse study, maybe it's like tenfold increase in the number of lines that we use, or like number of times that we try and experiment. But one thing to also keep in mind when it comes to variability, reproducibility, that there's real biological information in variability, right? So some level of variability that we see inherently has that biological information that is relevant, and it interacts with the genomic background. So it's a nuanced discussion whether variability that you see is coming from a technical or versus biological sources, and it's something good to keep in mind.
Thank you for your informative answer. The next question comes from YouTube. Can bioprinting applications offer a solution to the optimization need of 3D multicell systems?
Mm-hmm. Absolutely, yeah. So bioprinting, we're thinking about bioprinting in a number of different ways, but I'll just highlight a couple of them. So one issue that I sort of like alluded to was this lack of organization, and bioprinting approaches that we're now thinking about is related to introducing cell populations that secrete this what's so-called morphogens, secrete this growth factors or patterning molecules that activate or antagonize signaling pathways towards inducing a certain regional fate. And one of the ideas that we're sort of thinking about more and more is whether we can use bioprinting to really spatially restrict where we put these cells, so that instead of like bath applying some of these molecules, which is how we usually do our patterning, we can instead introduce cells that secrete these molecules in a sort of spatially restricted manner, so that our organoids, every time receiving those signals from very defined spots in space, and then whether that can be used to sort of control the cytoarchitectures spatially a little bit. Others have thought about using bioprinting when it comes, about it's more about building scaffolds and putting an organoid and then building a scaffold and bioprinting, for example, other cell types that you really don't get in organoids, like vasculature, microglia, things like that. So I would say it's a thriving sub-branch of our broader field, this bioengineering approaches related to bioprinting.
Thank you for your answer. The next question it comes from YouTube again. Could the assembloids of the future even bring together organoids from different systems, allowing the study of multisystemic diseases in which the immune system comes into contact with the nervous system?
Yeah, that's a fantastic question, and the answer is yes. If I'm understanding the question correctly, this is the peripheral assembloids that where's the cardiac, where's the gut, that you can sort of introduce to this neural organoids. And there's definitely a number of labs that are very interested in this kinds of questions. When it comes to neuroimmune interactions, I think a lot of labs interested in introducing microglia ectopically, sort of generating this. It comes from a different germ layer, so it has to be generated separately, separate from the corticals or brain organoids, and then they're basically transplanted onto the organoid. So people have published and been thinking about these kinds of approaches a lot, and it's not something that we do in the lab, but there's definitely a lot of potential. I would say it's sort of more of a dormant part of the field. I think it will definitely in future be a staple. I feel like of what we do, this source of assemblies.
Thank you for your answer. I'm moving with the next question. What are the gold standard and popular practices in designing experiments with assembloids?
Yeah, that was also another great question. So these standards sort of differ from lab to lab. Just because I come from a disease modeling background, we do have to sort of put in the numbers to be able to say, that there's a meaningful difference between patients and healthy individuals. And this just as a standard practice, we've been sort of reporting in our supplemental tables precisely how many organoids from how many differentiations, how many iPSC lines, and this, at least that it is out there. There is a certain level of variability when it comes to assay to assay. Again, if you're doing sort of electrophysiology, which is relatively low throughput, you're sort of limited in your numbers. But when we're doing calcium imaging or so that migration imaging is it's a higher throughput assay. So there are practical considerations, but I'm differing from like giving real numbers here, because it will again depends on power analysis and this and that. But you run a little pilot and then you see the robustness of your phenotype, and then you run your power analysis to tell you exactly the sample numbers that you would need to have. I mean, all I can tell you that it does require multiple lines, multiple differentiations, and from each differentiation, multiple organoids or assembloids. But if you can always go back to our papers and see exactly what kinds of numbers I'm talking about.
Thank you for your answer. I'm moving with the next question. This one comes from the YouTube. Recently, lab-grown clumps of human brain cells were transplanted into the brains of newborn rats. Is there any ethical issue for this application, such as humanized animals?
Yeah, I think this is a good question. There's definitely a very, that it is, yeah. So I'm sort of hung up on the word humanize because this is the word. It sort of depends on where you draw the line. Is it in putting one cell of a human neuron into the cortex is humanizing, and if that's also that's not humanizing. Why putting an organoid humanizing? And if that isn't the humanizing, what is it actually doing? That it's humanizing the animal. Is it making it smarter? Is it making it suffer in a way that a human might suffer because of the increased cognitive capacity? And that's why you're running into an ethical minefield. So I think there are rules and regulations in place that prevent anybody from doing very early transplantations, and that's just a legal requirement that you just have to abide by, and you cannot transplant these things. I don't remember the exact numbers now, at this very early stages where you might say, okay, this is a humanized rat, where like 70-80% of the brain tissue is compromised from human cells. But beyond that, it is a little bit difficult to draw the line and to really know what we mean when we say humanize. And I think this is ultimately a ball of cells still, right? So they are, people have shown that they are integrating into the rat, and the rats are not suffering. So I feel like that is the sort of the end goal of any ethical discussion related to organoids, which is related to any kind of mouse work or rodent work. That is the animal suffering or not? And we have very clear indications, right? Like when an animal suffers, and then we can sort of like work around that. But again, sort of like a non-answer I gave. But yeah, that also there's a lot of people that are more qualified in this than I am that can give a better answer.
Thank you for your answer. Another question is, do other neurotransmitters secreted by interneurons also play a role in TS migration phenotype?
Yeah, excellent question. This is not something we looked at. It's but yeah, interneurons do secrete other things. There's other sort of upregulated pathways that I have not shown you that could contribute to the TS phenotype. We sort of like picked the top picks, and then when we saw that we were able to get good rescue, that is sort of where we left the study. But that's a completely reasonable question to ask, whether there are other pathways. And honestly, I all I could say is that we have not looked into that. But excellent question.
Thank you for your answer. The next question is, will TS defect lead to changes in network integration and maturation of cortical interneurons?
Yeah. I did not, again, a very good question. I did not have time to show you, but we did look at network changes in assembloids. And what we saw was that it's a bit of a complicated question, because the network dynamics prior to the arrival of interneurons are already different just within the glutamatergic population. And then when interneurons come in, being hyperactive and GABA being an excitatory signal, it really drives that hyperactivity even more. So you get this really intense, this global hyperactivity, waves of activity that you don't really see in the controls. and a subset of these patients have epilepsy, and we always get asked whether that's sort of related to any kind of seizure-like activity that you're modeling. It's hard to say, but we do see network level changes, definitely. When we looked at the stages that we looked, we did not see any major maturation defects, but we have unpublished results showing that if we sort of let these interneurons really mature longer than what we reported, we do see differences in maturation, that they are sort of lagging behind a little bit in their maturation state, electrophysiologically speaking. So there are, but the main question for us is that how much of this later integration maturation related phenotypes, how much of those phenotypes are really connected to the migration phenotype itself? And that's a harder question to answer. But yeah, so there are phenotypes all throughout the sort of like this in vitro cortical development.
Thank you so much for informative answers. Lastly, I want to ask that what advice would you give to the students studying in the scientific fields?
I would say read. This is a fairly new field of study. It's a really sort of hot topic to, people that have not been organized scientists are getting into the field. And so there's an explosion of studies, but nevertheless, it's a relatively young field. And if you put in the time, it's surprising how fast you can catch up with the state of the art. So that is a sort of, practically speaking, an advantage for any of you who might be sort of interested in this kind of work, that I would just go and read and try to sort of grab as much of what's going on as you can. And I would suggest reaching out. That's really, I feel like, when I was applying, when I was trying to, I was born in Cyprus, and then I left Cyprus 2004-ish, and then before that I was trying to find my way, like how can I get connected with people so that I really find out what I really like to do. And people told me then, and people have been telling me all throughout my training over decades now, that you just have to get yourself out there and really just reach out to people. And network is such a critical, essential part of this. But you first need to find your passion. Like you need to find, whether it's this or AI or regeneration or anything else. And I think exposing yourself, finding that thing that really keeps bugging you and exciting you, and you keep going back to that thing. And once you find that thing, really like chasing the people, and people, I think, connect to that. Like if you're really passionate about something and then you reach out to them, they're also passionate about that. They also went through the same set of steps. So once, it's kind of finding a kindred spirit in a sense, that they connect, they click, and whether that's through an email or through a conference or anything like that. So read and connect with people. That would be the two take-home messages I would say.
Thank you for your helpful advices. On behalf of myself and all our participants, I would like to thank you for spending your valuable time transferring your knowledge to us and for this informative presentation.
Thank you so much. It was a pleasure.
I wish you healthy days.
Thank you.
Değerli katılımcılarımız, Bioinforange platformu olarak düzenlediğimiz Bioinfocongress IV etkinliğimizde çalışmalarına yurt dışına dönen değerli bilim insanlarının katılımıyla toplamda 6 seminer gerçekleştirilmiştir. Bu kapsamda 12 etkinliğimizin 6 tanesine katılım sağlayan izleyicilerimize İngilizce katılım belgesi verilecektir. İçeriklerimizi, duyurularımızı sosyal medya adreslerinden takip edebilirsiniz. Hepinize katılımınız için çok teşekkür ederiz. Sağlıklı günler dileriz. Yarın saat 14'te microRNAs as biomarkers in cancer, opportunities and challenges konulu konuşmasını gerçekleştirmek üzere Sayın Doçent Doktor Pınar Uysal Ongener hocamız bizlerle birlikte olacaktır. Teşekkür ederiz.